Okay, that was quick. Thank you. I'd like to welcome everybody with us this morning in person and on our webcast to our public workshop, Implementation of Signal Detection Capabilities in the Sentinel System, which is being hosted today by the Duke-Margolis Center for Health Policy and supported by a cooperative agreement with the FDA. I'm Greg Daniel, Deputy Center Director in the Duke-Margolis Center for Health Policy. And today's discussion builds upon the foundation that FDA has laid over the last 10 years to improve the speed and efficiency of medical product safety surveillance. Launched in 2008 as a pilot program to assess known safety signals potentially associated with a drug, today the Sentinel system is considered a fully operational and core component of FDA's safety surveillance capabilities. Sentinel, as many of you know, uses a distributed data network that contains curated electronic health data covering more than 100 million people captured through routine care delivery. These data have informed many regulatory decisions made by the FDA and in some cases have even eliminated the need for post-marketing studies, which can require years to develop and are often very costly. Given these successes and growing experience with Sentinel's data infrastructure and innovative and evolving statistical approaches that we'll hear about today, the agency intends to further strengthen its core capabilities by exploring these new approaches for identifying potential signals of unexpected serious risk. It's important to note that signal identification, which you may also hear speakers today referring to signal detection, signal generation, all of these, we're going to use the term signal identification hopefully throughout the day, but they're all synonymous terms. These would not replace but serve as an important complement to existing passive surveillance systems and tools at FDA like the FDA Adverse Event Reporting System, or FAERS. Today's goal is to explore important statistical and operational considerations for implementing signal identification in Sentinel, as well as potential techniques that could be leveraged within Sentinel's distributed data environment. We're looking forward to having a robust discussion on these key issues throughout the day, and thank you for your interest in attending today. Before we get started, I do have a few housekeeping and items that I'd like to cover, and I'd like to briefly go over today's agenda. We'll start with an opening presentation by Michael Wen and Monica Munoz, who will present the agency's initial thinking on signal identification in the Sentinel system and overview key topics where stakeholder feedback would be really helpful and important to help inform the agency's strategic planning efforts. This will lead us into our first panel discussion starting at 9:45 a.m. to consider important statistical considerations for implementing signal identification. So general things like reuse of data, hypothesis generating and hypothesis testing within the same environment that bring up lots of challenges and concerns and ideas around statistical issues. This will take us to our first break at 11 a.m. and we'll return for a panel session to discuss important opportunities to support effective communication of signal identification information between the FDA and key stakeholders. We'll break for lunch. Coming back after our afternoon break, we'll take a deep dive into the potential techniques, the actual approaches that are being considered and that many in the room are working on. The limitations and readiness for implementing such methods within the distributed environment. After the lunch, we're going to span that deep dive across two separate sessions. One session is stocked full of presentations of potential techniques that could be used. After that session, we have a panel of stakeholders that will weigh in on what they heard in those presentations, maybe bring up other ideas. But across those two sessions, we're hoping to have a really good deep dive into these potential approaches, how they can be used, and how they might be implemented. So before we begin, I'd like to just go over a few additional housekeeping items before I hand it over to our opening presentations. We'll have time in each session, as you'll see in the agenda, for discussion with the audience. We have microphones up front, and we plan in each session to open it up to the audience for Q&A. Wi-Fi information is about to be on the screen. There you go. The network is Meetings for Hyatt. There's no password. In addition, I want to remind everybody that this is a public event, so that that is also being webcast online. We have many viewers who have dialed in online with us today. The recording of this webcast will be archived on the Duke Margolis website after this event. But as a heads up to speakers and panelists, while we do have lots of time for Q&A, we also have a lot of presentations. So we'll have Elizabeth, who's right here in front, that will be holding a time card up for our speakers to make sure that we keep on time. And then also feel free to help yourself if you're in the room to beverages that we have out front near the registration table. And then lastly, lunch will be on your own. There are a number of restaurants in this hotel and in the vicinity, and we also have maps of these restaurants at the registration desk. Finally, as a reminder, this meeting is being convened under a cooperative agreement with the FDA, but it is not a federal advisory committee or a full Part 15 hearing. So we will not be voting. We will not be trying to reach consensus. Today's discussion will be a success if we hear lots of ideas on how signal identification could be used and incorporated into the Sentinel system, and concerns, or also ways to overcome those concerns. With that, I'd like to move on to our first session and formally introduce our speakers. And I'll ask them to also join me up on stage right now. So first will be Michael Wen, is the FDA Sentinel Program Lead and Deputy Director of Regulatory Science Staff in the Office of Surveillance and Epidemiology at the Center for Drug Evaluation and Research. Following Michael's presentation, Monica Munoz is Deputy Director of the Division of Pharmacovigilance 1 in the Office of Surveillance and Epidemiology within FDA's Center for Drug Evaluation and Research. With that, I'll turn it over to Michael. Here's the...
All right. Good morning, everyone. It's great to see everyone here. I'm talking on behalf of a very, very large team, and I'll just say thank you for folks like Gerald Delpan, Bob Ball, Judy Zander, and Michael Blum for really supporting this meeting, making it happen, and sort of giving us that kick in the pants to say we should be doing this in Sentinel. I'll just say a note, Judy Zander specifically came to us from to FDA a while back, and one of the first things she said to me was, when are you going to start using Sentinel for signal detection? And I said, we're working on it. I just need a little bit more time. And that was about a year ago, and I'm just happy to say this is where we are now. So I'll be talking about signal identification in the Sentinel system, past, present, and future. So my plan for the talk, we're just going to talk about 4 things. We'll talk about the motivation for initiating signal detection in Sentinel. We'll talk about the approach to building a signal identification program in Sentinel, our launch of a pilot, an operational pilot in the coming year, and then establishing a Sentinel scientific community around this topic and around Sentinel in general. So without further ado, we'll start. But this can't work unless we obtain scientific input, and that's why we're having a public meeting. So this is a fundamental goal of the meeting. So it always starts with the Institute of Medicine, the Future of Drug Safety report. I just pulled out section 4.2, this recommendation here, because it always reminds me what this really brilliant group of investigators and researchers told us to do many years ago. 1, increase the intramural and extramural programs to access study data from large automated databases. I think we're there. Include these programs in drug use patterns and to find background incidence rates for adverse events of interest. I think we're there. And to develop active surveillance of specific drugs and diseases in a variety of settings. And that's where we're talking about today. So following, obviously, the Institute of Medicine report came FADA, the FDA Amendments Act of 2007, where Section 905 mandated that we create an active post-market risk identification and analysis system. And you can see right in that wording, risk identification, is signal detection or signal identification. And if you dive down into the text that Congress gave us a little bit farther, you see here in section CC that says, other data as the Secretary deems necessary to create a robust system to identify adverse events and potential drug safety signals. And that's where we feel is our federal mandate, our congressional mandate to do such, to do signal detection in Sentinel. This then went on in the story as we launched the Mini Sentinel pilot, and Melissa Robb and Janet Woodcock wrote in this paper defining our early goals, where the system created under the auspices of the Sentinel Initiative will help FDA identify and investigate post-market safety signals. And they echo the framework that was created in the IOM report for signal generation, signal refinement, and signal evaluation. And that framework continues on to this day, starting from the IOM report, taken up by our leadership at FDA, and passed on to us in the operating divisions. So that's where the motivation came from. Then we started developing and testing these programs. The first thing we had to do is we had to create the foundational methods. We started with evaluating these methods in simulated data so that we knew exactly how the methods would perform. We created a suite, a repertoire of different approaches, which I'll get into a little bit more. And then we knew, once we started generating these hypotheses, we needed an easy way to follow up alerts in a rapid fashion. And so we created the infrastructure to do so. So some of this stuff a lot of this stuff has been published. This is the simulated data analysis that Dr. Judy Moreau led, looking at statistical power of post-market safety data mining. We then developed the propensity score-based tree scan method, and then we set up the infrastructure for evaluating alerts. And this is all on the internet. Then we went out to test it with products. So we tested it both with shorter-term exposures and longer-term exposures because each posed different challenges for the method. And we tested on vaccines and antibiotics. We tested on statins, long-acting reversible contraceptives, and diabetes drugs. And you can see some of that stuff here. You can see the results from our Gardasil analysis and the analyses done on diabetes drugs online. So then we held a public training on this last year. This was the second day of a public meeting, where you can see here in the arrows below, you can see we specifically highlighted tree scan analyses. The tree scan programs are online here. The user's guide is online as well. So we've tried to make this as not only robust and tested, but also as publicly available as possible. And so then we're here. Now that we've gone that far, now we're here. And that's really to discuss the challenges of implementation. And there's two major challenges that we're going to talk about today. The first is the statistical considerations for signal identification. And you'll see here in the background a commentary on data reuse. Just to show that it's not just FDA that considers this an important topic, but other academics and researchers in the scientific community believe that this is an important topic to discuss. We'll also talk about how to communicate results from hypothesis-generating studies. It's inherently uncertain, and Terry Toigo is going to lead a great panel this afternoon on it. So the next steps: conduct a pilot for signal identification in the coming year, learn as we do it in action, grow and enhance the toolkit, and establish a best practices framework. So that's where we are. All right. So second topic: approach to building the signal identification program in Sentinel. These are at highest level the approaches that are out there in the field, and which ones are available in Sentinel. You could, for example, use a pre-specified panel of select outcomes. You can do one product and look at all outcomes. You can look at one specific outcome and scan all products going backwards. Or you can do an all-by-all analysis where you're doing all products and all outcomes. And each one of these has its own pros and cons and trade-offs. We've developed capabilities in the first 3 in Sentinel so far. And each one has a place in Sentinel. If you use the first one, this is really coming out of the Vaccine Safety Data Link. And they've had a lot of experience doing active surveillance there. They're prospective surveillance of a select panel of designated medical events. We could do the same thing using our level 3 tool. We also have tree scan and drug scan. There is no one best method, and this is why we've set out intentionally to create a broad repertoire, a broad toolkit. If you just look at our inferential analyses that we've done in the last 3 years as an example, you can see that 60% of the time we've used a propensity score design. About 40% of the time we've used a self-control design. So we knew right out front we needed more than one tool. And so you see here we started with a self-controlled version of tree scan. Then we built that to tree temporal, which basically combines a self-controlled with a temporal scan statistic so that you don't have to pre-specify a risk window. And then we built on to a propensity score method. And I'll just give a shout out to the Duke Margolis team. The discussion guide that they put together is probably the finest discussion guide I've seen for a public meeting. It explains each of these methods. The actual innovators of each approach wrote out an explanation of how these tools work in there in a really nice table. So if you need to figure that out, or if you need a cheat sheet on it, it's right there. The other thing is you can't do signal identification unless you have a foundation of high quality data. And this is where the Sentinel Operations Center has been a pioneer in this area. This just shows at a high level the 7 steps. I won't get into it in great detail, but 7 steps of really ensuring that the data are reviewed for quality control and are characterized and fit for purpose. It's, in the blue are steps done by the Operations Center. In purple are the steps done by the data partners. So it's what you see is a partnership between the data holders and the coordinating center to do this. I'll just focus a little bit on steps 4 and 5 here because this is really the meat of it. In step 4, you're looking for model compliance. You're taking data from, you're taking source data and translating it to the Sentinel Common Data Model, and you're applying level 1 and level 2 checks on it. It's over 900 checks done, and on average, you're finding 44 flags that have to be resolved prior to going on to the next step. So this is an important step in making sure that the data that FDA analyzes is checked and characterized beforehand. Once you check for model compliance, and that's just compliance, then you review and characterize in step 5. And this is where you have level 2, 3, and 4 checks, and this is an additional 500 checks, where on average you're getting about 10 flags that have to be resolved. And this is an iterative process. Every single time you extract, transform, and load the data, every time you refresh it every quarter, you're going through this key process. And both the Sentinel Operations Center and the data partner ultimately approve each ETL, and so that we can have confidence in the information that comes out of Sentinel. All right, so let's turn to the operational pilot then. How are we thinking right now about how to do this? So here's our proposed signal identification process. I chose an image of a funnel because a lot of information, a lot of judgment, a lot of choices go into the decision to launch signal identification. And the output, hopefully, is either no signals or a handful of signals. But obviously, it starts with a summary of the clinical trial data, the integrated safety summary at approval, and the prescribing information. And so then FDA will select one product, choose the study design or tool that we will use, whether it's self-controlled or propensity score or some other tool, or a combination of those tools, and then conduct an analysis. Then we will review and classify the statistical alerts. And I chose the word statistical alerts here specifically because that's what it really is. It's just an observed unusual frequency and has to be reviewed and undergo clinical correlation. And then we integrate those statistical results and review them in the setting of all the other information that we have. And from that, we come out with identifying outcomes for further evaluation, if there are any. And what's nice about this is that we're broadening the user base at FDA. We started Sentinel with the Division of Epidemiology, and now the divisions of pharmacovigilance will be taking this on with the Sentinel program team. And that's why we have Monica taking on the second presentation. And any signals that come out of this will still be evaluated by the divisions of epidemiology. So we are growing this, the program into FDA's operating divisions. All right, so how are we going to do that, right? So we are going to adapt the lessons learned to this new program. We didn't start from nowhere. We started from a decade of work with setting up the active risk identification and analysis system. So I just, at a high level, there's 4 dimensions here. There's the initiation or the trigger. When are you going to, or what's triggering you to do this? The timing, the process, and then the communication of the results. And so with ARIA, or the active risk identification and analysis system, the initiation was a serious safety concern. If you identified a serious safety concern, you would start ARIA. The timing was determined by the statistical power and study design considerations. And the process is now fully established. We have roles, templates, processes across all FDA divisions. And then the communication, we have existing templates, web pages that show not only what we're doing, ongoing analyses, the results, as well as the regulatory outcomes. And you can see that all online. It's growing month by month. And so we're going to take that foundation and translate it to signal detection. So the initiation is now by using a risk-based approach that Monica will talk about a lot more. And we'll have to determine at the time of licensure, is the system fit for purpose for the drug of interest? Sometimes the drug may not work with the methods or the data available. For example, if it's an orphan drug that's only used by 100 people in the US every year, that we may not be able to do this, and FAERS may be more than enough. If it's an oncology drug that's used inpatient on really, really sick folks, this may not be the right choice. All of these considerations come into play. We haven't decided those best practices yet, but all of these types of decisions come into play. The route of administration, the use, things, and the regimen and things like that. The timing. Here, unlike it's driven by statistical power and study designs, the timing is really a balance between timeliness of signal detection with statistical power. If you do it too early, you're not going to be able to detect rare events. If you do it too late, some folks would argue that you've done it too late to make a meaningful intervention. So this is where we're going to have to figure out what the right balance is. And the process is we'll develop and pilot these roles, these templates, but we're going to build upon the existing ARIA process. So we're not starting from nowhere. And the same thing with the communications. And Terry's presentation is going to go deeply into this. So, all right. So last thing, establishing a scientific community. We're starting to invite new investigators on the Sentinel projects. Here you see us doing an evaluation of TreeScan with sequence symmetry analysis and information component temporal pattern discovery. So we partnered with Nicholas Noren, who will be talking today, and Jesper Hallas, just to show that we're thinking broadly about this because there is not one best method. We're also continuing our public engagement through the posting of study protocols. So this is a protocol to develop a global propensity score for data mining and tree-based scan statistics. So in a regular propensity score analysis, it's a specific outcome. You know what you're going to do with that propensity score model. But now you're looking at 8,000 outcomes, so what is this universal global propensity score that you're going to develop? And what is it going to contain? That's what this is about. And you'll see here, we posted it for public comment, we got questions, we answered the comments publicly, and you can see that track record here in our response to public comments, and we will continue to do this. There's also a couple of planned future projects. That we're going to develop we're going to launch a project to develop sequential TreeScan to enable multiple looks over time. So as I said before, part of the problem with signal identification is, when are you going to do it? When are you going to do it? Here, if you know you can do it prospectively multiple times, that obviates some of the anxiety and the concerns with that. We'll also evaluate TreeScan for pregnancy outcomes now that we have a mother-infant linkage. Can we do a better job in the pregnancy population? These projects are coming. We're not going to stop. at Signal Identification, we've got a lot of other things going on to advance the Sentinel system, and these are just two recent meetings that were led by Bob Ball. One to improve the efficiency of outcome validation in Sentinel. The other is to advance the Sentinel system in general, and what you're seeing are three major themes coming out. One is that we are going to explore opportunities to leverage advances in machine learning, natural language processing, and artificial intelligence. We will also expand the Sentinel Common Data Model, and we'll enhance existing data sources, particularly with electronic health records. So we're advancing on multiple fronts in Sentinel that I think will provide an even better foundation for signal identification in general, as well as enhance our ARIA tools. So last slide. In summary, establishing a Sentinel signal identification program fulfills a congressional mandate and expands the utility of the system. Sentinel signal identification can build upon the prior experience using the Sentinel system, and it will be integrated into existing regulatory processes. So we already have a process for label change. We already have a process for track safety issues. We're going to leverage those processes. We're going to invent as little new as possible because we already have a strong foundation. The next steps include a pilot program and establishing durable processes and best practices, and we'll continue to publish these. And we will continue to engage the scientific community through meetings just like this, expand the analytic toolkit, and disseminate lessons learned. And I should also put a plug into our Sentinel annual meeting now in April. We moved it out because I kept worrying about snow. So we moved it out to April, and so hopefully there shouldn't be any snow in April. And FDA is interested in obtaining input from this meeting. So thank you very, very much for being here.
Thank you, Michael. So this morning I'll give you an overview of our current pharmacovigilance framework and our current data sources that we use and where this opportunity fits into our current framework to use signal identification with Sentinel. So signal management comprises of all the activities around a particular safety concern, from signal identification to signal refinement, signal evaluation, and taking a regulatory action to mitigate that risk. In signal identification, we may identify a safety concern through our ongoing surveillance of FDA's adverse event reporting system, the published literature, and from the sponsor or the applicant's periodic safety reports. But we may also become aware of a safety concern from other sources in our review of other data. This may include regulator exchanges, other sponsor submissions, clinical trials, and other inputs like inspections out in the field. So any piece of data may represent a signal concern, and safety concerns from any source move to the signal refinement stage. It's at this stage where we have a preliminary consideration of the potential causal association between the particular product and the safety concern. We consider the potential public health impact, and we look at all available quantitative data about the safety concern, including data beyond where that safety concern may have originated from. Those signals that represent important potential risks move to signal evaluation. Here we assemble a multidisciplinary team of clinicians, epidemiologists, statisticians, chemists, really anybody who can contribute to evaluating the safety issue, and we look at all the data that's available. And this, importantly, for today, may include an epi analysis. And so from that evidence, we move to regulatory action. These often include communication, labeling modifications, and other actions that may mitigate the risk. And so while today we're focused on signal identification, it's important to understand this context for some of the discussions we have today on methods, choices, reuse, and communicating safety information. So we can zoom back into signal identification and talk about our current practices from a Division of Pharmacovigilance perspective. We screen FAERS and publish literature on a weekly basis, and this includes reviewing individual reports and articles. And we look at disproportionality analysis to identify any patterns that may represent safety concerns. We also perform cumulative analyses of the same data and sponsors periodic safety reports, as well as other sources that may be pertinent to particular products. And we do this using a risk-based approach to identify which products and at what frequency. The risk-based approach includes things like how long has the product been on the market? Is the product particularly complex or manufactured complexly? Is the population exposed, a special population such as pediatrics, and what we knew at approval and from the clinical development program about both the risks and the benefits of the product. So when we think about our current signal sources in the post-marketing setting, there are a couple papers that inform this. The first one here is an analysis of FDA's drug safety communications between 2007 and 2009. What these authors did is they went back to the public postings of the drug safety communications and looked at the data summaries and pulled what evidence source informed that drug safety communication. And if they couldn't figure it out from the drug safety communication, they looked internally in our databases. And what they found was 50% of the drug safety communications were informed by FAERS data. And we use drug safety communications as a tool to communicate important emerging risks to both healthcare providers and patients. Similarly, in a follow-up article, these authors looked at all the safety-related labeling changes in 2010. This included about 400 different products that had new additions to their label. And they went to internal files and looked at where did the evidence come from to support this labeling change. And similarly, they found that 50% were informed by spontaneous reports, followed by clinical trials and PK studies, and I think observational studies was in fourth place. And it doesn't mean that these data sources were the only data source that supported that labeling change, but spontaneous reports were a key piece of the puzzle in a lot of these safety-related actions. And so FAERS, we know, is a valuable source of safety information. We use it for quite a lot. It's particularly good at detecting rare and acute events. Things like Stevens-Johnson syndrome anaphylaxis are kind of its wheelhouse. It captures all products and settings of use, so things like compounded products, over-the-counter products, and things that are obtained throughout the commercial and non-commercial settings of use. And so we can get insights from that that we may not be able to get from other data sources. And importantly, it also provides a patient perspective. FAERS contains reports from healthcare providers and anyone from the public who would like to send information to FAERS, and we facilitate that through the MedWatch program. But we know FAERS has important limitations, and I'm not going to belabor these, but ones to keep in mind are the unknown denominator, underreporting. This is a voluntary reporting system. Stimulated reporting, particularly after things get a lot of media attention, and variable information quality. And all of these things make it difficult to evaluate signals with long latency like malignancies, worsening disease, or high background rates. And these limitations preclude us quantifying the risks. And so when we think about those disadvantages and contrast it to Sentinel, we can think of some advantages that Sentinel has and why these systems would complement each other. So Sentinel, we know what the exposure denominator is. We have exposure and event capture that's not dependent on a voluntary process, although there are other issues such as coding. We're looking at longitudinal data, so we have a better idea of what that patient's medical history is and their trajectory after the event. And because of these facets, we're able to control for confounding using various methods. And so as we think about the current sources that we use, we're heavily reliant on passive data. And this, today, we're talking about the opportunity for active hypothesis-free signal identification in Sentinel. And then we think that these two together will be a good complement. And so as Michael mentioned, and you will hear again today, we intend on implementing signal identification with Sentinel as a pilot basis. All of our tools have strengths and weaknesses, and the pilot is going to teach us a little bit more about Sentinel's strengths and weaknesses for signal identification. Importantly, this doesn't mean that we're abandoning our current pharmacovigilance approaches and our data sources. but we intend to complement these data sources with Sentinel, and we're excited for the new opportunity to do that. So, thank you.
Okay, thanks for those great presentations. We're going to see, maybe we'll take a couple minutes for any quick questions for either of our presenters based on what they presented. Okay, I guess not. Okay, so you're off the hook. We'll move right into our first full session, which we have built in a lot of time for discussion with the audience. Again, thanks to our previous presenters for a great overview and better communication of how FDA is viewing this and what some of the plans might be. This next first full session we'll have a broader conversation on the key statistical concepts and considerations concerning systematic error and biases that need to be addressed as part of the FDA's regulatory framework. As Michael pointed out, there are two big major questions about implementing signal identification within Sentinel. One is, what are the key, regardless of the method, what are the key statistical considerations and issues? And then number two, how best to communicate those with the audiences. So both of those two major topics are the topics of the next two sessions. And then I'd like to further note that in the afternoon we're going to dig into then the specific techniques that are going to react and respond to addressing these statistical challenges that we'll be discussing in this very next session. So I'd like to go ahead and ask our panelists and presenters to join me up on stage. We'll be kicking off this discussion with a presentation from Mark Levinson, who's the director of the Division of Biometrics 7 in the Office of Biostatistics and the Office of Translational Sciences at CDER. Mark will give an overview of important statistical topics such as what I mentioned before, the reuse of data, so using the Sentinel systems data for signal identification, and then, how would you use that same data to test and confirm hypotheses. Mark will give an overview of those issues as well as the need to develop a regulatory framework for signal identification. Following Mark's presentation, we'll hear from a panel of reactors who will discuss their views on these specific issues, including Darren Toe, who's Associate Professor in the Department of Population Medicine at Harvard Medical School and Harvard Pilgrim Healthcare Institute, Jahari Jahari, Vice President and Head of Epidemiology and Benefit Risk at Sanofi, Mary Beth Ritchie, Director, Epidemiology, Medical Devices, and Real-World Evidence at RTI Health Solutions, and then Simone Pinheiro, Director of the Division of Epidemiology 1 in the Office of Surveillance and Epidemiology at CDER FDA. Mark? Okay.
Well, thank you, Greg, and good morning, everybody. So I'll be talking about statistical considerations for implementing signal identification, and though I'm presenting today, much of this work has grown out of an internal working group led by an employee at FDA, Rima Izam, and Michael Gwen and others were involved in this working group as well. Okay, so I have three goals for this presentation. I'm going to present what I consider a primary use case for how we're going to do signal identification, followed by signal refinement or evaluation. And both these exercises, the identification and the refinement and evaluation, will be done in Sentinel in this use case. And throughout this talk, there are distinctions between refinement and evaluation, as Monica pointed out in the last talk, but they won't necessarily be central to my presentation. So I'm going to group them together in my talk. In the discussion, some distinctions may come out. I'm going to clarify the objectives of this use case to make it clear what we're trying to do and what we're not trying to do, and I think that will be important, as you'll see. And also, we'll set the stage by introducing some concepts and terminology that hopefully the panel can make use of. Okay, so this is the basic process of this use case. So signal identification followed by a refinement evaluation exercise. So a drug is approved in year 0. At that stage, we decide how to integrate Sentinel into this risk-based pharmacovigilance program that Monica spoke of. As Michael and Monica said, not all drugs will be appropriate for use in Sentinel, but we expect many will be. So let's assume that a drug is appropriate for this exercise in Sentinel. So sometime later, when there's enough data, and we make them a little bit more rigorous as the talk goes on, there might be a signal identification exercise. So this is the one drug, many outcomes scenario that Michael spoke about. And then, based on the signals we see from the signal identification, as referred to as statistical signals, some of them are going to be followed up. Some of them are going to be obviously not causally related, but some of them will require further follow-up, and that will be sort of refinement or evaluation stage. And that may occur soon after the identification stage, or it may occur somewhat later, depending on the availability of data and the specific needs of the problem. So I'd like to provide some details to this that help flesh out this use case. So again, we're addressing a case where Sentinel is used for both the signal identification and the signal refinement evaluation. And we propose to use all or some of the same data in both the identification and refinement evaluation stage. There will be other approaches to this that perhaps the panel will discuss, but this is the use case I'm going to present. So this is this data reuse. We're going to be using data both at the identification stage and also at the refinement evaluation stage. And the point of this, well, certainly one of the motivations is to maximize the use of available data. I mean, Sentinel is a very large system, encompasses so much of the U.S. healthcare system. It's very rich. We like to make maximal use of the data. But the important point here is the objective of this second stage, the objective of the refinement or evaluation. So we're trying to strengthen the evidence for or against by reducing bias and confounding. It's important also to note what we're not trying to do. We're not trying to seek an independent replication or reproducing of the signal. That's not what we're trying to do. Again, this is going to be fleshed out more in the talk. But if you understand our objectives of why we're using the same data, or this may help calm some concerns. Okay, so this is typical bullseyes about bias and variation that we're all very familiar with. So I'm trying here to explain the point of the refinement evaluation stage. So the bottom left, I'm not sure, point to, yeah. So this is an unbiased precise estimate. This is a biased precise estimate, an unbiased imprecise, and the biased imprecise estimate. So in this refinement stage, we hope to remove some of the bias, and I'll be talking about the biases we specifically have in mind. So we hope to move from sort of the right-hand bullseyes to the left-hand bullseyes, from identification to refinement. So what are the typical biases, and what are the remedies for those biases? So these are basically, in some sense, mini pharmacoepi studies. So there's going to be differences in the prognostic factors between comparator groups or other confounders in the self-controlled case. So this is a confounding problem. The other problem is outcome ascertainment. We may not perfectly ascertain, well, we won't perfectly ascertain the outcome. We may even have differential outcome ascertainment. Now, it's important to note that the initial signal identification is trying to account for some of this. It's trying to account for some confounding, and it's trying to use the best outcomes available. But it's doing this for many, many outcomes, and the confounding control or the outcome ascertainment is not tuned to the specific outcome. So these are general sort of confounders that might be relevant for this drug for a range of outcomes. And the outcomes have to be relatively simply defined in order to evaluate many of them. So coming out of the identification stage, we still expect some biases. So the fixes for those is to now specifically kind of tune in on the specific outcomes that we're interested in. So we'll do more careful consideration of confounding and adjustment. Like, what are the confounders for this outcome that are relevant? We may also realize at that stage that we don't have the necessary confounders, but at least we have a better understanding of the confounding. And then we could do more specific outcome definitions, or even in some cases chart reviews to get better outcome ascertainment. Other things that also in the pharmacoepi world we might narrow the analysis population. Okay, so now let's get back to this data reuse, which is really kind of the big topic here. Is, we're going to be using Sentinel both for the identification and also the refinement evaluation. This is reusing data. This is a concern. So what are these concerns? Well, the left-hand side are things that people worry about nowadays in science about how to do an integrity and competence. Like fabricating data, using inappropriate methods, not documenting things well. These are certainly worrisome, but it's not really necessarily a statistical concern. So we're not going to discuss that, at least I'm not going to discuss that in my talk now. But what we are concerned about are more of the statistical aspects of data reuse. So that's analyzing data in enough different ways to observe an association, selectively focusing on those analyses that produce statistically significant results, and not properly accounting for multiple statistical testing. And there's variations of all of these as well. So these are the concerns that we're going to try to address. And there's two broad ways we're going to try to address this. One is through pre-specification and transparency, both of the plan and the results. And this will be somewhat talked about further today in another session, but it's an important part of the remedies for concerns about data reuse. The other concern is the other remedy is proper attention to statistical testing and errors, and that's what I'm going to focus on and drill down for the remainder of the talk. So let me introduce some statistical concepts that hopefully will also be useful to the panel as they make their remarks. These are these type 1 and type 2 errors that come up in statistics. Type 1 in this setting are what we would call false positives, concluding there's a signal when there isn't one. This is something we usually try to probabilistically bound with our alpha, maybe set at 0.01 or 0.05. We usually try to control this. The second type of error is the opposite, a false negative, concluding there's no signal when there is one. And this is related to statistical power. So what are we trying to accomplish by, what are the statistical considerations as we use the same data for signal identification and refinement and evaluation? So this big box it's supposed to represent a Venn diagram. So in the box are all possible outcomes that can come out of the signal identification phase. And because we're dealing with type 1 error now, we're going to assume there's no true signal. We would like to use, or at least I propose that we use at the first stage for signal identification, a procedure where we have some control of the type 1 error, or some understanding of the type 1 error. So coming out of that signal identification, we're in this blue box. There are some false positives, but we understand the probability of that happening because we understand the properties of the signal identification procedure. Now, within that blue box, we can refine and evaluate the outcomes we detected and still control type 1 error. This is not kind of a free lunch to do everything. There are still many ways of introducing additional type 1 error, but if we understand the type 1 error coming out of the identification stage, and we can continue to control for it in the evaluation, the refinement and evaluation stage, and be left with something at the end that has type 1 error control. So the other part of this is bias. What's happening with the bias? Here I'm not assuming that there's no signal, but there may be bias. So if the big box represents the total bias in the procedure if we did no control for confounding or we had biases associated with outcome ascertainment. We expect that the signal identification procedure does do a fair amount of control for these biases. But as I said earlier, it's made to address many outcomes at once. So it's not doing the best job for bias control for a particular outcome. But in the further refinement evaluation stage, where we're really focusing on a specific outcome or a few set of outcomes, we could reduce the bias further from what we get from the identification stage. So that again is the objective of this evaluation refinement after identification. So where does this leave us? So we've done signal identification and we've done refinement and evaluation. We've used the same data, or much of the same data, for both stages. So if we do this properly, we would propose that we have an understanding of the probability of false positives. And we've also reduced biases, or if we haven't reduced them all, at least we have understanding of the residual biases. So that's the end result we hope to achieve by this process. Just a few other considerations before I have my concluding slide that I hope will be useful in our discussion. So there's no right choice for type 1 and type 2 errors to be set. We may tune them to achieve specific sensitivity, finding true signals, or specificity, not finding false signals. Different cases may require different type 1 errors, type 1 and type 2 errors. In order to do this, we may have to delay analyses in order that we have sufficient data to have the necessary power. And finally, another consideration is, this is, at the end, we don't envision this is going to be a binary decision, like after the refinement or evaluation, we're going to say there is or is not a signal. We hope that we've reduced as much bias as we can. We hope we understand the type 1 error, but ultimately it's going to require further assessment of the signals. And that's going to involve the same sort of criteria that are important in every stage of the assessment: the biological plausibility, the magnitude and uncertainty of the effects, and what we understand about the residual confounding. So, in conclusion, we propose that we can do signal identification and signal refinement evaluation with the same data source, but only if our goal is to reduce bias and not to provide replication. And only if we have some understanding or control of the type 1 errors, type 1 and type 2 errors, both in the identification phase and the refinement evaluation stage. And pre-specification and transparency of the plans and results are prescribed. That's going to be important to avoid the appearance that we're just looking at everything and just reporting what we think is worth reporting. Okay, so that's my presentation. So I thank you for listening, and I look forward to the panel discussion.
While Darren's coming up for his presentation, I just would like to add my welcome to all of you. I'm Mark McClellan, and sorry not to be on at the beginning of this panel. You got started a couple of minutes early, but that's good.
All right, thank you, Mark. So my name is Darren Toh. So I would just be quick, since I don't have too many thoughts. So I think I will start with this slide. Mark sort of mentioned the data reuse issue. I think that I will be more specific. The issue is not about data reuse, it's about reuse of the same data source, because you can think of this most simplified version of a signal identification activity, which would just involve two data elements, the exposure and the outcome. If we are talking about data reuse, you might be arguing, are you using the same two data elements? I think the answer is no. We are mostly talking about whether it's appropriate to reuse the same data source that give rise to the signal or alert. So Mark sort of mentioned that the goal is to sort of reduce biases by analyzing additional data elements or information. So in this case, it will be confounding factors or factors that might be associated with misclassification or selection bias. And I personally thought that would be appropriate in many circumstances, because you are not re-analyzing the same data elements. You're actually introducing new data or information that will help you reduce the biases that arise from the same data sources. I think that it will be even more appropriate if the methods that you're using are what some people would call orthogonal, meaning that maybe in the signal identification activity, you analyze the data in one way, but during the signal evaluation or follow-up, you use another method that sort of query the data in a different way. But to me, as an epidemiologist, I think that the more important issue, if you move from signal identification activity to signal refinement or evaluation, is whether you are still talking about additional statistical tests during the follow-up period, or you are more interested in quantifying the risk and sort of understand more the uncertainty of the risk estimate. So that to me is more like effect estimation part of sort of a surveillance or research activity, and the consideration about multiple statistical tests could be different depending on how you look at the signal follow-up activity. So Mark also mentioned the balance between false positive and false negative. So from many perspectives, I think that it will make sense to try to have a system that will minimize false positive, because if not, then Sentinel will be sort of flooded by signals that the system had to follow up, and then they might not be able to sort of direct the resource to the more important issues. But on the other hand, if you have a very specific system, you might actually miss some actual signal. So I think that the challenge or the trade-off of balancing the two very important issues is something that is worth discussing. So I personally would be very interested in the thoughts of different stakeholders in which one would be more important for the different stakeholder groups. In terms of being transparent and explicit about the analysis, which is one of the remedies that Mark mentioned, I think that should be pretty easy to achieve in Sentinel just based on our 9 or 10 years of experience, because Sentinel uses a distributed data architecture, meaning that the data are actually not centralized. All the analysis require the queries to be pre-specified, because that's the only way for you to get the data or the output back. So I don't see a concern about not being explicit about the analysis, even when we move on to the signal detection activity. My final thought is whether FDA or the community in general is more interested in the safety of a drug compared to no use of the drug, or compared to use of another drug that is on the market. So it's more like a comparative safety question that I'm trying to pose, because most of the traditional signal detection methods have been built to look at the safety of using the drug versus not using the drug. I don't know whether this is something that FDA is also interested in when they are doing signal detection activity in Sentinel. It might be true for some circumstances, but for others, it might be more interested in comparing drug A versus drug B, for example. So I also have one final thought, but I forgot what it is. It must not have been important. So I probably will stop here. Thank you.
Okay. Thanks, Darren. Next is JJ.
Thank you, Mark. Darren helps me a lot because he mentioned what I have in mind, but let me opine first on type 1 error, type 2 error, false positive, false negative, sensitivity, specificity. It is related. And it's easy for us to say fine-tune. In the end, it's arbitrary. So here's my position. Sensitivity is very, very important, but given the quality of the Sentinel, or the superiority of Sentinel compared to, let's say, FAIRS, sensitivity will take care of itself. So to me, at least based on our pilot, it's less important. We did 2 pilots using 2 different claims database, and the sensitivity basically doubled. So the question is on specificity, how on false positive. How have you managed that without compromising sensitivity too much? And we could discuss the theory of statistics to death, but the bottom line is practical reasons. So my comment is more on how to deal this pragmatically. And I like the fact that the FDA planned to do this on the newly launched drugs, at least in the pilot, because that way you will really take care of the true signals to be detected, but also the false positive. It's easy for us to say false positive and true positive, but let's step back a little bit. The true and false thing is not easy to define. You need to first deal with the standards. And standard is not easy either. You want to deal with label. Yes, label may be a good thing, plus no signal in the system, but it's not easy. So I like the approach that the FDA is taking. Start with the newly launched drugs, maybe within 2 or 3 years. Start, instead of, Michael in the beginning mentioned, the final point, maybe multiple drugs, all drugs for all events. Maybe start with one single drug and see how it works. And again, in theory, you could play the threshold of sensitivity and specificity, but in practice, it's difficult. So start with that. Another one is on use and between, again, the point is between detection and refinement using the same database. Again, the issue maybe here is of course, we have to follow all the statistical principles. But the point that I want to raise is maybe practical. FDA Sentinel is not uniform, everything is the same. Maybe you should consider using splitting the data. And I believe Darren and colleagues have published this in the past on refinement, but maybe you could start with doing signal detection on, let's say, insurance health database and refinement using healthcare database, because there are different databases in the Sentinel. Another one is maybe splitting regardless of the types of data. You could split randomly. But the very fundamental practical question is, if you're talking about data that is newly launched, you may not have enough uptake to split. So that the question about orthogonality comes into play. Maybe you could do the same thing as long as the method is orthogonal. But then at the end of the day, I agree with Mark mentioned, and what Darren mentioned as well, that maybe it's okay as long as it's not the repetition. Because the point is, you will learn more on type 1, type 2 error. You will adjust for that. You will adjust for confounding better. You will minimize bias better by better measure of outcomes and exposure. Another point I would like to consider to discuss over here is the repeat of the analysis of the data used for signal detection itself. So before I mentioned detection and then refinement using the data source, and I mentioned about splitting based on different types, splitting randomly and so on and so forth. But when you market the data, as the drug is longer in the market, there may be different signals in different point in time. So I would like the FDA to consider doing signal detection using Sentinel not as a one single shot, but in different points in time. The argument may be, okay, you're repeating again. I don't think so. You know, the data will grow, maybe more components, more information, and it is important for us in the industry to know the signal later on that may need more induction period. Last but not least, I don't want to talk too long, is we have been discussing over here advanced, fantastic method, but it's all conventional. T-scan, propensity score, self-control case series. The one growing field that is currently in use is the artificial intelligence. The use of it that I have seen more now is not detecting signal directly, but artificial intelligence to change the data from unstructured, such as social media, text and everything, into a more structured. That's when you apply artificial intelligence as we speak now. And then once it is structured, you apply T-scan, propensity score, and so on and so forth. Why don't we consider applying this artificial intelligence as well even to a structured data. And I think this is a promising field. The key challenge is how to make sure we validate, make sure we find the truth, make sure we get what we want to get. I'll stop there.
Thanks very much, JJ. And next is Mary Beth.
So I think I'm going to stay with these same themes. We seem to hit some juxtapositions here where we have the false positives and the false negatives, and both are very important. And I really like Michael's funnel in his talk, because to me that's a really nice embodiment of this juxtaposition, where we want very, very few potential signals to come through at the end of detection, because we want to be using our resources well for the public health. But we want the real safety concerns to be part of that very small number as well. And so dealing with the false negatives there is really important, because if this is a true safety concern, then we want to know that as soon as possible all along the way. In addition, we have a juxtaposition of Sentinel and the vastness of data that we have, but we're wanting to look at new products. And so in this vastness of data, we're dealing with a really, really small numbers issue. And so we're trying to deal with using this data well. And so because of that, I would really think that for the detection piece, we would be wanting to use all of the data, because we have such sparse data as we are learning along the way. And we want to be able to identify safety signals quickly and efficiently. With that, for me, if we have a protocol or a specification laid out that has information on signal identification as well as refinement in it, if all of that is pre-specified, then it will feel much more like a more typical epidemiological study. And so it won't feel like we're parsing the data over and over. And so I think that that might help us there. In addition, we talked a bit about the measurement of the outcome and how that can cause bias. And it really can cause some difficulties, but there are other measurements that are part of this as well. And we have a little bit more control if we're looking at one drug over how we're measuring the exposure. To Darren's point, we have traditionally thought about measuring with and without a drug. It's a little bit of a different measurement if we're looking at one drug versus another drug. Our assumptions of a non-differential bias between a binary exposure leading to a bias toward the null goes away if we're looking at two different drugs. And so we can have systematic bias that is overwhelming any potential signal that might be there in that particular scenario. If we are looking at a novel drug and we are looking at time before and time after use of the drug, we may have very, very different situations, and we may have lost exchangeability. And so I think that there's some difficulties there that we may want to be considering as well. So with that, I think that one thing, in addition to what's already been said, that we may want to be considering is an absolute measure of particular outcomes that is a threshold level that we may want to consider a signal detected if we go above that absolute threshold. It may be something that we can build in with these other comparisons so that at the detection phase, we're really able to quickly focus on the most significant, most potentially concerning statistical considerations.
Thanks, Mary Beth. And next is Simone.
Hi, good morning. Simone Pinheiro, FDA. Thank you for having me. So I want to just step back a little bit and provide a broad overview of the current processes and data that we have for post-market surveillance, with focus on the signal evaluation, refinement evaluation piece of it. So talking about what we have now, which currently does not involve the signal identification abilities in Sentinel, and then talk about what I think it's going to add to what we currently have with some considerations around those. So as we all know, drugs, it's nearly impossible to know all the drug adverse events that are related to a drug once the drug is approved. So the FDA uses several approaches for drug surveillance to evaluate and monitor the safety of drugs after approval. So you've heard from Monica. So the goals of the surveillance are to assess unknown serious risks related to drug, assess signals of serious risks related to the use of the drug, and identify unexpected serious risks when available data indicate potential for serious risks. So a signal identification falls in the first goal, and the two latter ones refer more to refinement and evaluation of the signal. So you heard from Monica. So how do our signals generated? And you heard from Monica Munoz in her presentation about the data sources that we currently have for identify signals, spontaneous reports submitted to fairs, periodic reports, published studies, and so on. And how does the FDA determine that a signal needs further or warrants further evaluation? And these are very complex discussions that involve many disciplines, clinicians, epidemiologists, biostatisticians, safety evaluators, and so on, to determine whether a signal warrants further evaluation. Once the determination is made, what is the next step once the determination is made that a signal deserves further evaluation? Everyone in the audience probably knows, but I'll remind that since FIDA, Congress passed FIDA in 2007, the section that mandates that the FDA develops the risk identification and analysis Sentinel ARIA system is linked to the section that gives us authority to require post-marketing studies. So since 2016, where Sentinel ARIA has been fully implemented in the process in our framework, once the FDA determines that a signal deserves further evaluation, and that will be done with observational studies, we need to determine whether that signal can be addressed in Sentinel ARIA. And if it can, we'll work with our colleagues, with other stakeholders to conduct that evaluation. And if ARIA cannot address that safety question, we may be issuing a post-market requirement for that safety issue to be addressed in a study conducted by the drug companies. And then we'll work with them from that point on in terms of reviewing the protocols and providing feedback for the protocol after that. And how would a regulatory decision be made? What will impact regulatory decision making? At the end of the day, we would be considering the totality of the evidence. That would include not only finding from these efforts, but also everything else around that drug safety issue. So that would influence the regulatory decision, if it will be made, and which one, what regulatory decision will be taken, and so forth. Now, how do I think that a signal identification abilities in Sentinel would add to that process? I think it would augment the currently robust system they already have in place. I think it potentially may allow us to more efficiently identify unknown signals with some considerations. I think that we need to use, and we will, implement a thoughtful use of these signal identification capabilities. So at the end of the day, the Sentinel ARIA system uses repurposed data. So there's some scenarios in which it may make sense to use it for signal identification. Some others it may not, and Michael mentioned some of them. Perhaps the study population is not ideal, or the exposure cannot be adequately observed in the system. So all these things will have to be taken into consideration, fitness for purpose. I talked about the potential more efficient identification of signals, and of course, speed and efficiency may come, but we'll still need to wait for accrual of data. So I don't know exactly what that will mean, but however long it will take for the data to accrue for us to be able to use the system to identify the signals. And just reiterating a little bit what Mark brought up in his presentation is that what are we talking about in terms of reusing the same data source for signal refinement? I think here we're talking about really the goal of it is reusing the Sentinel data source to help better understand the hypothesis, so helping remove and decrease biases at that point. And how would these efforts, these refining efforts would accomplish this? perhaps by using more targeted adjustment for confounders, once we have a particular explosion outcome pair, we may be able to employ more targeted confounding adjustment. Perhaps you may narrow or better define the study population in these subsequent analyses. Employ more specific identification of outcomes, perhaps that may include more specific algorithms or even validation of outcomes may be needed. These analyses may include, probably will include, a lot of sensitivity analysis, subgroup analysis to help us better understand the findings. But at the end of the day, these are not considered, from my perspective, an independent confirmation of results. Yeah, but this is not the goal of the refinement. And just to close, my closing thought is that these analyses would be conducted using the same scientific rigor that we employ for refinement evaluation analysis. That would include pre-specification of analysis before we move forward with them. Thank you.
Thanks very much, Simone, and I appreciate the emphasis on scientific rigor in this important new expansion of Sentinel capabilities and activity. And maybe we could start off by having a little bit more discussion about the issue that came up in Mark's talk and that many of you picked up on, related to data reuse, or as Darren may have more correctly put it, data source reuse, since there is a sequential process here getting to more sophisticated analysis. And I get Mark's point about why this is not, the goal here is not to seek independent replication. But some of you raised some questions about whether there might be better ways to approach this, and I'd like to have a little bit more discussion on that. These include steps like having the more refinement stage include a broader range of data elements and maybe even additional data sources. An idea that's used often is splitting samples if there's sufficient power available, and then emphasis from Mark on through on pre-specification, but especially as these methods are being developed, maybe some very high level pre-specification might be difficult. So those are some issues that have come up, and I appreciate any further thoughts that you all have after hearing this discussion about how to address this general topic of data source reuse as part of implementing this program. Anybody want to start? Darren, I remember we discussed some of these issues at meetings years ago.
Yes, and actually,
on you first.
correct. I actually had to miss that meeting. So I think that the idea of splitting the data is so intuitive that it must be a good idea. But then when you take a closer look, like what JJ said, if we are going to focus on new medical products, you might actually not have the sample size that you need. So actually, you might not actually achieve the statistical power that you need to do the signal detection activity. I was involved in a work group that was affectionately called the Data Reuse Work Group many years ago in the Mini Sentinel pilot. One of the members of the group brought to the attention of the entire group a paper called "Don't Split Your Data." That is exactly the title of the paper. And they use it more like a Bayesian framework to argue against splitting the data, both on the false positive and false negative side. So again, I'm not a statistician, so I don't fully appreciate the paper, but based on the Bayesian framework, they argue that it is never a good idea to split the data. I actually don't know whether this issue is settled or not, but there apparently there's a school of thought that they sort of argue against splitting data in terms of doing this type of work that we are talking about today. But if you ask me, if we do have the sample size, it still makes intuitive sense to me to maybe consider splitting the data. So I'm torn in this area, and the experts in the room or on webcast might know more about this than I do.
I have something, Mark, on
Yes.
that. So suppose in the situation when we have no choice, this is a new drug, rarely used, we don't have enough for anything. So forget about splitting. Now the question is, can we still do, this is before we go into what Mark mentioned. So it's different. You know, in refinement, you deal with things better, in a better way. But there's another way, another approach to doing it, maybe on the safer side, is, again, don't neglect the practicality on this is not simple. When you try to detect signal, which means you try to see if there's some pattern out of thousands of drug event pairs, even when you have only one drug. So maybe for detection, you will use certain methods that will allow for speed without sacrificing the quality. And then in the refinement, you do it more deliberately. Maybe you use a different design, different method that will allow for a better confounding control. That's one thought that comes to mind. So in other words, you don't have to use the same exact design methods between detection and refinement, even if you don't split, even if you use exactly the same data source.
Any other thoughts on this topic? And maybe push a little bit on this pre-specification issue. And I get the point about wanting to have the hypotheses fully defined in advance. It does seem like, given what you all just said, if you're really trying to get it right, to get to that small corner in the box that Mark put up on his slide, there may be some drug or class-specific issues that, as you really start looking into the topic, get more and better specified as the work is going on. It seems like it may not be possible to do sort of full pre-specification, especially at some of the early stages in this work. Thoughts on that?
Well, I think that it's very difficult to pre-specify every potential outcome in this work, but if we're looking at the drug outcome pairs and we want to pre-specify the drug well, then that does help us, especially in the detection phase. I mean, we like to think a drug is a drug is a drug, but in reality, we have different dosing, we have different modes of administration, we have lots of different things that could happen with any particular drug that is on the market, just in how that drug is given to patients. And then within the data sources, we have different ways that we may be finding that information. And we can pre-specify all of that. And so I think if we pre-specify that early in the detection phase, then that suits us well to be able to say, this is how we're defining it. And we may need at the refinement stage to be tweaking that a little bit. But I think for detection, that does help us. We remove some of the bias there.
I mean, it does strike me with Sentinel data being what it is and getting better, and computing power being what it is and getting better. It's not going to be that hard to do at least what you're calling sort of stage 1, 2 early analyses on pretty much every drug and outcome combination. It's how much different the work, how much additional work will be needed beyond that to get to confidence that you're really getting to that on-target, accurately estimated step. And that may be a reason for focusing on certain disease areas or certain drugs. Does that seem like how this is likely to play out?
I would assume we don't want to do everything up front. And so I think starting with a disease area, starting with a particular set of drugs makes the most sense. I think starting with areas where we know there's going to be lots of new drugs is a nice place to start. But then also starting with if we have seen in the past a particular concern in a class, the new drug that's going into that class, go ahead and test all of that out now in the detection phase.
And developing those additional steps at the level of, again, as we're thinking about a pilot phase, individual drugs, drug classes, manufacturers that volunteer. How would you think about the trade-offs in focusing in on a specific drug or a drug class? What are the considerations that are likely to lead to good places to start this effort?
Maybe I could start, Mark. So the question is, I think one specific drug is a good place to begin with. But if the question is which drugs you want to start with, I think that's a different story. But the benefit of having one drug in the pilot instead of all drugs within the system is it will be easier to handle the positives, to follow up and to differentiate the true positive and the false positive, and we will understand the nature better. So moving on, so it is more on the volume. Yes, technology is moving so fast. Even in the future, the differentiation between detection and refinement might be getting blurred, more and more blurred in terms of speed and in terms of data quality. But to answer your question, I think having one specific drug of your choosing will enable you to really refine the tool itself. Because at the end of the day, if you have too many drugs, you will have so many positives. Again, we don't know if it is true or false. It will take time for you to understand the method.
I would disagree slightly with that. I think one class is probably better. For one drug, it could be very, very difficult to understand what you're seeing without the context of the rest of the class. Choosing a class where there is still an active pipeline would be appropriate. Choosing a class where there's already something on the market would be appropriate, just so we have enough context of what's going on in the clinical world to be able to understand what we're seeing.
Now, the factors that go into that choice, whether it's drug or class or something else, obviously are going to include things like statistical power and the ability to reliably identify an effect, as we've been talking about in the data source reuse context. I'm going to turn to a little bit more specific focus on considerations around addressing confounding and bias and how that might go into the initial choices here. So we talked about how many of the methods that described, I guess what JJ described as sort of the usual ones, tree scan, propensity scores, and maybe some additional insights coming from AI and assisting and making data sets more comparable or complete or other ways. Any additional comments that relate to this very important issue of starting in areas where confounding and bias can reasonably be addressed through these methods, or sort of the best places to start with those kinds of considerations in mind?
So I can probably start. I'm slow in taking up new technology because I still think tree scan is a very cutting edge method.
Just because it's established doesn't mean it's bad.
So as Michael mentioned, so now we have a propensity score based tree scan approach, which is trying to handle confounding and other factors at the signal identification stage. And my understanding is that the propensity score model is more generic than typical pharmacoepi study. They are using some sort of genetic metrics to address sort of higher level confounding factors. But then if you sort of think along the line of addition and advancement, without thinking about the data source reuse issue, you can foresee a day where the propensity score based tree scan will be as robust as a typical pharmacoepi study. And then, as JJ mentioned, then the line between signal identification and signal refinement might become blurred and blurred. I don't know whether that's a good thing or bad thing, but I think that is just a natural path of development. I do think that one of the strongest ways to account for confounding is to identify an appropriate comparison group. So as Mary Beth mentioned, like comparing drug A versus drug B, especially the drug B is used for similar indications, will really help with minimizing biases. But then you are answering a different safety question. So there are trade-offs between answering the right question and selecting the appropriate control group. But if you are just talking about reducing biases, I think that at the design stage, you can do a lot to remove or minimize a lot of confounding factors. And then for the others, you just use some statistical approach to account for other biases. But my personal thought is that design would really help you a lot.
I was just, Simone again. I was just going to add that in addition to the sophisticated tools for the signal identification methods, we also have the refining. We have the opportunity to decrease and minimize bias at the refinement stage with further methods beyond the signal identification to better understand the hypothesis.
My take, Mark, on that is, if I understand your question correctly, is how we take care of confounding issues, biases. Again, the technology and the methods become so sophisticated that it's not a matter of which methods you choose to address the confounding factors. Because let's say you know that 20 confounding factors you need to take into account. You could handle that using different methods. But the point is, what will make a difference is the quality of the data itself, of the depth and the breadth of the data, the completeness of the data. Now, many data sources right now doesn't have BMI. Maybe in the future it will be part of it. And maybe the quality of BMI now is not as good, but in the future it will be perfect. So to me, the difference makers is in the completeness and the quality of the data, whether it be the main exposure and outcomes and confounding factors, more than the methods and the technology itself, because it will catch up very quickly.
So if I just can quickly add as well, so not only the data, but also a very competent team behind it, evaluating and interpreting the findings of either of these methods.
I want to come back to that issue too, but kind of continuing down this line of focusing on methods and signals that will get to reliable and important results. Several of you, I think, alluded to the importance of a threshold or a level of certainty or concern around a risk that would trigger kind of the further steps and action here. And Mary Beth, you talked about, just like in Mark's diagram, there are only a few, hopefully very meaningful signals that are coming through this extended process. Does anyone want to talk a little bit more about what those thresholds might look like, what kind of considerations they would put into place, especially at the early pilot stage of this effort?
Sure, and I would actually tie that back to Darren's comment about the hypothesis. So, the big goal of all of this is to find the public health concerns and remove them. And so once we start pulling that back down, well, how do we, in the signal detection phase, how do we find those, statistical somethings that may be potential safety concerns down the road? And so we could have a hypothesis that is based on is what we're seeing for an outcome for drug A higher than without drug A. We could also have a hypothesis is with drug A higher than with drug B, or we could have a hypothesis that it's higher than some pre-specified threshold. And so all of those are slightly different questions that could kind of lead back up to we've detected something that's worth looking into. All three of those questions need to be handled slightly differently. So when we start doing that, talking about a threshold is very different in each of those situations. For that third one, where we have this like pre-specified, because Sentinel has already, and many other programs at FDA have already been looking into background rates for adverse events. We have some information there on what might be appropriate to utilize, given what's already being utilized in the divisions of pharmacovigilance. And so I think there we're good. I think because of what we have with refinement and the signal refinement in Sentinel, we can utilize somewhat similar thresholds in the detection phase to figure out what to move forward. Though I know that resources are scarce, and as we're trying to figure out how to balance what's coming out of that funnel to be looked at further, I would advocate for some sort of severity threshold to be applied to it. An outcome that happens more than anticipated, that is a very severe, very harmful outcome, I would weight that more heavily to look at more quickly.
Any other thoughts? See some nodding in agreement with Mary Beth.
Well, I agree with Mary Beth in terms of taking seriousness of the event into account in defining the threshold, but we could also add more component, and I think Michael mentioned about statistical signals into it, and which means could be not only the significance, or not everybody likes significance, but it means something, as well as the magnitude of the association, hazard ratio of this or ratio of that. So the combination between seriousness. The key as well is I don't think we should be, especially in the pilot process, we should be fixated into one set of criteria and threshold. That's the point. We need to develop and to see what is the most suitable based on the objective. Is it really you want to be something ultra sensitive or something that you want to compromise? The thing is, well, the best philosopher in the world, Mike Tyson, said, everybody got a plan until you got punched in the face. So, well, you feel very, very comfortable with the series of criteria and thresholds until it doesn't work. So, plan accordingly, be inclusive in the criteria, and then have a very clear method. Do you want to handle sensitivity or false positive?
Yeah, back to my concern, it may be hard to completely pre-specify everything in life, which I think was what Mike was trying to convey. Thank you all for already lining up. We were going next to comments and questions from those of you who are here on these important topics related to statistical considerations for implementing the Sentinel identification system. Signal identification system. So I'll start over here and alternate. Please just briefly introduce yourself when you ask your question or make your comment, and please try to keep it concise so we can get through all of these. Thank you.
Hi, can you hear me?
Yes.
Hi, I'm Michael Wynn. I just spoke.
Yes.
So this is good. So I stepped up intentionally to sample the hors d'oeuvres first, hoping that other people would step up to get food. One question that Darren, you brought up this issue of orthogonality, and I just want to double click on it a little bit, because this is going to be a two-step process where you have identification and refinement. Can the panel comment on what is the threshold for orthogonality? So there's some examples right here, right? So the clearest is a different study design. You start with self-controlled and you move to propensity score. I think that's orthogonal, but if you use a different comparator, or if you do a different outcome definition that's more specific, is that orthogonal enough?
Or a somewhat different data set, but not completely different.
It might not be based on some people's arguments. If you just switch the comparison group, but still use a core design. I think the purest way of doing something completely orthogonal is to use a completely different design in a different data source. But we are not talking about different data source. But there's a gradient, and I don't have an answer for you, because the hard thing about creating a threshold is if you ask 10 people, 12 people who have opinions. And so it's never easy. But I think that the end goal is coming back to what we've been talking about, trying to minimize biases. And any way that you can think of as a valid way to reduce bias, to me, is an appropriate option.
I think that raises sort of a larger point, that there's not going to be simple decision rules about whether a signal or not a signal. And I think you might be able to come up with some analyses that are very orthogonal, like looking at temporal patterns as opposed to comparative incident rates. But ultimately, I think you're going to be in situations like Darren described. There's going to be different degrees of orthogonality, and it's not going to be hard to quantify. So I think ultimately there's still going to be a lot of expert input to making sense of these analyses, and we can't fool ourselves to think we're ever going to get over that.
Diana?
National Center for Health Research. I wanted to ask you all to comment on an issue pertaining to confounding. Obviously, One of the great advantages of these huge data sets is that you can control for a lot of confounding variables. But sometimes confounding variables are important in terms of subgroup differences. So for example, age can be a confounding variable, BMI can be a confounding variable, having diabetes can be a confounding variable. All these things can affect outcome, but sometimes certain types of patients are going to have a worse outcome with a particular drug. So how do you deal with wanting to control for confounding, but at the same time look at subgroup analyses? And actually, let me just say that goes a little bit into that funneling idea, that maybe what we want to do is not just funnel how many signals there are, but whether signals are very specific to certain groups.
Comments?
So in terms of subgroups, we have the ability, depending on size, of course, look at subgroups in the data. Now, you'll never get around the fact that you'll need it doesn't matter how wonderful the data are, you still need the experts evaluating that. So there may be situations in the FR where there are certain confounders, important confounders, that are not captured in the data. And maybe in some of the situations, that particular data source may not be the best one to use. So you'll always need the expert judgment.
It does seem like one advantage of this whole approach is the potential for identifying subgroup effects that may be different around risks. So this is an important goal of the effort.
I have a little bit to add. I think that the two facets to that question. Number one is the subgroup analysis means that there is a potential differentiation in the role of a drug and event in different groups. And typically the reason to go into certain group is prior knowledge of that in trial, and others that we call it in epidemiology effect modification. The impact of drug in one group is different from the other. So the reason to go there typically is prior knowledge. But, and again, to what Mark said, given the enormity of the data, we're able to do that. Now, technically, it's not complicated. It's the best way to see if age truly modify the impact of drug A on an event is stratification. That's maybe the most straightforward.
Thank you.
Stephen Evans, London School of Hygiene, Tropical Medicine. I'm a panellist in the next session. Try not to say anything I'd say there. Number one, statistical considerations must be subservient to public health ones. The consequence of that has two things, in my view, in relation to what you've said. In relation to reuse of data, it is going to be surely far better to split the data 60-40, not randomly. Random splitting is nearly a total waste of time. Non-random splits, because the extra 40 is not gaining you anything like the power that you have in your 60%. That extra 40% doesn't get you 50% more power or anything like that. The second one is that you must begin with drug classes, as Mary Beth said. I think that focusing on individual ones has two consequences. One is it can cause switching to other drugs that have the same or an even worse problem because you don't know about it. All right? And it also means that the industry is happier. They squeal like crazy when their drug is picked on out of a class.
Thanks very much for the thoughtful comments. Any reactions?
Thanks, Steve.
So just about the splitting of the data, I don't think it's about 60 versus 40. I think that the comment was 100 versus 50 or 60. So the argument was that 100% of the data used to, in signal generation would definitely give you more power than any splitting. That's assuming that you are not doing the same analysis in the split data. So one will be used to generate a signal, the other will be used to confirm the signal. And then the concern or the argument was that it might not always be the best approach to use.
Thank you. Hector Isurieta, FDA. I remember years ago I was with Darren and with Mark in these debates about splitters and non-splitters, and as it happens often to me with age, I changed my mind. So I don't think we should split the data. Here's the reasoning. We have signal generation, what we call signal refinement, and what we call hypothesis confirmation. The third one does not exist. In observational data, observational studies, the problem is not really statistical, it's just bias. You know, whether we have unmeasured confounders, whether the groups are comparable, etc. So the third group does not exist. Everything we do is detect the signal, refine the signal. In order to refine the signal, everything you have explained is useful. You know, finding comparable groups, compare drugs that are comparable, etc., so you inform the decision maker. But I would argue that no study by itself, no observational single study, will reach conclusive results, unless it's randomized, and that's a completely different thing. So because that third step, the signal confirmation by one study alone, should not really exist, we as reviewers, we want to see the entirety of the data, the quality of the data, whether we have compared group, we use negative controls, did we detect, check for unmeasured confounders, whether we use, as we are in our group right now, using external databases, surveys to look for behavioral changes, health-seeking behavior, etc. I want to look at that data, and I don't want to split any data. I want to look at the entirety of the information. As you said, you know, the quality of the research group, the experience of that research group also has a weight. But bottom line, I don't think the debate is about splitting or non-splitting. We should not split. We should analyze and make decisions. But that's my opinion. I like to hear.
Thanks very much for the comment. I appreciate it.
Good morning. I'm Patrick Robert. I'm the head of safety for Pfizer. Well, one of the major confounding factors, and JJ alluded to that, is the lack of completeness. I would say I would call it, you know, the lack of medical information, of the poor medical quality of many of the cases that we are collecting today. Could be for spontaneous cases, or also I think even in the claim databases. So we are missing a lot of medical information. Understand that AI can turn unstructured data into structured data, but at the end it doesn't increase the medical information. So, and we all know that these poor quality cases also have described masking effect. So at the end, you know, they delayed the identification of safety signal. And we know that all the increase in case volume that we are seeing today, at least for us at Pfizer, is made of social media cases, and these social media cases contain no medical information whatsoever. So the quality is very poor. So my question is to the panel, you know, what is your recommendation for dealing, which I think is a very important confounding factor both for spontaneous reporting and for claim databases or in the distributed safety model. Thank you.
Good question. I think we only have time for a brief response to a very broad question right now. JJ,
well,
do you want to?
Since Patrick mentioned my name, so let me try this. But I think the solution to this is not necessarily, and I agree with you, the quality of the value created based on using AI and others will depend on the raw data itself. And if something critical is missing, then it's missing. So the solution to this is the best is to make sure we have a better data source. So hopefully, and it has a lot to do with also our public health approach here in the United States and the health practice as well. If people are required or people try and start to give as complete information and as accurate as possible, then the quality of data will increase. But that is a broader issues.
Well, we're certainly aiming to get better and better and more complete data. In the meantime, we're trying to deal as best we can with the important public health questions with the best possible methods and data use here. So really appreciate the discussion on how to do that. So
Hi, I'm Harry Seifert from GSK Vaccines. I have an ongoing concern about bias and confounding. Thank you. bias and confounding, especially when you started talking about looking at new drugs or new vaccines. And so my question is, I understand you could use propensity score to try to adjust for that. Do you really have, that's a terrible thing to say, how much confidence do you have that the propensity score really adequately captures or has enough covariate data to really adequately capture comorbidity?
Yeah.
And I think you can tell my bias from that question.
Well, any brief comments on this? I'm guessing the answer may be it depends.
Oh, the answer definitely depends. But I think that that's a nice way for us to utilize the entirety of the system. Because we have a detection phase, because we have a refinement phase, because we have a phase where we can do more studies, I think that we don't have to get it right in one particular moment. That as we learn more about what the confounders may be, we can include that. And so I think especially in this detection phase, having something that's more broad is useful, because then as we are moving toward, from this may be some statistical aberration to this may be a signal to this may be a safety concern, that we can refine both what we know of the exposure and the outcome and what we know of the confounders.
Yeah. I appreciate you bringing it up. It does seem like there's a lot of careful thought and attention going into this issue, and more will be needed as this effort continues. So we've got just a few minutes left. I want to get through these last three comments, please.
I'm Thomas Moore with the Institute for Safe Medication Practice. I wanted to just add a sort of a balancing comment about type 1 versus type 2 perils, to focus a little on the type 2 peril, which I think is our central problem with all adverse event detection. And it's twofold, which is one, to give you some context, I think it's certain whether, and I hope it's a drug class rather than a single drug, when you start looking, you aren't starting from ground zero as if these were going to be like flowers coming up in the garden. There are going to be clear signals from the NDA, this pooled safety analysis, and other places. And when you look at those data, I guarantee you are going to find that several labeled adverse events you don't see in this data, which leads me to the second part about type 2 error, which is if type 2 error possibilities are abused, and that is to say lead to the false conclusion that maybe something you thought was a drug adverse event now can be disregarded, it would be really a serious disservice. So I think that careful thought about not only the type 2 error possibilities here, but how the type 2 information is presented and used is a really important part. The second part on data reuse, I'm not going to go into the statistical part of this, but as a policy, I would recommend don't put it on like a straitjacket. As someone who's worked with a lot of observational data, you're going to get down to the point and say, you know what, to pursue this signal or that signal or the other signal is really not appropriate for this data. I would even suggest that you actually have a sort of conscious decision point in which you evaluate whether or not your findings, and it's the underlying data that are my big concern, whether your findings are actually, your underlying data sets just not suitable for pursuing those particular events.
Great. Thanks very much for the comments and the emphasis on the fit for purpose. Jesse?
Yeah, hi, Jesse Berlin from Johnson & Johnson. I've found over the years, I'm going to pick on Stephen Evans before I pick on everyone else. Over the years, I've found occasionally I have to help him translate from English. Now, I think the point he was making has to do with replication in different data sets rather than just random splitting. And to that point, I want to remind people that Sentinel is made up of many data partners. And you actually can generate, you do generate, by nature of the distributed process, you generate a data point, a summary estimate, and a confidence interval from each individual data source. But those are not generally reported. So how are we to understand the replicability, the variability within and between data sets if we're not going to get it reported?
Question, a brief comment on that, or just something to
yeah, so that means I have to wear the sensitive hat. So Jesse's a comment about like data source specific effect estimate. So that is available to FDA. It is not always published because we don't publish everything in peer-reviewed journal. But and then there is also has something to do with the agreement with data partners. So it's just but it is there.
Just the same answer I get every time I ask this question.
Yes. And if you look at the recent publications, you are starting to see more and more site-specific effect estimates because as the system mature, like data partner feel more comfortable presenting the data in that format.
Okay, well, thank you, because I just want to encourage more of that.
Yeah, of either the site-specific estimate reports or at least the magnitude of site-specific variation underlying the estimated results.
Can you just quickly add? I was just going to add to say that the FTA often consider that, and that's also very helpful in terms of helping us determine consistency of results across.
Just because it's not publicly reported doesn't mean it's not considered. Yes.
My name is Martin Kull of Harvard Medical School and Brigham Women's Hospital. Thank you for a very stimulating panel. I have a comment on orthogonality, but before that I want to say of the two uses of data reuse, the one of trying to evaluate potential biases and get rid of them by more correlated adjustment, I think is not only something that we can do, is something I think is absolutely critical that's done after we find a statistical signal. And the presentation by Mark, I think, was the best presentation I ever seen to make that case very thoroughly. So thank you very much for that. On orthogonality, it's related a little bit to statistical independence. And sometimes we can use the same data source to get a statistically independent analysis. So for example, what's the difference between orthogonality and statistical independence? Suppose we do a completely different study, different data, but the same design, we will have the same biases, but it will be statistically independent because there's completely new data. So the random errors are completely independent from the original studies. And to have that is a very good thing. But sometimes we can do that with exactly the same data source. I'm going to give you an example from what we did in the CDC vaccine safety data link. We had a signal of an excess febrile seizures after the MMRV vaccine. And we had evaluated the number of seizures 1 to 42 days after the vaccine and found that there were more seizures after the MMRV vaccine compared to the MMR vaccine. So how do we then evaluate that signal? Well, one thing we did is we say, okay, we condition now on the fact that we found this number of signals, 1 to 42, and then we see at what time do they occur after the vaccination. Because that information was not used in the original analysis to generate the signal. So now we said, okay, among all those who had the seizures, when did they happen 1 to 42? And we found that 7 to 10 days was a big peak in the seizures. So then with this kind of a cluster analysis to see that there was a cluster peak there that was statistically significant. And that analysis using the same data source is statistically independent of the first one. And that's sort of a very strong thing to sometimes do that. That's not something we can always do, but sometimes we can do that. And that's sort of a nice sort of way of doing signal evaluation or refinement using statistically independent analysis. So just an example.
No, I appreciate the example. Thank you very much. We are out of time for the panel. I don't know if there are any final thoughts that the group would like to leave with. I do want, as this process goes forward, to, a few of you raised the issue of resources necessary. These are not going to be easy questions to address, and it's important to develop a system that maintains a high level of confidence from across all of the stakeholders, many of whom you've heard from this morning. So hopefully we can come back to that topic as the day continues. Any final thoughts before we wrap up right now? No. Thank you all thanks for an excellent discussion. We're going to break until 11:15. We'll resume in about 12 minutes.
Thank you.
Way back to your seats. Thank you.
Are these? Yeah. Somebody else is yeah, he's changing these out. This one, I think,
That's mine. That's a fresh one, yeah. Okay, I'd like to welcome everyone back to our next session of the day. As we heard in the first session, a lot of discussion about the statistical challenges regarding the use of the same database to both identify and follow up on potential signals. Given this potential for a result of uncertainty, there's a critical need, as you've heard in that discussion and earlier, for the FDA to effectively communicate findings to stakeholders through processes that align with FDA's existing communication and transparency initiatives. Our next panel will discuss potential opportunities to improve the communication of signal identification information by building on FDA's foundation for stakeholder communication, while also aligning with the guiding principles of Sentinel, including privacy, security, and transparency. Now I'll go ahead and introduce our panel. Terry Toigo is the Associate Director for Drug Safety Operations in the Center for Drug Evaluation and Research at FDA. Stephen Evans, who I think will be hopefully joining us soon. I didn't realize he wasn't up here yet. Is a professor of pharmacoepidemiology. Here he comes. Stephen Evans is a professor of pharmacoepidemiology at the London School of Hygiene and Tropical Medicine. And then Joanne Waldstreicher is the Chief Medical Officer at Johnson & Johnson. Mary Frances Schubert is Vice President, Clinical Safety and Risk Management at Merck. And Diana Zuckerman is President of the National Center for Health Research. So I'll go ahead and hand it over to Terry to get us kicked off with a presentation on FDA's initial thinking on communicating this result uncertainty.
Thank you, Greg, and good morning, everyone. Let's see. The focus of my comments is communication and transparency, and I plan to cover 4 topics in the 12 minutes or so that I've got. I'll review some past drug safety transparency initiatives at FDA. I'll discuss how we're thinking about transparency and Sentinel signal identification. And then I'll discuss the Sentinel signal how Sentinel signal identification fits into our current regulatory processes for communicating about drug safety issues. And then finally, I'll talk a little bit about our plans for a pilot framework for communicating information from Sentinel signal identification. So FDA has long provided information on drug risks and benefits to healthcare professionals and patients. In the past, including the early MedWatch years, we provided that information when we were certain of its significance or it prompted a regulatory action, such as a labeling change. In 2005, FDA decided to make important drug safety information available earlier than we had in the past. But that meant there was a period of uncertainty while FDA and the drug sponsor evaluated new emerging safety information to determine whether the safety concern in fact related to the drug and whether the regulatory action or another action was appropriate. In 2007, we replaced the Drug Watch with the Index to Drug-Specific Information webpage. This page has evolved over the past 10 years. It doesn't include all FDA drugs. It only includes drugs that have been the subject of a drug safety communication. And when I took a look last week, there were about 400 drugs listed on this page. In 2008, we launched the what we call the 921 page, which required, it includes information that we were required to make public under Section 921 of FDAA. We post signals of serious risks or new safety information identified using the FAERS database, and I'll talk some more about this later in the presentation. This slide summarizes the Sentinel transparency initiatives, and I'll talk more about each of these in later slides. But as I was preparing these slides, I took a look at some of the history related to Sentinel and transparency because it's not something I work with every day. So in the 2010 Sentinel report, we said, and I quote, "The agency has a number of mechanisms in place for communicating drug safety information, and we will follow similarly transparent methods of publicly communicating relevant and timely safety information obtained through Sentinel evaluations." I think we've followed that plan. There's one thing I want to point out before leaving this slide, and that's there is a prominent disclaimer repeated on many of the Sentinel web pages. It's a long disclaimer, but the relevant point for this discussion is, and I quote, "The fact that FDA requests and receives data on a particular product through Sentinel does not necessarily mean there is a safety issue with the product." So as we work to integrate Sentinel signal identification into FDA pharmacovigilance activities, our transparency goals remain the same. We're committed to placing knowledge acquired from the Sentinel system in the public domain as soon as possible. And as we do this, we need to balance the desire for transparency with the preliminary nature of the results from signal identification studies. The statistical alert that Michael referred to represents the possibility of a new safety concern, not an actual new risk. It will require further study and clinical correlation. It may be later refuted, refined, or strengthened, dependent on further investigation. And when the further investigation is done, there may be regulatory action, or FDA may decide that no action is necessary based on available information. And before I leave the slide, I want to mention another goal that I forgot to include as a bullet, but I think it's important. And that's to mention our PDUFA 6 commitments related to enhancement and modernization of the FDA drug safety system. Those commitments included user fees to support the expansion of the Sentinel system and integration into FDA pharmacovigilance activities. And second, there is a commitment related to timely and effective evaluation and communication of our post-market safety findings related to human drugs. So meeting our PDUFA commitments is also an important goal of this initiative. As we think about our challenges to transparency, we consider what we want to achieve and what we want to avoid. And as I said, we're committed to placing knowledge acquired from the Sentinel system into the public domain as soon as possible. But there's always going to be some tension between the goal of having practitioners and consumers informed as early as possible about safety concerns and the potential for unintended consequences. We don't want to share premature information that might lead to drug discontinuation or drug avoidance with the consequent loss of a drug's beneficial effects. We don't want to inadvertently contribute to patient anxiety with uncertain information. We don't want clinicians to become desensitized to safety alerts and as a result ignore or fail to respond appropriately to such warnings because of alert fatigue. So finding the right balance, particularly with a new program like this, will be very important for all of us. And you've heard Michael and Monica, You've heard Michael, Monica, and Mark discuss how Sentinel signal identification will augment and not replace our existing systems. Our goal is to strengthen an already robust pharmacovigilance framework by building upon our prior experience with the Sentinel system. And just as my colleagues described how the activities will be integrated into our existing processes, so too will the communication with sponsors and the public be integrated into our existing operational framework. So how do we build upon the existing transparency initiatives in Sentinel? As Michael described, we adapt our lessons learned from ARIA to a new program of signal identification. This is a summary slide for how we plan to incorporate lessons learned. Information about the analysis tool itself, when FDA uses signal identification, and how we communicate about signal identification results and regulatory outcomes. And I'll talk a little bit about each of these in the next slides. The public development site is a cornerstone of FDA transparency. It includes analytic tools, analytic packages, and instructions on how to use everything. Previously, we shared study protocols. Now we share the analytic package used to execute the analysis with every parameter that FDA used. And this is the level of transparency we plan to have for signal identification. This graphic displays the total number of analyses conducted by FDA in Sentinel's ARIA system since its launch in 2016. As you have heard, there are several types of analyses, including summary tables, level 1 analyses, and level 2 analyses, and more technical information on these analyses can be found on the Sentinel website. Results from these analyses are posted on the assessment page as they become available during the evaluation process. And I'll show you a little bit more in the next slides. We will add signal identification to the existing use summaries as the information becomes available. You'll know when and how often we run a signal identification in Sentinel. Our communication with sponsors and the public will be integrated into our existing operational framework. Our experience with fairs helps inform our approach. The screenshot on the right shows how we meet the requirements of Section 921 of FDAA. We post potential signals of serious risks or new safety information identified during the fair, identified using the fair's database during the indicated quarter on the slide. So we post this 4 times a year. Appearance of a drug on this page means that FDA has identified a potential safety issue, but it does not mean that we have identified a causal relationship between the drug and the listed risk. If, after further evaluation, FDA determines that the drug is associated with the risk, we may take a variety of actions, including requiring labeling changes to the drug. And as you can see from this slide, in other cases, we may decide that no action is necessary at this time based on the information available, and we update the web page accordingly. Our plans for transparency related to signal identification in Sentinel are similar to what we have done with ARIA. In the screenshot on the right, we summarize how select analyses conducted in Sentinel's ARIA system have been used by FDA. Each ARIA analysis listed in the table contributed in some material way to inform an important regulatory discussion or an action. And Sentinel's ARIA system was one important source of the evidence considered. It's important to mention that the Sentinel data have informed many CDER regulatory decisions. In the past 2 years, we conducted Sentinel studies on 9 potential safety issues associated with 5 products in lieu of conducting a PMR. Such post-marketing studies typically require years to design and complete each at a cost of millions of dollars. This slide shows a mock-up of the pilot framework for communicating signal identification information. We would summarize how select signal identification analyses conducted in Sentinel's area system have been used by FDA. And as my FDA colleagues have discussed, CDER's Division of Pharmacovigilance will select products, choose study design or tools, conduct an analysis, and review and classify statistical alerts identified in the analysis. And just as they do with potential signals from FAERS, they will integrate the results with other sources of information and determine which statistical alerts will need to be further investigated. And as you can see from this slide, those alerts identified for further investigation would be posted on the Sentinel website, similar to what we do for FAERS. The expectation is that the results from the signal identification and analysis can contribute to the FDA regulatory process. And just like with the FAERS reviews of potential risks posted on the website, the further investigation can provide evidence that alleviates concern about a particular safety issue and might lead FDA to determine that no regulatory action is necessary based on the available information. So in summary, Sentinel is a new signal identification tool that will augment but not replace existing systems. And as we work to integrate the Sentinel signal identification into FDA's already robust pharmacovigilance activities, our transparency goals remain the same as they have always been with Sentinel: to place knowledge acquired from the Sentinel system in the public domain as soon as possible. We intend to integrate signal identification activities into our existing regulatory processes, and we need to balance the desire for transparency with the preliminary nature of the results from signal identification studies and post results in a meaningful but not misleading manner. So finding the right balance, particularly with a new program like this, will be very important for all of us. We'll learn from the pilot program and we'll look forward to your input. Thank you.
Thanks, Terry. We'll move to Stephen.
Thank you very much, Greg. Thank you for inviting me. I took the liberty of visiting Duke campus just before coming here and learnt about the benches that they burn, having visited UNC. Some of you can learn about that. Lots that you are doing, in my judgment, is great. Better in many instances than what we do in the EU, but not in every. I think we need to remember we're here for patients. Patients are the people we should be communicating with, as well as the people who we are serving. If we communicate clearly to patients, then the health professionals will also understand it, and the companies will as well. In terms of communicating uncertainty, our main object should be to reduce uncertainty. And I've got 9 things that I want to say about communicating. First of all, even if the primary analysis is done on a relative scale, odds or hazard or relative risk, then you must report absolute risks. And it's very important, in my view, even with signals, that absolute risks are reported. You should also report the absolute risks of benefit. You need to find a way of stating that. Somebody in the previous session mentioned benefits. We need to put these things in context. We need, I think, to have a more sophisticated, a more general approach to investigating the adverse events that you're studying, forgetting about drug effects. When you say that there is a signal for this adverse event, what is the epidemiology of it? And Sentinel should be able to give you that epidemiology. Break down by age and sex and what incidence rates are, and patterns over time. I think that I'm in favour of the grouping. The grouping you use with ICD-9 seems to be entirely sensible with its hierarchies for the tree scanning. But grouping, I tend to be a believer in Bayesian hierarchical models, but the tree-based things may work. One has to remember that ICD-9 codes, indeed all ICD, was planned for what? What was the purpose of ICD? No, death certification, right? And it was not designed for health things. It was designed for death certificates. That's the way it was originally designed. It's been modified to do it, and we need to be aware of the limitations of any of our coding systems. Fourthly, we need to look at the power of the randomized trials. That was mentioned in the slides from Michael a little earlier. And we need to see what possible effect in the adverse effect that we're talking about as a signal could have been excluded in the trials. What was the power in those trials? And we need to have some numbers. I believe in Kaplan-Meier curves for the exposure to drugs over time as part of the, should be in an EPAR or in a risk management plan, the actual numbers who are in the trials. The next thing we need to know is the time pattern. I would like to see for each signal the Kaplan-Meier cumulative hazard or Kaplan-Meier type curve for the drug that has the signal over time from beginning of treatment with a sensible timescale, and any comparator, either a non-use in your group or ideally other drugs. Confounding by indication does not necessarily mean that there's zero effect of this drug. So you need to be careful in future in just eliminating signals that you think are confounding by indication. You shouldn't, as I said a bit earlier, look at one product only, and I don't think you should look at one outcome. And as Jesse interpreted my incomprehensible English and thought, we need to have very clearly the variation in the rates of these adverse events across data sources. Thank you.
Okay, great. Thanks, Stephen. We'll move on to Joanne for some comments.
Thank you so much. Thank you so much. First, I want to thank both of you for very thoughtful presentations. And I really want to commend Sentinel FDA on this incredible work. I am really supportive of this next phase of Sentinel and proactive safety surveillance. By way of introduction, I'm the Chief Medical Officer of Johnson & Johnson across all three sectors, pharmaceuticals, which is called Janssen, medical devices, and consumer products. And my group is an internal group, but independent of all other groups, including commercial as well as R&D. And we consist of both safety as well as epidemiology and other policy efforts and efforts across the company. I really appreciate being invited here today to share my perspective on a communication approach specific to signal detection results, recognizing the potential for result uncertainty, both false positives as well as false negatives, all with an eye toward our shared goal of enhancing patient safety. I want to applaud the FDA for putting forward an approach for proactive detection and also for acknowledging other approaches, which will be discussed right after this session. Since we're so early in understanding the role of real-world data in signal detection, I think it's important at this stage to acknowledge that there may not be one perfect approach at this point. As we heard earlier, we're still in the learning phase of development of the right methodology or methodologies, or the right way to do these analyses. That's why I hope that moving forward, FDA Sentinel will take a very collaborative scientific approach involving various stakeholders and different methodologies so that we can all learn together rather than either a single approach or an approach that just includes one stakeholder, meaning FDA, in terms of the methodology and the actions. So instead of focusing on communication of statistical alerts, and I love that, those words rather than signals, it's statistical alerts. Instead of focusing on those alerts after signals are detected and vetted, which is very important, and I know that my colleagues up here will focus on that, I wanted to focus on what I think is most important, and that is collaborating together in the process and the methodology of these alerts or signal detection and interpretation, even before there's something to communicate. Without working together, one can envision that we will get into a cycle that I think everyone in the room recognizes, whether you're at a regulator or at a company or in academia. What will happen is that FDA Sentinel will start doing these really great analyses, great work, a lot of work on proactive signal detection and interpretation. And then what will happen is every single company in its own silo will also start doing the same thing, proactive signal detection and interpretation. We'll have a lot of new work using different methods, different data sources, different signals among the various stakeholders, many false positives, different false negatives, and many different interpretations, each with a level of uncertainty that, as we've discussed. Personally, I think there's great benefit for what I would say a more diverse and inclusive and collaborative process. Working together, we can avoid this situation and bring together the best experts, the best methods, the best data sources, including those internationally, and the best results for patients and healthcare providers. I try to think through the lens of what I know everyone here feels is our collective responsibility, working towards a more learning healthcare system, whereby we learn from all the data we capture and all the analyses we do to directly impact and inform clinical practice. And I also think that we should continually challenge ourselves and look at the impact after changes are made to clinical practice to see if the actions that we've taken have led to improvements in healthcare. I think that no single group will best accomplish our collective goals, and I think that we should develop some sort of trusted third-party scientific convening body, some way to work together to look at multiple methodologies. For example, we could consider launching a collaborative pilot that looks at, and at the risk of being accused of squealing, I believe also we should look at a class rather than a single drug. And then use each of the different methodologies to understand, is there a convergence of which signals are detected? Let's evaluate new signals, and also if we are detecting those signals we expect to see, say from clinical trials. Do we get confirmation with the different methods and the different databases, or just as importantly, do we get different answers from the different methods and different databases? And is that telling us something important? Finally, what are we seeing in other countries and other databases? Some of the scientists and databases in other countries are of very high quality and can contribute tremendously to this important endeavor. And many countries are now moving towards a common data model, the OMOP common data model we saw that was just announced a few days ago from IMI2. In addition, and this was mentioned before, touched on a little bit when there was a discussion of subgroups. Once a signal is detected and there's agreement in interpreting this signal that it's clinically meaningful, we could work together to do predictive modeling or subgroup analyses so we can identify those at the highest risk and the lowest risk. And then when we communicate to patients and healthcare providers, we have very meaningful messages to share about which patients are at the highest and lowest risks. And finally, after communicating, we can look together at the data again to see if the impact of the signal communication has led to meaningful outcomes for patients and public health. This is how I see this effort making a great contribution to enabling a learning healthcare system. Before I turn it over to my colleagues, I wanted to add one more thing. As we start to think about this new effort, I think we should also think about what we should stop doing. Through this new effort that FDA Sentinel is catalyzing, we can now envision having the capability of gaining great insights from real-world data, claims or electronic health record data, systematically and in a more timely way. While we're thinking of the great value we'll gain in better understanding post-marketing safety with this new approach, let's also think about data and work that is of very low value and asking if we can stop doing things which generate a lot of effort with little to no value. I was very interested before and really believe that in something that was mentioned by Simone and Mary Beth, and that is that we all want to use our resources together, use them well, and to answer important public health questions. What about the resources and work that we do that does not answer important public health questions? And what I'm talking about is potentially simplifying the collection and processing of certain post-marketing, mostly non-serious adverse events that contribute little to understanding of benefit and risk, especially those from programs like patient support programs. These activities require a lot of time, effort, and expense both at the regulators as well as the individual companies. You may be aware that Transcelerate recently did an analysis across 11 countries and over 1 million adverse events, individual cases from patient support programs, and we found, although it took a lot of effort, there was very little benefit for the reporting of, and analyzing of these adverse events. So just to wrap up, I really want to applaud FDA Sentinel for putting forward this fresh approach to proactive signal detection. I hope that FDA Sentinel will consider a diverse, inclusive, and collaborative process where we can put together the best minds, the best data sources, and a real diversity of approaches, as we'll see at this meeting, and also an approach that lets us frame our collective efforts in the context of a learning healthcare system, whereby we collaborate and test multiple methodologies, we learn together, we use predictive analytics and subgroup analyses when we do believe there's a signal to be able to define those at the highest and lowest risks so we can better communicate to patients and healthcare providers. And then we plan to re-look at the data after signals are communicated to see if our communication has had a positive impact on public health. Thank you.
Okay, thanks, thanks, Joanne. So we'll move to Mary Frances.
Thank you. I'm going to try and focus on some complimentary remarks because I do think a number of the other panelists have spoken on some important topics. I'm going to do it in two dimensions. One is in my current day job, and I lead, as my British colleagues, with a team of medics who are looking at all of the safety data for the products in our company. And so we're the end consumers of all of the data sources that are coming in, whether it's epidemiology data, clinical trial data, spontaneous data, adding in the Sentinel data in the signal identification space, and really trying to understand how to filter that. And as it relates to communication, you know, the stakeholder community for FDA's communication schema is much broader perhaps than people realize. It's a global stakeholder community. We have global regulations in many other markets where we as the sponsor need to proactively notify other agencies of alerts around our drugs. And so the cascade effect of every communication that FDA makes has a ripple effect around the world, and it triggers multiple simultaneous investigations. And to the points that were just made, they're independent variables and independent data sets. And so there is a cascade effect that the PRAC will start looking at something, Health Canada will look at something, the Singapore agency will start looking at their data. And so we are generating an accumulation of data, which I think is all in our benefit, but in a lack of coordination. And so I think when we talk about the resources applied downstream, I think it becomes multiplicative, you know. And so I do think those are things that, as a stakeholder community, we need to think about. I do think that, you know, secondly, putting on my healthcare provider hat as practicing physician for many years before joining industry, and the ability to understand the complexities and uncertainties that we're all struggling with here in the room as a group of reasonable experts in the theory. Most practicing physicians may not have the visibility into the uncertainties, and even having trained in a place where evidence-based medicine and literature reading and those types of things was very helpful. Okay, sorry. You know, there's still really the interpretation of what those communications mean and how do I apply it to my medical practice. FDA has clearly recognized some of the pitfalls, and that balance striking is really going to be important. So how do we engage some of that recipient audience of those communications to understand what they need in the communications to really understand the level of uncertainty? I think that's another piece of how do we craft communications that are meaningful, helpful, and understandable to the relevant target audiences. Because we all want the information, but we have to have it in a digestible manner. And I think that the areas there that we struggle with, especially when you get down to patient-level communications in an area that we want to try and continue to enhance, and I know FDA has been looking at how do we do risk communication in a number of dimensions, the health literacy of the target audiences we're reaching out to is another critical factor in the interpretation of these health communications, these safety communications. So I think figuring out how to engage more broadly the relevant stakeholders, not only in an impact assessment of what that communication may cause in the healthcare practice, but in the nature and how we communicate and actually the words we use around those communications so they're understandable. I think Stephen mentioned some of those aspects as well. It's going to be really critical to the success of the early alerts. In reacting to the comments in the first panel, when you looked at that diagram around the lead time of when you have a signal to when you actually may have an outcome, there's a long gap depending upon the uptake of the product, all those different variables that will let you do that more confirmatory analysis. How do you describe to the recipients of the communication what that lead time's going to be so they know up front not to be expecting an answer tomorrow? I think those are aspects of communication that we need to think about. So when I think about it, putting on my sponsor hat, putting on my healthcare provider hat, I think those are one of the important considerations. So I agree with as soon as possible, but it has to be given the robust context so that that information can be digested and not acted upon if the intent is to no action right now, we're still investigating. And I think that's where the consequences of those alerts without the appropriate context does result oftentimes in unintended consequences of lack of adherence, which is a healthcare public health problem to begin with, and you can accelerate lack of adherence by alert with a long lead time until we have an answer. So I think those are some of the things that we can partner together to try and work on is not just the frequency or the timing or the threshold of when we communicate, but actually the nature of the communication with appropriate context to make sure it's understandable for the relevant target audiences around the world and within the U.S. who are going to be actioning in their medical practice and in their patient guidance around that. And then I think, my last thought is really, kind of taking into account the burden on the healthcare system and going back to when I was first practicing and direct-to-consumer advertising came out and patients would come in with a long list of questions because of what they heard from the DTC ads. The FDA communications trigger that same kind of questioning. I heard it in the New York Times or the Philadelphia Inquirer about this safety issue that FDA is alerting about. What does that mean for me? And so I think as we look at our healthcare system, to be a learning healthcare system, we have to support those conversations between the healthcare provider and their patients by giving them robust information to be able to understand the nature of the communication. So those are my thoughts for the day.
Thanks. Thanks, Mary Frances. Diane?
Yeah, let me know if you're getting a little noise.
Yeah, there was some feedback, but they may have fixed it in the back.
Great. I'm Diana Zuckerman, president of the National Center for Health Research, and for those of you who don't know what we do, let me just briefly explain that we conduct research, but most of the time we're scrutinizing other people's research and also trying to explain that research to patients and to providers and sometimes to policymakers. So a large part of what we do is communicating often very complicated information. So of course the issues that we're talking about on this panel are very relevant to our work. One of the things that becomes really clear when you talk to patients, but even when you talk to providers, is that so much research is focused on how good is this product for everybody and how, what are the safety problems for everybody, and that's not what patients want to know. They want to know, what does it mean for me? So if I'm a woman, maybe it's different. Or if I'm over 65, maybe it's different. Or if I have a high BMI, maybe it's different. So all of these issues that are part of this whole idea of personalized medicine indirectly are really important to patients. And it isn't just FDA that's not focused on those subgroup analyses. You can't find them in most medical journal articles either. And so the vast majority of information we have is really based on everybody and not on particular types of patients. I want to mention another problem which Stephen raised and is really important, and that is the need to talk about absolute risks and absolute benefits. It isn't just, you know, maybe this doubles your risk of something terrible happening, but maybe it doubles it from 1% to 2%. But the same thing is true for benefits. There are many medications on the market and many treatments that patients are being urged to have because it reduces certain risks by 35% or 50%. And again, sometimes it's from 2% to 1%. So those absolute risks as opposed to relative risks are really important to communicate to everybody in this healthcare systems that we're dealing with, and obviously especially to patients when we're talking about the Sentinel Project. So I want to also go into this whole question of how do you balance providing information in a transparent, timely manner to patients who will want to know it, with raising anxiety, providing warnings that may end up being incorrect. These are the big issues that we're dealing with on this panel. I do want to say that I'd like to see some harmonization of those concerns across the centers at FDA. There are many, just as an example, many diagnostic devices, diagnostic tests. One example I think would be low-dose CAT scans for lung cancer, where the false positive rate is something like 90%. I mean, it's really, really high. And it's particularly bad for low-dose CAT scans because usually, if it's an ambiguous finding, which is often is, patients are told to wait 6 months and be tested again. So you get 6 months of anxiety, not just a few days of it. So we need to think about anxiety not just with Sentinel, but with everything that fits in together with information and treatments that we're offering to patients. I think the big issue, obviously, is that the FDA is a public health agency. And as a public health agency, the goal has to be providing information that will improve the public health. And that, again, gets into complicated issues of what are the risks of warning patients about a possible problem versus the risks of scaring patients so that they don't take a treatment that could have helped them. And that will partly depend on what the alternatives are. And so again, I agree with Stephen and others that we need to look at comparative effectiveness and comparative safety issues, not just one product, and also not necessarily just a type of types of drugs, but various treatments that might be available. So basically, I think that I haven't seen the evidence, and I'd certainly be interested to hear what is the evidence of how much harm does it do to patients when there's a warning that's premature and ends up to be perhaps incorrect or perhaps specific to a particular type of patient but not to all patients. So I think one of the ways that we can reduce that concern about losing enthusiasm for certain treatments is to make sure that this, the analysis that Sentinel does is specific as much as possible to finding out who is most at risk. And that can really vary, obviously, by age, sometimes by race or ethnicity, sometimes by other, comorbidities. I mean, there's just many different issues that can be very relevant. And one of the things that we always talk about at our center is the need to have a subgroup analysis that looks at safety and effectiveness of a product for a group, not just looking at, is this more effective for men than women? Because honestly, as a woman, I don't care if it's more effective for men than women. I only care if it's effective for me. And same way with safety. I don't care if it's a bigger risk for women than men. I only care how big is that risk for me as a woman or me as a person of a particular age group that I won't go into. In summary, again, FDA is a public health agency. That has to be the major goal of this project and of these analyses. Transparency is really important and a key for trust for patients and for providers. Early warnings are essential, and they just need to have those appropriate caveats. I think Terry talked about that, and yes, you have to have appropriate caveats. You don't want to unnecessarily scare people when you don't know yet what you have found, but that doesn't mean you shouldn't let the public know what you have found so far. Again, subgroup analysis can be key both in terms of making sure that patients and providers know what the benefits are and what the risks are in absolute terms so as to not unnecessarily scare people off from particular treatments, but also to provide appropriate warnings. And I don't think we're quite ready yet to get rid of the adverse event reporting system. And I will just, even for the lower risk products, just on a personal note, I recently found out that a medication I was taking was the reason I was getting these weird hives. I mean, I just had no idea. And hives are not serious, but boy, they were annoying. And so I wouldn't want to be in a situation where that kind of information is not public. I was able to find out that it was a reported adverse event, and when I stopped taking the medication, they went away. So sometimes that information is useful, even for side effects that seem pretty trivial. Thank you.
Okay, great. I'd like to thank all of the panelists and the presentation by Terry. A lot of different views and useful information. I'm going to make sure that we have time for Q&A from the audience, but first I'd like to maybe probe a little bit more on a couple of themes that I heard come up from the group. What specifically to communicate, how to communicate that, and then a lot of discussion around sort of the need for more collaboration. So I'd like to touch on all three of those things. But first, what gets communicated? And this is where there seems to be a lot of concern, and where we really do need a good balance of not communicating in a way that results in premature discontinuation or even worse adherence than what we might already see unnecessarily, versus boy, we do need to communicate something. And so I want to hear from, maybe weigh in across the group a little bit more on, what should get communicated. And it's important to put context and to maybe have more specific information of what we're seeing by age group and by gender and all of these things. But at the same time, it might be something where the drug isn't even causing the adverse event that we're seeing. So how do you, how do you deal with that, that sort of tension that you all, any additional comments based on what you heard from your fellow panelists?
So, I certainly think, as I tried to say, I think there's some elements, we have to describe the level of evidence in a meaningful way that's understandable by the broad practicing community. Because I do think that will help inform that provider to have conversations with their patients. And I think the other piece that I thought would be really important is really understanding the time horizon on when we're going to have a more definitive answer, because I think that level of uncertainty becomes very challenging for that provider-patient conversation in understanding that. Because I think that's where, how long are they going to have to live with that uncertainty? I would agree with Diana that, kind of understanding what the totality of what we do know, right? Do we know that there's any trend towards a predominant population or an elder risk, or I think that type of information. And so that really gets to the timing of when do you have enough information to make a meaningful communication. And I think that's where the tension in the system is around as soon as possible, but as soon as possible with enough information for appropriate context. And I think that's going to be the challenge for FDA, to really kind of have enough inputs to know what's that tipping point around when do I need to communicate. And I think that speaks also to the front end filter of what signals rise to a level of magnitude that warrant a proactive communication when there is still uncertainty, as opposed to the non-serious ADR that we may not communicate till we're certain. And I think those are the things that I would say should be factored into the thinking around that.
Any other comments? Stephen?
I think that when we look at spontaneous reports, their major gain is the human suspicion behind them. When we look at patient safety programs, there's no human suspicion and their value is close to zero. And I think that the PRAC at the EMA made a crazy decision in pressing companies over that. But the consequence, I think, is that we have been used to spontaneous reports where we do not have denominators, and we do not have, even with the nicest of our spontaneous reporting algorithms, we do not have any genuine estimate of relative risk in usage. We may have an excess of observed over expected, but it doesn't necessarily relate to relative risk. Now, with this system, we have the great advantage that we have denominators, we have time exposed, we have doses, and we have all sorts of information. And I think it would be a great mistake if we simply have single point estimates and we start even using statistical confidence intervals, because they're not going to be helpful to patients. We need to have some ballpark figure that the statisticians won't like of the uncertainty in the excess risk that this has. Now, I think we did this quite well many years ago with hormone replacement therapy in the UK. We actually said, if you take combined hormone replacement therapy between this age and this age, if you didn't take it, you would have this many breast cancers, and if you do take it, you will have this many. And you have the actual numbers that you expect to have over that time period. And I think that if you're going to communicate sentinel signals identified, you ought to do something similar, if you possibly can. And I don't see a good reason why you can't. So that's the what.
So maybe push back a little bit on that and get the everybody seems to be very friendly up here, and I want to get some controversy even more. So great. So we communicate that, but I guess the challenge that what I'm thinking in my head is that this is like the earliest step of the process, and if we're communicating about the results of a tree scan analysis that was purely signal identification, and we reduce some of the bias, but certainly not all. Next steps would be, more hypothesis testing or signal refinement and evaluation, so that we can at least start to potentially probe cause and effect, or at least strengthen the association that we're seeing. If we take that very preliminary result, where we're not even sure yet, and try to put it into a communication that patients would, if I take this drug, then I might experience this or that. Is there still the challenge there of communicating that before you've done all of the additional analyses that you probably should, or that at least you plan to?
I think it's a bit like the ethics of randomized trials. I think it's unethical not to do them. All right. I think if you're going to communicate anything, you've got to communicate something that is really helpful to patients and to prescribers. Now, I'm not too worried about damaging the companies by raising scares unnecessarily. All right. I think eventually people get to realize. I remember when people talked about making spontaneous databases available publicly, they said, oh, this is going to lead to all sorts of terrible scenarios, and it didn't happen in general. There will have been the odd lawyer who's raised problems, but that hasn't been a general problem. And I think that when you get out doing this, at first there will perhaps be some concerns, but people, the public are not that stupid in spite of their voting behaviour in the UK. I can't comment on voting behaviour anywhere else, but I can in the UK. But that if we communicate that we think that there is a problem that might increase your risk of liver disease, serious liver disease, from a rate of, let's say, 1 in 10,000 to perhaps 2 or 3 in 10,000. So that in your community, and we often, and a previous chief medical officer in the UK talked about your street, your village, your immediate surround, your thing, or your city, or the country, and you talked about the size. One person in this who takes the drug will have that sort of problem. I think you've got to do something of that kind, and you have the data to do it. It is no good having some complex statistical method and describing that in great detail and then giving a relative risk that doesn't help you. The man ringing the bell in the City of London asked, "What are you doing it for?" "It's to keep the elephants away." "Well, there aren't elephants within thousands of miles from here." "Well," he says, "you can see I'm reducing the risk by at least 10,000. So we need to make sure that we have that kind of context, and we also need to have the benefit. You know, there's lots of controversy over the benefits of statins, and people have talked about the absolute risks of coronary heart disease, and patients are beginning to get to understand that your risk of coronary heart disease will change a great deal. And when, Diana, you finally reach my great old age, you will realize that, you know, lots of these risks have accumulated, all right? But when you reach 75, you can't die a premature death any longer.
Great. Thanks for those comments. Diana?
Yeah, I just wanted to add, really to support what you just said, Stephen, but also just to say, to go along with that I think that none of these risks are going to be that high. You know, it's not like we're going to miraculously find, 50% of people are dying from some drug. The risks are going to be pretty low, and so the benefit of letting people know that there could be a higher risk than was thought, or maybe even a higher risk than some other treatment, but that the risk is still, below 5% or below whatever percent it is, let alone 1 in 10,000, people can understand that. And, there's all kinds of, some people are risk averse and some people aren't, but at least in talking to physicians, mostly what I hear is that when you tell a patient that something has a risk of a side effect of 10% or below, they say, "Oh, well, it's only 10%. And if you say you have a chance of living longer if you take this drug and it's a 10% higher, they say, "Oh boy, great, that's wonderful." So a lot of patients really are focused on they like to see the good news, and they don't worry so much about the odds of the bad news. So I don't think we have real good evidence that very small risks, obviously, we're not going to know exactly what the risks are until we're further along with the analysis, but I think the preliminary findings are not going to be that scary.
Okay, go ahead.
Come back and be controversial, if you like. If you want to learn about communication, go and read the Vigor trial report from, was it 2001, in the New England Journal of Medicine. What a terrible mess. What things that bordered on scientific misconduct, reversing the basis for MIs with rofecoxib compared with naproxen, compared with GI effects, and analyzing the data on MIs in the crudest way possible, whereas analyzing the data on GI benefits and emphasizing the slightly less serious ones, Go and read that, and it's an example of almost exactly how you shouldn't do it, right? And there is some absolute risk in there, but not enough. And there is balance of risk and benefit, and you see that the risk and benefit of MI and CHD is there. I think rofecoxib should never have gone from the market, incidentally, but there we go.
Great comment. So I do want to open it up to the room, and if this is like the last session, there should be quite a few people. As you're lining up, I've got one more question to the panel, and I do want to quickly touch on Joanne, the one point that you made around the collaboration. Clearly point taken around collaboration around methods and approaches for actually doing the signal identification, but you also pointed out how a collaboration might help improve evaluating whether or not the communication itself is effective. And so if we look at what Terry had presented, by showing an example of what the row might look like as the communication and doing it quarterly, like FAERS information is reported out quarterly. I get the sense from the group that maybe because we're not doing, this is not just numerator data, that it is also inclusive of the denominator, that an additional step might need to be taken to better communicate, clarify, what that result is. So comments on how the collaboration or collaborative approach might be able to help FDA and collaborators along with thinking about that?
Yeah, I mean, one could envision a trusted third party taking on, let's say, classes at a time, working together to say this quarter we're going to start framing out how we're going to look at this certain class of drugs or a certain therapeutic area or disease. And then doing the analyses, doing a variety of analyses, again, looking at not just different methodologies, but different databases. And then when you find the signals, doing the predictive modeling, looking at subgroup analyses, if there is something real, finding a good way to communicate it together. And then that same group could plan, we anticipate that the use of this product would go down over a certain period of time. A lot of times all that's followed up is just on the use of the product rather than the outcome. So for example, did the incidence of MIs go down or the incidence of GI bleeds go up, after a certain action rather than just the use of the product. And you could plan that rather than waiting, years and just by chance some academic or interested company or regulator looking at that. It could be planned so that we can make sure that our actions, our collective actions actually did result in a positive benefit to public health. I did want to just add one thing since you mentioned the adverse effect. I wasn't talking about all, it has to be done in an evidence-based way. In fact, the example that you brought up, you mentioned, it was already in the label, and that's when something is a mature product, and if you have a patient support program, and I'm not talking about all reports, just, it has to be in an evidence-driven way, looking together to see what's low value versus what's high value to add to the product labels. Because many times, and we have this example, we looked at 1 million adverse events in a patient support program, not in spontaneous reports, but patient support programs, which is what you mentioned, that it didn't add anything to the labels, right? Either the information was already in there, it came from patient reports, et cetera. So I just wanted to make sure you understood it has to be a data-driven way that benefits patients without generating extra work that doesn't add benefit. That's the philosophy.
Okay, great, thanks. So I'll go to a question over here.
Hi, I'm Sheila Weiss, consultant. Thank you for the panel. There's a lot of really important points here. And as an epidemiologist who's actually taught data mining, I also like to second going to think about how we use words very carefully. If we're data mining in the, for finding signals, we usually call them alerts, and then because they're statistical. And then the next point is they have clinical evaluation, and then it becomes a signal. And if that's the case, we might want to really think about when and what we communicate to the public. As epidemiologists, we know there's a lot of studies that get done and later disproven. Is it Chinese food that's bad, red wine, butter, margarine, cell phone use, and the same with the drugs. Is it one drug? Is it a class of drugs? These things take a lot of thought to design a good study. So when you're doing this type of exercise, you're really trying to wring out a little information from the data, but it doesn't have any clinical veracity. So really, I would suggest that communicating a fishing trip or data mining expedition is probably more harm than good. We really have to wait till the next stage. And at that point, really there needs to be a conversation is, what signals do we follow up on? You don't want to necessarily just go for the one with the biggest odds ratio because it might be biased, but what are the ones with the most public health information? So again, really thinking through each step of the process and what moves from a statistical signal onto a actual second study, and when, at what point is that really of value to communicate to anyone? Thank you.
So a suggestion to maybe not communicate after that first stage where it might still be an alert and not yet a signal because you haven't done the subsequent analyses. Any comments or reaction to that?
So, I mean, I think my understanding is there is going to be a clinical filter before any communication would happen, and my FDA colleagues are nodding. But I think even when you have that first clinical pass, that concept of signal is still not well understood in the broader medical community, that a signal still warrants further investigation. So I think that's where understanding what the investigation plan is, if you're going to communicate, sharing that so people know. I think the other risk, and we see this all the time in spontaneous reports, as soon as something gets out there, a wave of reports comes in. I think that same stimulated reporting effect could influence the databases you're looking at, because physicians may tend to enter more events into their coding. They may enter more observations that may further confound or make more difficult the interpretation of the subsequent analysis that's going to be done to further confirm that signal or refute that signal. So it may be harder to refute the ones that should be refuted because you end up with excess of reporting or excess of coding. So I think those are some of the risks that I see, and that really drives the timing of that communication relative to when you're going to do that subsequent analysis.
Yeah, I just want to add, as someone who works a lot with explaining research results to patients and helping them understand risks and benefits, I think most patients would think an alert is worse than a signal, so you really do have to be careful. It reminds me of tornado warnings versus tornado watch. Which is the worst one? It's so hard to remember. And I think alert sounds kind of scary and signal kind of doesn't, but.
Terry?
So I think when Monica described how the safety evaluators do their work, they look at lots of signals, and even the 921 postings. Not everything is posted there. It's when the denominator for the 921 postings is what we currently call TSIs, track safety issues. In the future that may have a different name, but that's the denominator. So something has already risen to the level where we're going to do an evaluation that Monica talked about. So we're not communicating about all of the alerts that come out of Sentinel. It's a thoughtful process. But even so, it's still trying to think about not scaring people and finding that right balance. But, and Diana, the sensitivity about the words and how what we think, I mean, that's a good point.
Great. Next comment or question?
Yes, good afternoon. I'm Ira Crafting from the FDA, and the panel has raised some very important educational points. I did want to present for discussion a practical example of communication and to get the panel's response in this exercise. To bring everybody up to the initial thoughts, gadolinium-based contrast agents have been around, as we know, for 30, 40 years. It was discovered in the last few years that there is deposition of gadolinium in the brain after getting brain and other body organs. After getting some of these scans, the scans, at least in the United States, are done through the day and night. It's hard to avoid at times. This is a phenomenon discovered in animals and in some human autopsy studies. It did lead in the Kingdom and the EMA to removal of the marketing application of certain of the gadoliniums as a precautionary measure. Here in the United States, we took a different tact, issued a warning, and it seems to have generated a global taking sides, as Dr. Zuckerman might have mentioned. When we do things, the FDA, various agencies around the globe have either removed some of the agents or followed the FDA warning pattern. So with all that background information, where we have a phenomenon without a clinical consequence so far, how would you communicate that to the public, and how would you approach those steps? And as a final question, would Sentinel even be helpful in that total regard? Thank you.
Thanks for the question. Any comments, or Stephen?
I was a dissenting opinion at the EMA on that issue, but I think that it would be very difficult because a lot of it was theoretical and not actual clinical. When it came to the nephrogenic systemic fibrosis, there was clinical evidence. And so if you were to use that as your example with gadolinium agents, there was actual clinical evidence, and then you could communicate. So I think that you need to distinguish between things that are theoretical. I don't think that Sentinel signals will be generating theoretical things of that kind.
Great. Comment?
Tom Moore, ISMP again. Most of the problems that I wrestle with in the drug safety business every day are insoluble or really create tremendous problems. And interestingly enough, listening to this, I'm not sure with respect to the Sentinel signal detection that we really have a problem. It seemed to me we have a problem about communicating accurately about drug risks, and I thought that Stephen Evans did an outstanding job of explaining the kind of information in a broad sense that we need to communicate to public better than we do today. But laying at the door of a pilot program for signal detection, being all of these issues, I think, is way too heavy a burden. I thought that Teresa showed an example here that's very similar to preliminary communications the FDA already makes that look perfectly reasonable to me. And I read drug safety communications every week, and it's not unusual to see an emerging risk and a possible risk. Patients go on the new dashboard and come back with patient deaths to their physician totals, which of course unfortunately is a mistake. But what I want to say is it really is a complicated world. It is really worth possibly a whole workshop to say, can we organize this in a different way? But I think that the fundamental principles I've heard here, that it ought to be transparent and it ought to be disclosed, and that have guided Sentinel since its start, are really good principles.
Okay, great. Thanks for the comment. And Bill?
Yes, it's William Dumashal at Oracle Health Sciences. I think there's an aspect of the methods discussed in the first session, which was a very nice section, that will also influence the idea of risk communication that we're talking about here. And that is, when worrying about the multiple comparisons problem, the focus is so much on hypothesis testing rather than estimation. And it's kind of well known. I mean, if a signal shows up as verified even against multiple comparisons, then it's probably going to be an overestimate, just because of the effort needed to do that. And similarly, of course, if you can't pass the multiple comparisons test, then the estimate seems to plummet to 0. And that's certainly going to affect the risk communication issue. If you use my preferred method of Bayesian estimation, and you focus more on estimation rather than testing, you end up with, what the Bayesian posterior mean is typically called a shrinkage estimate for a reason, because all the estimates shrink, especially the significant ones. And so if you have to produce estimates of absolute risk or whatever, if you do the calculations starting with the shrinkage estimate rather than the number that popped out of them, multiple comparisons hypothesis test, I think you'll be better off. And this is not only true for the existence of the basic signal, but it's probably even more true for the subgroup comparisons that was also discussed quite a bit. Again, if you look for the age groups or the genders or whatever, past medical history that seems to produce the biggest effect, again, and then sooner or later you're just going to quote that, especially if it passed some kind of multiple comparisons, group comparison test. If you get shrinkage estimates of all those differences between subgroups, it'll probably be much of a less scary issue.
Any comments or suggestions? Certainly goes into the methods used that give results that might more or less be clinically interpretable.
As I said, I'm not clever enough to be a real Bayesian, but I believe in Bayesian hierarchical models and shrinkage towards things that are in that hierarchy. So I support everything that William or Bill said. I would just add one minor thing, and that is, if we're uncertain about whether to communicate or not, how about randomizing it? Okay, provided you've got a way of assessing the impact of what you expect to find as a good or a bad influence of it, try doing, in your initial days, randomizing whether you communicate or not. You know, there'll be some things that you'll be certain that you ought to, and some that you're uncertain about whether you should. Those, randomize.
Yeah, I just have one follow-up comment here, and it raised a point to me in that for the more learned communities who've been looking at Sentinel communications over time, the transition from being more of an evaluation tool to a true detection tool, data coming out of Sentinel may be weighted more heavily than the traditional track safety communications that you've done so far with spontaneous, because everybody recognizes the limitations of spontaneous data. So I think as we transition into communicating via the Sentinel tools, what we're seeing there, it may be given more weight by the recipients than what we have done historically, because the understanding of the limitations of spontaneous data are well known. And so I think we just need to, as we transition there, I think there may be some unique communications in that new phase of work coming out of the pilot, that they're not giving undue weight, because they are essentially a signal that warrants further evaluation at that point in time. So,
sure, Diana?
Yeah, I just want to add, I mean, we've kind of talked around it, but the difference between statistical significance and clinically meaningful differences. And it seems to me that's going to be something that has to be dealt with, and that's a lot more subjective than statistical significance, but things can, especially in these huge data sets, they can be statistically significant, but what difference does that make to any individual person? It might make an important difference on a public health level, and that is important, but in terms of patients understanding what's the likelihood of this affecting them, I think it's
great
crucial.
Point. Okay, we are out of time for this session, but one last quick comment.
Yes, my name is Anna Sharpman. I work for the Food and Drug Administration. We should not forget that we are working with non-traceable data. When we are talking about the need to have chart reviews, is an example of the problems that we have with this data. It's not electronically traceable, then maybe what we need to start doing is start to think about how to make the medical data interoperable from the primary data, and then we will be able to get electronically traceable data. We will ameliorate the problem of having to ask questions. Maybe we will get to a situation like with weather maps, that we don't need to ask questions, but the data is allowing us, speed on its own, and let us know where the meteor or whichever is going to hit. Thank you.
Okay, great. Thanks. So we are going to go to lunch. So as you contemplate solving the data interoperability problem, as well as randomizing whether or not we communicate or not, you can do that for an hour. So we'll be back here at 1:30 There are some restaurants in the area. Again, there's a list of the available restaurants at our registration desk out front. And I'll see you back here at 1:30. Thanks a lot. Stage. What do you want to do? You can do it from your seat. Yeah. I don't need to see it. Okay. Yeah, you can do it from your seat. This is me. Have to look and see where we are. We've got a. Well, I'll pass this down. Yeah. Okay. Okay, so I see that the real statisticians have come back after lunch. Okay, so this morning we discussed a lot about FDA's vision and key considerations for how signal identification might be implemented as well as operationalized within a regulatory framework. This afternoon, we're going to build upon this discussion and take a much deeper dive into the technical details and specifics of signal identification approaches that FDA is considering for implementation. It's important to understand how these approaches might fit within Sentinel's distributed data network, as well as their key strengths and limitations of the techniques that you're going to hear. I want to remind everybody that the clear goal of this session is not to select the single one best technique. It's most likely that there will be a suite of techniques depending on the clinical area, the context of the safety concern, et cetera. And so it's important to understand that we envision a suite of options and tools that can help complement and support and augment the existing capabilities of the Sentinel system and regulatory needs. This panel will start. Hi. Oh, okay. Yeah, no problem. Okay, this panel will start with a presentation that will help orient the audience to key characteristics of these techniques, and then we'll have a series of focused presentations on each specific approach. So I'd now like to introduce our panel. We're very glad to be joined by Martin Kulldorff, a biostatistician in the Division of Pharmacoepidemiology and Pharmacoeconomics at the Brigham and Women's Hospital. Judith Morrow is an assistant professor in the Department of Population Medicine at Harvard Medical School and the Harvard Pilgrim Healthcare Institute. William Dumichel is the chief statistical scientist at Oracle Health Sciences. Martine Schweme is a research fellow and director at Janssen R&D and is a visiting scholar at the Biostatistics Department at UCLA. And then Niklas Noren is Chief Science Officer and Head of Research at the Uppsala Monitoring Center, which supports the World Health Organization program for international drug monitoring. So now I'll turn things over to Martine.
Thank you very much, and it's a great pleasure to be here. So I'm going to give a little bit of a landscape background of the different signal detection techniques. And the common theme here is that we are looking for things that we didn't expect to see. So if you want to know if drug A causes a health event B, this is not for you, because then you should do like a regular pharmacoepi study. So this is when we're looking for things that we don't really expect to see something. So data mining is useful for that, when we simultaneously evaluate multiple drugs and or multiple events. So we have multiple drug event pairs. And there are some common goals of data mining methods. And of course, we want to find the unknown adverse reactions, the unexpected adverse reactions if they exist. We want few false positives, or if there are false positives, we want those that are easily explained to be obviously false positives. We want sufficient power to detect rare adverse reactions. And of course, if there's a known adverse reaction, then we want the data mining methods to find them, because we want them to find both what's known and what's unknown. Now, when we do data mining, we can have 4 presentations here. We can sort of view it as 4 separate things, but we can also view them as different combinations of design features that we can do for data mining. So data mining has different design features, and these could be combined freely to create any hybrid approach that's your favorite. So it's like when you go to the ice cream store, you pick a cone or a cup or different type of cones, and you pick your flavors and your toppings, so you can create your own ice cream sundae with your favorite ingredients. So you can view the same thing with these data mining methods. You can pick one thing this and one that, and sort of create your own favorite thing that works well for your particular study. So what are the key features? Well, one is, of course, we can use different types of data sources. And most of these methods can be used for different data sources, even if some tends to be more popular for certain data and others for other data. Another key feature is the risk window, when we sort of are thinking that this drug might increase the risk of some event. This could be identical to the exposure period, and sometimes that's the case. In other studies, we have one or more pre-specified risk windows, like 1 to 14 days after the incident exposure. Or we could do a temporal scan where we simultaneously evaluate hundreds of potential risk windows, sort of 1 to 14, 1 to 15, 2 to 18, 7 to 21, and so on, and hundreds of those. So that's one thing, that we can sort of pick one of these and combine it with any of the other features that we're discussing. Another key feature is the comparison group. We could compare with all individuals, with healthy individuals. We could have users of different drugs, or we could use different self-control methods, either with pre-exposure or post-exposure. So this could also be used in combination with these other features that we have. Another one is covariate adjustment. Sometimes in data mining, we don't do any adjustments, and that can sometimes be appropriate, especially in self-control methods where we have automatically adjust for any between-person variation. And sometimes we might adjust for age, gender, or concomitant exposure, or we do propensity score matching, which was mentioned this morning. So that's another thing that we sort of can select any of these. And so far, those are sort of general towards any pharmacoepi study, because we have to make those choices in any pharmacoepi study. But then there are certain things that are unique also to signal detection or data mining. And one is the granularity of the outcomes. So if you look at multiple outcomes, we could use the collection of very specific diagnoses, such as all the ICD-9 codes, ICD-10 codes. Or we could use a smaller collection and look at all the more general group of related diagnoses, like liver disease or heart disease, so bigger groupings. And sometimes it can be better with one or the other, but if we don't really know what we're looking for, we could also want to simultaneously look for both the very specific, very fine granular outcomes, as well as the bigger ones at the same time. So this, again, we can sort of choose either of these depending on our situation. Another key feature is how to deal with multiple testing, because we're looking at, for many potential drug event pairs here. So one option is to do no adjustment. And in fact, we maybe do no formal adjustment, but informally we'll always do some kind of adjustment, sort of in our mind. Another one is to do some kind of informal adjustment, say, well, we want a 95% confidence interval to be greater than 2 or greater than 1.5 or something like that. There's another informal way of adjusting for the multiple testing. Another one is to do formal analytical Bonferroni type adjustment, which is the most common way to do statistical adjustment for multiple testing. But that works well if we have independent studies, and if there's a lot of dependency, it sort of falls apart. And then we can instead use formal Monte Carlo adjustments with random data generated under the null hypothesis. So these are different ways to deal with the multiple testing issue. And then another thing that was mentioned this morning also is how do we present the effect estimates. So we don't have to do only one, we could do multiple, but we could do relative risk odds ratios. We could do some empirical based shrinkage estimates, or we could do risk differences or attributable risk as an option. Another thing we have to sort of decide on is the frequency. We could look at the data only once. So maybe a couple of years after the drug has been approved, we look at the data once, and that's it. That's fine. Or we maybe want to look at it every year for a few years. Or we could try to do real-time surveillance when we sort of continuously get data, maybe on a weekly basis or so. So that's another aspect, the frequency of how often we look at the data. And then the last one is the size and type of the net that we're casting for this. We could look at one drug and thousands of disease outcomes. We could look at one disease and thousands of drugs. We could look at thousands of drugs and thousands of outcomes. Or we might be more specific for a specific population, and for example, only look at pregnant women and birth defects. Or we could also look at drug-drug interactions. So those are different features. So all of these can sort of be combined just like your ice cream sundae as to your favorite data mining methods. And some of these has been used in practice, but you can also come up with something that's completely new and unique to you, just as you can at the ice cream parlor. So when you sort of think about these 4 talks that's coming, you can think of them as a very unique method, but you can also instead think of them as, okay, this method used this thing here, and this feature there, and this feature there, and this feature there. And that sounds nice, except this one I don't like. I'm going to change it to that one. So this gives sort of a bigger toolbox, if you want, to choose from. And as you listen to these talks, you can sort of now tick through, like Judy's talk, okay, she used this one, this. Bill used this. And if you note that down, we'll have an exam at the end of the session that see how many correct ones you have. So thank you very much.
Okay, so next we have I'm going to leave it up to the presenters. If they want to go up to here or if they feel comfortable, I can give you the clicker to go from your seat. So it'll be Judy next.
So folks, I have, is this on?
Press the button, yeah.
So I have the pleasure of having a great setup today because the Sentinel system has utilized TreeScan most heavily, and I'm going to be presenting on TreeScan today. And so there was a lot of discussion this morning and also things that Michael presented that will seem very familiar to you. I also happen to be seated next to my very frequent collaborator and mentor in the use of TreeScan, and so there's a lot of pieces that you will see come up again, including the features that Martin just talked about. And so just to lead off, so tree-based scan statistics are enabled by a freely available program called TreeScan, which you can download and which is available for PCs, Macs, and Linux users. It is a signal detection or a data mining method. It does scan electronic health outcome data that have been grouped into hierarchical tree structures, and I'll give an example of that in a minute. You'll see that. And it does, as Martin was saying, and if you're thinking of your flavors here, there is an automatic adjustment for multiple hypothesis testing, and this, in fact, is using the Monte Carlo method to do that adjustment. So what I'm going to be focusing on today, as Martin said, there's many different types of, this was sort of the safety net feature that he was talking about. So what we're going to be really focusing on is when we pick one particular exposure of interest and we look at many outcomes, what we've done when we've implemented TreeScan in the Sentinel system is use what's known as the Multi-Level Clinical Classification System, which is a product of the Agency for Healthcare Research and Quality. It organizes health outcomes by clinical groupings, and we use a tree that's been curated that has around 8,000 outcomes in it. There was a talk earlier today where there was a question about, well, do we really have to pre-specify every single thing we do? So in this case, we actually do pre-specify these 8,000 outcomes, and you can find that on the website. So there's a downloadable tree, and it is in the 9 era and the 10 era, so you can see outcomes that are in the ICD-9 framework as well as in the ICD-10, and they're listed there for you. So what does the data look like when it's arranged in a tree structure? I've put in here a small sample of the multi-level clinical classification system. I've also put next to it a MEDRA sample so that you can sort of see the similarities between them. At the lowest level term, or at the leaf level, is the actual diagnosis code of interest, and then it continues to aggregate up to, at the very top, you have the diseases of the nerve and sense organs in one case, or the system organ class. And the idea behind here was the feature that Martin was speaking about outcome granularity, which is that when you use a tree, you don't necessarily have to know how the clinician intends to diagnose something. And so, for example, in this tree that I'm showing you here, the ICD-9 code all the way on the left and all the way on the right, we see as the most frequently utilized in the Sentinel system for this type of event. So you see these are more general codes. They're either listed as other convulsions or just convulsions. And in different practices, they may choose to use the the one all the way on the left, and in others they may choose to use the one all the way on the right. And the advantage here is that you don't have to know which one in any given practice, because the hypothesis testing will be done at both of those levels, as well as the aggregate level of convulsions. So this is a slide I'm going to spend a little bit of time on, but one of the things that I wanted to emphasize, and one of the things we talked about this morning, is that really with TreeScan, the study design leads. And what I mean by that is the very first question that you have to ask yourself is, what is the comparison you want to do? Do you want to look at people and do something like a self-controlled design where you're comparing a period of time within person when they're exposed to a product, perhaps to a time before in a pre-control period or a distant control window period? Are you going to look at an active comparator design? Are you going to compare new users of medical product A to new users of medical product B? Are you going to look at some type of a comparison to a historical standard, and that's that stratified cohort design? So am I going to look at a current versus historical comparison, or perhaps to an unexposed group? So that was spoken about this morning as sort of, if these people were exposed, what about the time when they were unexposed, but not in a self-controlled design, but rather in an aggregate sort of structure? And so depending on what kind of comparison you want to do, again, that's sort of the lead. Once you know what kind of comparison you want to do, what kind of design you want to implement, then you can find sort of the TreeScan analytic model that actually matches the design that you intend to execute. And so there are different analytic designs that are designed to work with different, there are different analytic models that are designed to work with different baseline epidemiological designs. And in TreeScan, there's a concept of what's called an unconditional or a conditional. The difference here being, if in an unconditional analysis, external information drives the null hypothesis. So for example, in a match design, I might set the matching ratio. I may say, a priori, we're going to use a 1-to-1 matched ratio. That's an example of external information setting the null hypothesis, whereas a conditional hypothesis or conditional analysis is based on the information that's coming in from the incoming data set. And so how has TreeScan been evaluated this far? Again, Michael did a lot this morning to talk about this, so I can run by it really quickly. But essentially, we've used a balance of simulated data sets and empiric assessments. And the reason for the balance is basically the advantages and the disadvantages of both. In a simulated data set, you know what you know and you control the truth. And that's really a great advantage when you're looking at statistical characteristics, because you can put a risk into a data set and then say, okay, I'm going to figure out whether this method can detect it or not. And you've set the truth, you know what the truth is, so then you know its performance characteristics. Some of the disadvantages of that is that depending on how well you do your simulation, you can have things that are sort of more artificial and then more realistic. Empiric assessments are very useful to understand how the data will come in and what it will look like and how you will have to triage them and react to them, but then again, you don't know the truth. So you can't really say very much about what you're missing, because you don't know what the truth is in that actual data set. And so I'm going to walk through, these are a series of papers that were done that are using that grid from before of each of these designs. So these are all papers that have been done with a self-controlled design using a tree temporal scan analysis. So this is one of the flavors that Martin talked about, which is where you don't pre-specify sort of where your risk window is. You specify an observation window and then allow scanning to go across that observation window. So we worked with that with simulated data to look at power. The long and short of that paper is essentially, if you don't know what the risk window of interest is, and typically you don't in a signal detection study because you have 8,000 outcomes, and with 8,000 outcomes, the risk window for all of them is different. And so allowing yourself to use that scan to go across the entire observation window is really useful. And then additionally, we have work that's been reported out using this method with vaccines, both in the Vaccine Safety Data Link, which is run by the Centers for Disease Control and Prevention, and then also within the Sentinel system. And then we also have done work with drugs that is still sort of going to be, is in the process of in the manuscript phase at this point. But these are the things that Michael referenced before with the drugs of interest that have been studied. So then with propensity score match designs, there's been a lot of work here with simulation to look at using the propensity score match, which is again now you have an active comparator cohort, you have folks who are exposed to drug A and folks that are exposed to drug B, and then you use that in order to do a TreeScan analysis. For the gentleman from GSK who asked a question earlier today about how well the propensity score controls, this is a simulated environment, and it basically shows the degree to which you need to actually measure your confounders and how well you do, and it has a spectrum from full control to sort of the least amount of control, showing what the differential is as you go across. Additionally, the same group of folks are developing a global propensity score, which was briefly discussed this morning. And then finally, there's been some older work with the stratified cohort design, which used a referent cohort. So this was work done with actual drugs, and there were referent cohorts. And what I mean by that is, again, in a stratified cohort design, you've set up sort of what the expected rate of occurrence of events is based on whatever your referent cohort is, and then you adjust your observed events based on what that expected is. And so this is some of the original work that was done there, and there are many authors of this work that are in the room today. It's a little bit older, but it was done. And then there's some simulated work there for power studies. So some of the strengths of TreeScan, it inherently takes advantage of a hierarchical nature of clinical concepts. As I mentioned before, the investigator doesn't need to understand how a clinician would code a particular outcome, because essentially the hypothesis testing will be done at the leaf level or the lowest level of the tree, the diagnosis itself, all the way up to sort of aggregate clinical concepts. And there's an automatic adjustment for multiple hypothesis testing. It is a Monte Carlo adjustment, so it is not as severe as a Bonferroni adjustment. That's because the hypotheses themselves are highly overlapping in a way to control for overall type 1 error. The limitations, though, are that all the outcomes are treated identically across the tree, regardless of their onset or their severity, which is why we should be reminded that we are doing signal detection. And so essentially, there is further work to be done. Nothing that comes out of a signal detection exercise is the end. There has to be work that then characterizes and looks at, what is actually happening. Additionally, in TreeScan particularly, there isn't complex outcomes, so algorithms that say something like an outcome is defined as 2 or 3 codes that occur within a certain number of days of each other. That's not something that TreeScan handles. It's looking at the individual occurrence of a code. And finally, in terms of the limitations, most of them are inherent to the actual study design chosen. So basically, when you choose your design, you're sort of locked into already some of the limitations of that design, and those carry forward in the analysis as well. Finally, I guess an overall limitation that maybe that folks in the audience have sort of probably picked up on today is sort of the limitation of the data source itself. So we are looking at medically attended events. If I woke up this morning and I had a migraine and I didn't go see the doctor, no one's going to record it. It's never going to show up here. So those sorts of things, just to remind that we're always sort of drawn to the data set that we have and the limitations of that, and to keep that in mind throughout. And that's it.
Okay, thanks, Judy. Appreciate you going through that rapidly. Bill?
Thank you. So I'm going to talk about a series of methods that are, to use Martin's ice cream analogy, in Boston they're called mix-ins. And I think that's a good phrase, because these Bayesian ideas can be mixed in to virtually any other methodology. The idea is that the techniques, and especially the ones I'm going to talk about, which are in the hierarchical mode, where you have many different versions of the same problem, you combine these multiple examples to allow borrowing strength, which kind of means they're weighted towards each other. So you have to assume that there's a kind of an average effect that's somehow relevant to each of the individual possibilities. And so when you have many parallel problems, the Bayesian model assumes some kind of similarity of effects or sources of variation across the multiple problems. And they work best with the large databases where you can draw on multiple estimations and measure variance components, because typically there will be some measurement of a variance component that decides how much to shrink or how much to draw the different problem answers towards each other. The primary strength is that they often provide accurate adjustments for multiple comparisons. Now, interestingly, the multiple comparisons methodologies that Martin discussed, actually, he didn't mention the Bayesian shrinkage as a primary. And in fact, Bayesian methods haven't often focused on the multiple comparisons ability, partly because many Bayesian methods, which don't involve hierarchical and big databases, there's not a multiple comparisons problem there. But in these kinds of situations, the shrinkage does give a different kind of answer to the multiple comparisons problems. That is, rather than taking the standard estimation and then expanding the confidence interval to provide multiple comparisons prediction, the Bayesian methods kind of do the opposite. They shrink the actual estimates towards the null hypothesis, but the confidence interval is not actually expanded. In fact, it's sometimes reduced as a result of that calculation. There may be problems with this if the individual problem you happen to be looking at is kind of more like a black swan outlier that doesn't follow the general pattern of other examples. But that's a typical multiple comparisons issue no matter what. I'm going to talk about two examples very, very quickly of Bayesian methodologies, and they're just two things that some version of which could be mixed in. The first one is taking an example where instead of one versus all, or all versus one, or all versus all, it's maybe an example of several versus several, so that you're focusing on class effects and you're taking perhaps many drugs of a similar class and many adverse events that are somewhat similar medically, and then trying to sharpen those distinctions. You can certainly, you'd have a null hypothesis model for each drug event combination, which might adjust for other things like age and gender and other covariates, possibly even adjusting for concomitant drugs if you had patients that have taken many drugs. And the two-way shrinkage model assumes an analysis of a pre-specified set of drug event combinations, as I said, kind of several drugs and several adverse events. The rationale is that there probably will be a class effect, and you kind of like to do a certain fine-tuning about how similar drugs might be differing. Similarly, with a medical event, we've just been talking about granularity and how to choose granularity. And so you could have multiple events that might seem similar, but you'd like to kind of get straight whether, in fact, some version of them is different. There was a similar model related to environmental drug response slope estimates in a JASA paper of now 35 years ago. So that the example I'm going to give is antipsychotics and movement disorders. And I'm doing this in FAIRS, but it could certainly be done with the other kinds of data that we're talking about, longitudinal data. So here there's 6 antipsychotics listed there, and then 30 movement disorders, which were selected as the 30 preferred terms that had the greatest total number of reports across all 6 drugs with this particular version of FAIRS. And so there's a counts and an expected counts. The Poisson regression, the idea here then is to create a Bayesian prior distribution that could be used with these 6 times 30, 180 drug event combinations. And one possible thing to do would be to say, well, there could be a kind of a general effect of a given drug across the different movement disorders, and then there could be sort of a general effect of a given movement disorder with that class of drugs. And so you could imagine that Poisson model that's there at the bottom of the slide, in which there's essentially an expected count that's kind of a null hypothesis, and then with a multiplicative version of a drug effect and a particular event effect. And then you would take you would take the observed ratios of N over E and shrink them towards the result of that Poisson regression model. This is an example with this data. This is sort of like the raw data. I've used a color scheme to just represent the ratio of N divided by E. And of course, because a lot of the expected counts are so small, there's a lot of variance in that ratio. Now I've ordered the drugs and the events in a certain way to make things a little plainer as to what's happening. If you fit that Poisson regression model, then it becomes something like this. And the reason why that looks so smooth is because I've ordered the columns in the order in which the regression effect showed up from low to high, and similarly I've ordered the 30 movement disorders in that same way, so that you kind of get, as you go to the right and up, you get higher and higher predicted rates. But you sort of think of this now as your prior distribution, and this is the raw data. And then if you use the Bayesian methodology to decide how to shrink these estimates towards these estimates, you get something like this. And this is the two-way shrinkage estimate, which allows a nice combination of understanding that not only is there an overall class effect, but there's probably a kind of an improved definition of a class effect by fitting these regression models. And in fact, we have continued in conjunction with Laszlo Urban at Novartis. We've started looking at those distinctions that are showing up here and seeing with a lot of in vitro data to see whether certain molecular targets can account for these distinctions. The second estimate I'm doing in much less detail. In fact, this is the only slide for it. The idea is to try to take, and again, now we're back in the Sentinel notion of a distributed database, many different data sources. You get estimates coming back from each source, and unfortunately, they don't all agree. Sometimes and you'd like to have some kind of estimation or some kind of procedure for combining or getting a better summary estimate, or at least understanding what's happening. One possibility, if you think of this now as just a three-dimensional problem, there's database D and drug J and event K. You've got this three-dimensional problem, and you have observed and expected for each ones. And the notion is there's probably some kind of true disproportionality that you'd like to estimate. And you could just assume a kind of a multiplicative model, take the three two-way methodologies, the alpha, the DJ and the DK and the JK, estimate them all together using some kind of a Poisson model with priors that would have, for those three things, that would typically be a gamma distribution that works well with this Poisson data. And then having done that, you would get a kind of a smoothed estimate of those three-dimensional lambdas using just the two-dimensional priors. And from there, you could either stop there and say this is a pretty good estimate for what we found in each database, or you could use that as a central prior distribution and then shrink all of the N over E's towards it. So this is an example of, as I said, kind of two mix-ins that could be used. There's many others, many different varieties of each of these problems, as well as many others. But the idea is to use Bayesian concepts of fitting prior distributions to hierarchical setups. Thank you.
Okay. All right. So thanks, Bill. So we'll move on to Martine with some of, if you want to go at it.
Yeah.
Okay. Sure.
All right.
So what I'd like to start with is to try and be more precise in some of the terms that we've been using today. And so as we think about observational data that we want to use for signal generation, well, we can use that data for computing counts, how often does the outcome occur in the exposed, to look at associations, so is the exposure associated with greater counts. But also we can use observational data to try and estimate causal effects. Does an exposure increase or decrease the risk of an outcome? And just to be very clear about what we're trying to do with signal detection is we're trying to find signals that represent causal effects. We're not looking at associations, just statistical associations, not just at counts. We're really trying to look at causal effects. Also, there seems to be some maybe a misnomer, hypothesis-free. I've heard this term used already today. I think what it actually means is you just have many hypotheses. So, for example, Judy was already mentioning that she looked at 8,000 outcomes. So if we look at a single drug for 8,000 outcomes, we just have 8,000 hypotheses. And Judy rightfully remarked that a lot of them are related, so we can take that into account. But really, we're just doing causal effect estimations for 8,000 questions. And we actually already have quite a long history in using observational data to do that. We have many existing methods. For example, new user code method using propensity score adjustment, self-controlled case series, case control, etc. So we have already a long history that we can rely on when we do this. It's just at I would argue, at a different scale. Not only do we already have a large number of methods that we've used in the past, we also have established best practices for using this observational data. I've already heard many of these mentioned before, so we need to pre-specify what we do. We need to be transparent in what we do. That means sharing the protocol, and we believe also sharing the source code. So I was very happy to hear that the FDA is sharing, or Sentinel is sharing their SAS code nowadays. I would also argue we need proper validated outcome definitions, sensitivity analysis, study diagnostics, and we need to run an analysis on multiple databases. And like I said, I think that applies not just when we're evaluating one hypothesis, but also when we're doing multiple hypotheses like we're doing in signal generation. And so what I'm trying to get at is and I think this reflects what Mark McClellan also mentioned, there might be some fading of the boundary between, or at least in my mind, between what is signal detection and signal evaluation. And just to show you how these best practices can be scaled to what we're doing for signal, or want to do for signal generation, I'd like to highlight a project that we've been working on in Odyssey. And for those of you who have been at the Odyssey Symposium earlier this year, you will have heard me and Patrick Ryan and Mark Suchard talk about LEGEND, or large-scale evidence generation and evaluation in a network of databases. So I don't have a lot of time to go into details, but basically what we did in LEGEND is we performed an analysis for where we compared drugs, classes, and combinations of drugs and combinations of classes, all of treatments for hypertension. For all those comparisons, we looked at 58 outcomes of interest with well-defined outcome definitions. So in the end, we had over 500,000 research questions that we wanted answered. We answered them using a new user code design, using large-scale propensity scores and proper outcome definitions in a network of 9 databases across the world. And in the end, we produced 1.3 million effect size estimates. So these are causal effect size estimates for hypertension treatments. And so we not only produced these estimates, but each one of these estimates comes with a set of statistics and diagnostics. So just to highlight, I'm picking one here, lisinopril versus hydrochlorothiazide for the outcome of angioedema, where in the CCE database we observed a hazard ratio of 2. So we not only produced that estimate, we also produced a set of statistics that we think would be the ones that you would expect to see in a, for example, manuscript. So we actually, to drive that point home, we created an automated procedure that takes all that data and puts it in a report. So in that report, you, for example, have a description of the methods used to estimate this effect, the attrition diagram, a propensity score distribution, and a covariate balance, which would get to Darren's point, like, is this actually a valid comparison that we're making, the Kaplan-Meier plot, characteristics of the study population. And so we basically have that available for each one of those 1.3 million estimates. And so imagine that you have a signal generation system that provides this level of detail for every question that you're trying to look at. So we developed Legend with a specific use case in mind, which is very similar to how one uses the literature, in that we imagine that somebody might have a question, like a doctor or a patient or a regulator might have a question. For that specific question, they would check the Legend results and see what answer comes up. So that's one hypothesis with one answer, so no multiple testing. But there's nothing stopping anyone from using that same data to do signal detection. You could, for example, rank order by the lower bound of the confidence interval. But when you're doing something like that, of course, you need to do adjustment for multiple testing. And I think I've already heard very good ideas from both Judy and from Bill on how to deal with that. I'm not going to go into that. My point is that it's the same goal, to provide the best estimate. But that does lead me to another point I need to make. And one best practice that I've neglected so far. Well, how good is that estimate? You can do multiple testing, but that assumes that if you're, for example, correcting your p-value, that assumes that p-value is unbiased, that it actually has nominal characteristics. Well, how do we know that that's true? And so one of the things that we've been promoting in Odyssey for a long time is to include controls in your analysis. And so, for example, what I'm about to show you is a study where we included 200 negative controls. And a negative control we define as an exposure and an outcome where we know that there is no effect, and therefore the hazard ratio or the odds ratio should be equal to 1. And so I'm showing you here a plot. On the x-axis is the effect size estimate, the relative risk or hazard ratio in this case, and on the y-axis the standard error, which is the width of the confidence interval. And so the higher you go, the more uncertain you are. And everything below those dashed lines has a confidence interval that includes 1. And each one of these blue dots is one of these 200 negative controls. And so here you see the results for a cohort method analysis using a one-on-one propensity score matching. And you see that actually exactly what you would like to see. 95% of these blue dots are above the dashed line, so their confidence interval includes 1. So that's actually pretty good, what we call coverage of the 95% confidence interval. Compare that to this other approach, a variant of the nested case control design that we applied. You see that actually a large number of these same negative control hypotheses are below the dashed line. So certainly this method has much more bias, but on the flip side, it does have a lot more precision. These points are all a lot lower, so you do get much more information on the effect size. And so some methods are a little bit more in between. So I'm showing you the multiple self-controlled case series, which has both relatively small bias and relatively high precision. My point is that we need to have those estimates to get an understanding of how well the method works. With these estimates on negative and positive controls, we can evaluate performance. For example, computing the coverage of the confidence interval, measuring type 1 and type 2 error, area under the receiving operating curve, mean squared error, precision. All of these, I think, are important to have these metrics when we develop a signal generation system. We can actually trade off some of these characteristics. For example, type 1 and type 2 error can be exchanged by varying the threshold that we use on for example, the p-value. And we can apply empirical calibration, which is a process that we've developed in the past, where you can, for example, ensure that the 95% confidence interval has 95% coverage, but which comes with a trade-off that you probably lose precision. My main takeaway points is, well, the goal that we have is to reliably estimate causal effects, and that's the same whether we test one hypothesis at a time or many, which you might call signal generation. In both of those situations, we need to apply best practices even though we're doing them at large scale. So confounding adjustment, proper outcome definition, sensitivity analysis, study diagnostics, multiple databases. And we should always include negative and positive controls to evaluate and measure the operating characteristics of the system that we're using. And we can express those results using multiple metrics. And with that, I'm at the end. Thanks.
Thanks. Nicholas?
Great. All right, thank you. So we're moving from a setting in epidemiology where we're doing confirmatory studies, and we think carefully for a long time when we design a study specifically for one hypothesis that we're interested in. So we can adopt the settings and the parameters to that specific drug and that specific medical event. And then ideally, I know it's not always the case, but ideally we should be able to run the analysis and then just basically report and conclude from that. Into this other setting, which we've talked about throughout the day today, where we do exploratory analysis, and we have to think very carefully, maybe even more carefully, up front, because we need to design a methodology that will work for signal detection across either many medical outcomes or many drugs or both at the same time. And this is what we're talking about here. But also on the back end, we need to very carefully scrutinize those results and look at them in hindsight, because now we have a specific drug event association in front of us, and we need to look at the analysis that led to that. Like we talked about before, maybe we want to adjust the analysis, we want to look more closely at other possible explanations for what we've found. So today I want to talk about both those areas, both what we've done in our own research in terms of improving the methods for screening, for doing the first step, the exploratory analysis, but also the second step, where we look back at the data and we see how can we actually assess whether this is something that we think is a real signal. So if I start on the back, one of the technologies that we have developed is this statistical graphical visualization of temporal patterns in longitudinal data. We call it the chronograph, and it visualizes the temporal association between one drug of interest, in this case it's nifedipine, a calcium channel blocker, and one medical outcome, in this case it's ankle swelling. So if you look at this graph, you see, and maybe we start with the bottom panel, if you look at the center, this is a cohort of patients starting nifedipine, and the center of the graph where you see the asterisk is the day of the start of treatment with nifedipine. The bar chart represents the number of patients who had a recording of ankle swelling on that day, so in this case it's about 50. If we take one step to the right, we see the number of patients that had a recording of ankle swelling in the first 30 days after they started treatment. And if we take a step to the left, it's the number of patients who had that recording in the 30 days leading up to the start of treatment with nifedipine. And then we can go all the way to the end, which is about 3 years into the future, and all the way to the left, which is about 3 years back into the history. Now, we don't just look at those observed counts, because we know there's an issue in looking at observational longitudinal data where we don't have the same likelihood of capturing or ascertaining medical events in different time periods. So, for example, there tends to be, not just because of censoring, there tends to be a tendency to have more recordings close in time to the start of treatment. So this could, for example, represent clustering of doctor visits in time. So if you've seen the doctor 1 month, you may be more likely to go back another month or the subsequent month. Whereas if you haven't had an interaction with the healthcare system, at least in the UK primary care data that we're working on for some months, that may be less likely. We also may have asymmetries around the start of treatment, so that we see more recordings generally for a specific outcome after you start treatment than before. So we use patients who start other medicines as a background, as an external comparator, and that gives us an expected value for each of those time frames, and that's what you see as a blue line next to that bar chart. If we take the observed value for each time period, we divide it by the expected count, and then take the logarithm, so this is a bit complicated, that brings us to the top panel, and there we see basically it's a better way of visualizing the associations in each time period over that whole spectrum. So basically, if we look at these patients here with nifedipine, they're slightly less likely than patients in general to have ankle swelling before they start treatment. And then we see a quite dramatic increase just after they start treatment that subsides a bit over maybe 5 or 6 months, but they go back not to the baseline they had before, but a slightly elevation. So that tells us quite a bit for this known adverse drug reaction. Is we don't actually see any selection away. We don't see any really avoidance pattern that it drops just before. So it doesn't seem to be a selection away of patients who just had ankle swelling. We also see a transient effect, but also a more persistent effect over time. And this will be useful. And these are the kinds of patterns we would like to find in our signal detection activity, and then look at these chronographs. But to get there, we have the other challenge, which is the first phase of the exploratory analysis is to identify those interesting chronographs out of the millions or tens of millions of possibilities. So we've used for that purpose essentially a self-controlled design where we use patients as their own controls. So we're looking at a specific risk window and comparing to the time before typically they started treatment. But we don't just do that because we know we have this greater tendency maybe to capture medical events in certain time periods. We want to adjust for that. So we also use this comparator group of other startup treatments to calibrate that self-controlled cohort analysis. So we basically divide by that observed expected ratio that we would see in patients starting other medicines. And that gets us to this observed expected ratio that we're interested in, which is essentially now the observed number of events in the risk window of interest divided by a product of the expected, the number of events in that control period multiplied by the observed expected ratio in the comparator. So that is a way to get to an observed expected ratio that's essentially self-controlled, but also just for some of the general patterns that we tend to see in data. We use a statistical shrinkage, which is similar in nature, I think, to the one that Bill presented, but it's much more simple. So we don't do any borrowing of strengths. It's just a simple way to moderate our observed expected ratios towards the baseline assumption of one observed expected ratio, where the observed equals the expected. So we basically add a constant to the numerator and to the denominator, and that will shrink the estimates, moderate them to the null hypothesis. A key design choice for us has been not to use a single control period, but to actually use multiple control periods in parallel. We've found no single control period that will work ideally on its own. So, for example, we found that it's important to have one control period that's immediately prior to the start of treatment with the drug. So in the 30 days leading up to the treatment. This is useful in particular to eliminate associations that would otherwise be confounded by the underlying disease or the severity of the underlying disease. However, we also need a control period we've found that's situated quite far before we start the treatment with the drug of interest, because otherwise we're vulnerable to highlight false associations that are driven by reverse causality. So, for example, contraindications. If you have a medical event that will impact negatively the future likelihood of starting treatment with that drug, you will tend to see a dip in the chronographs before you start that treatment, and that could then lead to what looks like it's an interesting association, but when you actually compare to the baseline, long before that goes away. So we use those two control periods in parallel, and also a control period that's actually the day that they start the treatment, because we've found that even though in principle that could be acute events, most often it's likely to be related to the underlying disease or the indication for treatment. So basically we run 3 different analyses, and we require there to be a significant association for that risk window for all of those control periods. Great. And the method has been evaluated in the OMOP experiments and some other comparative experiments, and this is just showing you the results of the 2012 study, where the blue axis marks this self-controlled cohort, the calibrated self-controlled cohort method. And you can see that it performs relatively well compared to most of the other methods. So it's near the top or at the top of, in a few cases, against each data source that it was evaluated in. But we also see that the performance is far from perfect. We're nowhere near the AUC of 1 that we would like to see. So that we need to bear in mind as well. So I want to say just a few words about, I think, what has been the most interesting study perhaps we've done with this methodology is when we did try this in the wild. So this was part of the PROTECT project in Europe, where we actually mimicked how we would do signal detection in reality, similar to how we do it when we screen spontaneous reporting systems in pharmacovigilance. So we had 7 assessors. They would be epidemiologists or safety physicians, and a few of them were both, that would go through a random selection of highlighted drug event associations for that would have been picked up by this method, and then go through the motions that you would with a safety signal from spontaneous reporting system. So they would look at the label, is this something known? And if not, they would go and look at it, do we see other alternative explanations, or do we think this is something that would merit an in-depth evaluation? So we found that 25% of the assessed combinations were already known, so that's reassuring in a way. Many of the things that come out are known adverse drug reactions. It's lower than what we see in spontaneous reporting systems, which is more like 50% typically. We also see that 3 out of 4 combinations were dismissed because there were alternative explanations. I'm just going to very quickly show you 2 reasons. There are many different reasons for this. One that we found was residual confounding by the underlying disease. So we're looking at here an antibiotic, gentamicin, and eustachian tube disorder. And you see the pattern of an increased registration around the time of start of gentamicin treatment, both before and after. So we think this is just still there. And in principle, it should be possible to eliminate. The following one, I think, is much more difficult. This is hyoscine and endometriosis. So hyoscine is partly used to treat stomach cramps, and this could be a symptom of endometriosis. So we think this is a protopathic bias, and what's going on is that patients are having stomach cramps, being given hyoscine to treat this, and then they don't have the diagnosis until later on. And in some cases, it's quite far after they started the treatment. So in summary, we don't think there's a one-size-fits-all, but we have to try to find the best possible size or the best possible sizes that we can to get the best possible fit. And we should be realistic about a manual review being a necessity and try to find the best possible ways to do that in a sensible way.
All right, thank you. So it kind of sounded like you all described the same thing, just in different ways. But what I really want to know is, like, which one's best? And we're not supposed to do that for this session. So the question is really, you know, what are the characteristics of what you're trying to do that make each of your techniques sort of stand out? So each one is best at certain things and in certain contexts that you're trying to implement them. So if one was trying to design a pilot study that would sort of test these different approaches that you all described, you know, how would that be designed? What sort of things would need to go into that study that give, you know, different variations of conditions whereby you could actually look at these different techniques and sort of get a better sense of when one or the other might be more appropriate for a given context. So any thoughts on, like, how would the FDA sort of think about all of these different techniques or start testing these different techniques?
I think some of them are, like Martin said originally, very orthogonal. I mean, so there's no reason you couldn't combine, for example, the tree-based scan statistic with what we've done. I mean, that is a way to find good groupings of medical events. You don't have to specify the granularity up front. So that could definitely be run with our observed expected analysis that we're doing. So they are not choices that you have to choose between. I think similar with Bill's approaches. I mean, that's another way to handle the different possible granularities in a way with borrowing strength. And that could also be done. And again, I don't think there's a choice between these methods. They're really like we heard originally, like the Lego pieces you have to fit together and try to find the best way.
Any other?
So, Greg, yeah, I just wanted to, I think Michael talked about it this morning, but Martin and Nicholas and then also Jesper Hallis are all co-collaborators on a protocol that is actually on the website and is available for anybody to download and provide comment. We're in the 30-day comment period where it's going to take the same data set, which is going to use a self-controlled design, and then actually do all these techniques and sort of look at what are the differences. I don't expect that actually there will be a lot of differences, frankly. I'll just say that ahead of time. I don't expect that there's going to be, you know, wildly different behavior because there are a lot of elements that are very, very similar. Really, it mostly comes down to multiplicity control more than anything else. And so I think that's, you know, it's already out there in some respects, and so we can continue to do things like that as we go forward.
Martine, you presented, like, you know, 500,000 plus research hypotheses immediately tested. So could the techniques that Judy talked about, so that tree scan and then the other techniques that were included, could that easily be incorporated into the sort of approach that you described?
Yeah, so I think one of the main notions that I try to get across is this idea of including controls in your study as you run it, but also as you're trying to evaluate it. I don't think there is a reason why you couldn't do that. So if you want to compare all these methods or how combinations of these methods work, you really do need a proper evaluation. And so I've seen the protocol. What I missed in it is so there was a simulation component and there was one component where there was an evaluation basically without specifying what the gold standard is. So I would like to see a component added where we do have a pre-specified gold standard using real data and real examples.
Okay. Go ahead, Nicholas.
Maybe just to add to that, I think the difficulty with the gold standards, I think many times, is that if we get them at all, it's always the already known adverse drug reactions. That's what we can maybe agree on sometimes. And of course, what we're looking for is the unknown in a way. So I think that is the argument for the more prospective approach. That is I think, the approach there and what we did there. I think you need both, but I think there is a risk if we look at the already known adverse drug reactions, because they will be different. There will be risk minimization behavior, for example, that we might not see with the unknown adverse drug reactions. So I think that is the argument for that. But I can see, I mean, reproducibility and all sorts of other issues.
Well, so you've actually written that comment down already in a paper, so I'm well aware of that, and actually I completely agree with it. So the path that we've taken is that does not apply to negative controls, so you're actually free to go and use those. And then we use a process that is basically enhancing negative controls to become positive controls. It's a mixture of real and simulation, which I think might be helpful. But certainly just a set of negative controls might be good to pre-specify.
Yeah, I absolutely buy that. I think that's, use of negative controls, I think, like you've suggested and other people have said, is very good. Just as a baseline to see, are you highlighting things you shouldn't be highlighting?
Yep.
Okay, to Judy and Martin, given your knowledge of the Sentinel system, the way it exists and the way it's has been developed, any implementation challenges to, or some of these techniques that we just discussed more easily implemented into the process versus others, or are they all sort of just depends on what you want to do and they're just as easy to implement?
I think that for many of these techniques, they can be implemented in a distributed database setting. I think the key feature there is that we try to minimize the transfer of patient-level data. And what I mean by that is a data set where there's one row per patient. And so we don't combine patient characteristics from our different data partners. We try to find a way that's maximally privacy preserving. And so depending on the different technique, there can be adjustments that are made to make that available. So, in doing this pilot, we sort of went with we talked with each of the innovators and said, okay, this is the data set that we're going to bring back. Is that going to be able to be adequate for your needs? And we actually could do it without too much problem. That, though, is very particular probably to a self-controlled design where you have an automatic control for time invariant characteristics. And so, in that sense, it's easier to be privacy preserving than in other types of techniques.
Any other comments from the panelists?
Well, I just wanted to agree that a lot of times these are not alternatives, but they can be mixed and put together. And as far as the multiple comparisons controls, I also agree that the using negative controls is sort of a good idea. Using the positive controls and using an assumed gold standard is tricky. The work that I've done with the with Bayesian shrinkage as a multiple comparison method is, with simulations it works beautifully, but real data has problems just primarily because of so many unknown effects going on that you really can't adjust for.
Okay, Anna. Your mic may not be on. Adam, you want to check that?
We are working with messy data. For example, clinical laboratory data is not included most of the time because this is an important part of our medical assessment. We have more patients in the OMAP database in the US that I think 3 or 4 times more unique patients than the population in the US. Then we need to accept that we are I think that we need to accept that we are dealing with messy data. Then I think that to have a very good experiment, we need to select the best data resource, the one that we can have traceability to the original terms, and hopefully we will understand that we need to create an interoperable environment for medical data, because this, we are constantly reconfiguring the data and the tools, and this doesn't help patients. This creates a lot of medical errors. Then I think that you can please consider the data also, the quality of the data, because if not, we are not working in a real situation.
Great. Any comments?
Yeah, so I completely agree that data quality is very important, but I am going to disagree a little bit. It's never going to be perfect. So what we also need to focus on is not just data quality, but evidence quality. So some sort of an evaluation, that's basically my main point, is an evaluation of the evidence might actually be more important than, well, we always need to strive for more data quality, but in the end, we need to make sure that it answers the questions in a reliable way.
Yes, Amazon is getting into this type of work, and they understand perfectly well that they cannot work without having totally interoperability. Then I am hopeful.
Well, I think with data quality, garbage in, garbage out, it doesn't matter what great statistical method you have. If the data is bad, the analysis is going to be bad. So I think that is key. And of course, there are different aspects of data quality. If we go back to evaluating these methods, it's useful to do simulations. It's useful sometimes to have a negative control or if we have a gold standard. But ultimately, I think the only reliable way to evaluate and test this method is to use them in a real-world setting where we don't know what the answer is, and see what comes out. If we run a method and it's sometimes very clear that it's not working, or if it is working. For example, we did a self-controlled tree scan for 2-year-old recipients of a vaccine, and it doesn't work to do a self-control for 2-month-old because basically you're comparing diseases that somebody is 2 months versus 3 months old, and that's very different. If you compare somebody who's 50 years and 6 months versus 50 years or 7 months, they have the same rate of disease, but not a 2 month and a 3 month. So that was a situation where that didn't work, and it was pretty obvious. We have done other situations where if you look and you don't get too many false alarms, that's the good sign at the same time as you detect things that are very rare. So we used the tree scan, for example, for PPSV vaccine, pneumococcal vaccine, and in adults, and we found that there was some rash. That's natural because a lot of vaccines gives a little rash at the injection site. And then we found that there was an excess of spleen surgery after PPSV vaccine. So what's going on there? Do people who have the vaccine suddenly develop spleen problems and they have to go into emergency surgery? No, that's not the case. The case is that if you are scheduled to have spleen surgery, you're getting the vaccine as a preventive measure a week or two beforehand. And of course, the statistical methods can't differentiate that, so it's clearly a true signal in a sense, because it's causal even. It's causal, but except backwards in time. So that's sort of in a way, it's a kind of a gold standard, because there is a causal relationship that the surgery causes the vaccines before surgery. So we should pick up, a method should pick up those kind of things. But of course, it's nothing we worry about because we know what's causing it. So by running these things in a real-world setting, we'll learn a lot about it. We'll learn where the strengths and the weaknesses are, and we use the clinical knowledge that maybe I don't have, but other people have, to sort of judge if the results are reasonable or not.
The problem is this leads to a lot of uncertainty. and uncertainty, not knowing exactly if the signal is a signal or if it's not, this leaves a lot of uncertainty, and then the uncertainty leads to paralysis because we don't know how to interpret the results. We have seen in war situations, some people stay, some people leave. There's too much uncertainty. And I feel like using negative controls is a must. Is very important.
But by that same token, I think that using negative controls is a good idea in theory. In practice, getting people to agree to a negative control creates its own kind of paralysis, where people can't agree and then it stops the actual analysis going forward, which is why a simulated environment where you actually break all of the connections and then force there to be a negative control is a much more sort of streamlined way to do that.
But that's why we need complete traceability to the original data, and we need to incorporate clinical laboratory data, make it interoperable, because if not, we are not using one of the most important pieces of information. But thank you.
Yeah, thanks. So we are running out of time for this session, but we'll do a quick question or comment from Stephen and then Hector, and then we'll go into our last break of the day.
Can I just ask the panel what they think about the hierarchies? Because it seems to me that that is definitely the way forward, and we need hierarchies both for medicines and for adverse events. But how we group them isn't clear, because MedDRA is not good enough. You get hypertension and hypertension in there, and you need to think more carefully about things that are alternative terms where they would not cluster together in a single report. They're mutually exclusive. And things that are clinical syndromes that go together, and they are subtly different. And it seems to me that we haven't thought our way through that. And similarly for drugs, because I'm not always interested in things that are used for the same indication. I may be more interested in mechanistic things, as was suggested by others. But for other purposes, I am interested in things that are used for the same indication when I want to do analysis. So there are multiple dimensions.
Yeah, great questions. Any quick, fast
So I think
comment?
that is very, very important, critically important, and to have that hierarchy for the TreeScan methods, that's very important. I don't think there is one correct hierarchy to use. So, because outcomes can be grouped in different ways because they're written in different ways, and same with drugs, as you say. So, when using TreeScan, one approach that we have started to use just a little bit, but is to use multiple trees simultaneously. So we sort of have maybe 2 or 3 or 4 hierarchical trees on the outcome, and then we sort of scan first one, and the second one, and the third one, and the fourth one. And from a statistical point of view, that's not more complicated than having one tree. Computationally, it takes a little bit longer, but not usually too much.
Nicholas? Or let's go Bill and then Nicholas.
Okay, yeah, I think Stephen raises a very important point, that the notion of whether two things are close together in a hierarchy gets confused, because there's the notion of granularity, which kind of sort of says alternative views, alternative observations that don't occur in the same patient are close together if they sort of show up in the same situation. In many other cases, if you're trying to study drug interactions or syndromes, you really want to focus on things that typically happen to the same patients. And they're just two different concepts, and you'll have to use different statistical methods to find each one.
Nicholas?
Yeah, I think that's sort of the point I want to make. So
Okay, fair enough. Hector?
I will give you a hypothetical example. Let's assume I have a study on herpes zoster effectiveness, and then I use negative controls Some of them predict frailty, like hip fracture, and some of them predict healthcare bias towards healthcare utilization, like use lipomas. The lipomas are 10% higher in vaccinees, and the hip fracture are 40% lower in vaccinees. Those mean different things. And in this particular case, I could argue that I don't care about hip fracture. I only care about lipomas because that is the bias I'm looking for, healthcare seeking behavior, because I'm looking for outpatient consultations for herpes zoster, not hospitalization. My point is, I do love to use negative controls, the more the merrier, but it's not so simple. If you don't have an a priori hypothesis, you can get false results, as maybe I have or could have done in a hypothetical example.
But wouldn't you want a causal effect estimation method that's robust to both types of bias?
It's a long discussion. I would love to take it.
Okay. Okay, this was a great discussion. We're going to go into our last break of the day. We'll get, if we start trying to maybe get back at 3:05 and maybe make up some of the time that we're over for our last session of the day, which will explore a lot of these ideas from a range of stakeholders. So go ahead and take your break. Come back at 3:05. Thanks a lot.
Back to today's final session. This session is on stakeholder views on implementing signal identification in the Sentinel system. We've already heard some views on how this can be done in the last session, building off methods that Sentinel and other organizations have been using already on issues related to signal identification and strengthening. In this session, we want to make sure we are broadening the perspectives on these topics as much as possible. Again, this is a very important stage for FDA's efforts to continue to advance the approach to signal identification that we've been discussing today. So there's a good chance to hear from those of you who are here, but I'm very pleased to be able to begin with our panelists. First, Andrew Bate is Senior Director and Epidemiology Group Lead for Analytics for Worldwide Safety and Regulatory at Pfizer. Kenneth Hornbuckle is a Research Fellow in the Pharmacoepidemiology Group, which is a component of the Global Patient Safety Organization at Eli Lilly and Company. Miriam Sturkenboom is a pharmacoepidemiologist at the Julius Global Health Group of the University Medical Center Utrecht in the Netherlands. Jennifer Nelson is at the Department of Biostatistics and is a senior investigator at Kaiser Permanente Washington Health Research Institute and an affiliate professor of biostatistics at the University of Washington. And finally, last but not least, Bill Crown is the Chief Scientific Officer at Optum Labs. I want to thank all of you for being here. We're going to hear from each of them with perspectives on implementing signal identification in the Sentinel system, and then we'll have some time for discussion. So if you all have comments to add on how FDA might best move forward on these efforts, this is the right session to do it. And Andrew, I'd like to start with you. And as with the last session, feel free to stay there or come up to you all.
Thank you very much, Mark. Thank you very much, Mark, and thanks to the rest of the organisers for the invitation to present at this important meeting. It's really fascinating to see how the agency is exploring the potential use of the Sentinel network for signal detection. And in my comments, I'd like to try and do three things, really, which is, first of all, comment a little bit more on some of the methods we heard presented in the previous session, to give some context on some of the kind of emerging issues and challenges, particularly around the design choices, linking them to our experience with methodology pilot testing at Pfizer, and then thirdly, wrap up by giving some thoughts on how this all informs potential future directions. So, as we heard, there's a range of different approaches that have been presented in this meeting, and indeed there are many other methods that have been proposed for signal detection in this real-world data, these longitudinal observational databases. And I think this really encapsulates the challenges with signal detection in the data, that there's many different ways of approaching it and a myriad of challenges to tackle. Martin Kulldorff presented a great overview of many of those design choices and the core challenges behind them. And I'd like to focus on a few of the specific issues that came up then in those four methods that were presented. Before saying that, though, I do think we need to really think about the nature of the data. We've talked quite a lot about it today, but I think it deserves emphasis to say that we're dealing with data that is a secondary use of the real-world data, and lots of the thinking is informed by how we approach signal detection in spontaneous reports. But we have two fantastic elements of spontaneous reports that have made it the cornerstone of signal detection for so many decades. Specifically then the clinical suspicion, and increasingly patient suspicion of a potential link between exposure and outcome. And secondly, the data that is communicated in order to support that suspicion. So it's not just that there is a suspicion, but spontaneous reports that we can really act on have a lot of rich data associated with them. Of course, one of the challenges, as we've heard about today, is many spontaneous reports don't have much data on, and that's a major problem, and a limitation with the spontaneous reporting systems. But when we think of real-world data in the secondary use, in formal epidemiologic studies, we have to really, as we plan and post and discuss hypotheses that we want to test, we have to really think not only about the hypothesis we want to examine, but the nature of how that will manifest itself in recording in longitudinal observational databases. The comparators, the risk windows of relevance, enrollment time, washout periods, and other elements we've heard about today. So we carefully think about those issues from the context of hypothesis testing studies. Now, as we've discussed a lot today, that's not possible in the context of hypothesis-free signal detection, other than in broad brush strokes. So the clinical epidemiological review is essential. And I think that's one of the aspects that we need to think about, is not just how quickly and effectively we highlight alerts, statistical alerts as the term was used earlier, but also then how readily does that alert allow us to then further investigate it and that's a key challenge for us as well. We've talked a bit about coding, but I do think it does really encapsulate this challenge of secondary use. So we have MEDRA, a terminology for spontaneous reports designed specifically to facilitate signal detection. As we think about, and certainly this chimes with our experience, it's a real challenge to think about terminologies in these different real-world databases, which vary across databases as well. Do we look at individual codes, groups of codes? How do we define them a priori? To what extent can we vary our groupings of terminologies? The work from Dr. Marrow around the tree scan is very interesting in how they think about the hierarchical terminologies. And I did want to emphasize, though, that that's one of the challenges, of course, is how that tree is constructed. And it's very interesting to hear this idea of looking at different trees. I think in terms of understanding how we highlighted certain issues or didn't, understanding the variability that we see across different trees and different ways of structuring terminologies is very important. Builder Michel talked about Bayesian borrowing and these hierarchical models, and that's a fantastic idea, and we've looked at it, or groups have looked at it in spontaneous reports and clinical trials. And as was mentioned previously, though, the challenge is, what is it that makes things clinically, semantically similar? What is near, what's far? How do we group things? Is it reasonable to think about, for example, in MEDRA, system organ classes as our groups of interest? And I think that's an area that's very interesting, but more work needed to further pursue that, as with the tree-based scan statistics and how we best leverage the different trees. Martin Schumi talked about the Odyssey experiment, and it's very exciting from an informatics perspective to see that ability to generate those sorts of outputs. How, though, when we see these visual diagrams with tens of thousands, hundreds of thousand points, how do we go from that to facilitating objective, systematic, transparent review of the outputs? So that's very interesting foundational work that I'm really interested as well to see how that further develops. Nicholas Naren talked about the chronographs, which I think are very appealing in the way they try to look at variable time between exposure and outcome and persistence potentially. What we found, and we looked at all of these methods, or many of these methods and some other ones internally, was that it's very visually appealing, the chronograph, but obviously we can't review all of them one by one. So we did some work and have put a disturbance algorithm on top of the chronographs to try and facilitate where to focus our clinical review. An autoregressive integrated moving average model that looks to model the behavior of the chronograph and then look for interesting disturbances from it, not to ignore and throw away, but instead to say these are focus areas that might represent emerging statistical alerts that we then review further. So lots of really exciting methods work going on. I think, though, and this did come out earlier, but I want to re-emphasize it, we still have limited evidence on the comparative performance of these different methods, particularly in terms of important signals prospectively detected in real-world data. I think that's because this is a really challenging issue, but we certainly need to do more testing. Not just do we find the right things true and early, and how do we avoid false signals, or false alerts, but also how do we facilitate that conversion from statistical alerts, to use that terminology, to signals? How do we have clearly transparent understanding of why an alert was created, and the potential limitations with it or not so, and then so that we facilitate the ability to highlight for consistent clinical review and epidemiological review for further consideration. Therefore, with all of these issues, I think more collaboration is really very much essential. We need to look at making reference sets available. We need to discuss the strengths and limitations of them, whether that be simulated, real-world data sets, prospective signal detection, as well as doing retrospective and check that we can find things over time. We as an industry, I think, can contribute a lot in the collaboration here because of the fact that we tend to have an international focus and therefore have access to huge numbers of real-world data from different countries with different issues. I think that as well as many different databases here in the US, from oncology databases to other, and really understanding the amount of difference in performance across these real-world data sorts, I think gives lots of insights into, if you like, the fragility of some of these outputs and findings, and I think we can build that knowledge into how we further refine them further. I agree completely, there's no one perfect method, it's about developing a suite of methods. I would just name-check the taxonomy report from Josh Gagnon and others that was presented several years ago on different epidemiological study designs for different types of exposure and outcome. And I think we could therefore think about how we might look at a similar type of thinking around the different types of exposures and outcomes that we need to detect in this signal detection. Transparency on guidance around, we have lots coming out in terms of reporting of how we do real-world data studies, like the ISPO task force on healthcare decision-making for real-world evidence. There isn't much specific to signal detection in real-world data. There's CIOMS 8, but that's 10 years ago now. I think there's real need for us working together to look at guidance of how we can best progress this science and really work together in terms of assessing real-world performance of these methods. So to conclude, I think we need more capability development. I think there's some very exciting methods, all of which have strengths that we've heard about today. There'll be more to come. We need to really look at performance characteristics and methods. We need to think holistically. We need to not just think about real-world data or spontaneous reports. Yes, they have a complementary role. What is that complementary role? Spontaneous reports and how we handle them is pretty much how we've done since the 60s when there was no alternative. We have a responsibility to revisit how we think about spontaneous reporting as we get more capability of doing signal detection with other approaches as well. So collaboration with multiple stakeholders and methods development through public-private partnerships will be very powerful as we strive to contribute ever more effectively to our ability to ensure patient safety through the best possible signal detection we can do. Thank you.
Thanks very much. And next is Kenneth.
Thanks, Mark, for the introduction. I'd like to thank also the Planning Committee for the invitation opportunity to participate on this such distinguished panel. It's nice to be with you guys. Also, my main goal here is, first off, though, I'd like to commend FDA for exploring this area of drug safety in terms of signal detection, trying to also leverage the Sentinel network. Also, this has been a great discussion at the workshop. I really enjoyed the deep discussions and the number of topics that have come up. My main goal, though, is as a industry representative, is to really share with you a case example of how we have been using the electronic large healthcare data to help supplement doing signal detection with these large databases. So I really want to give you highlights and lessons learned from those activities. As well as, before going to that area, I'd like to just make a few general comments. First off, I think signal detection is a very, very essential component for any pharmacovigilance system. And like any other system, when you want to add additional activity, it's important to really determine and take a systematic approach. Okay, what are we trying to achieve? What are the objectives we're trying to achieve? What's the research question? Are we trying to look for, in this case, it's really not targeted events. We're looking at hypothesis-free events, all comers. And it's not the other area that's important, it's too, are we seeing an increase in severity of adverse events as well? The other thing is, in order to frame this, it's a lot of complexity. A lot of complexity with the different exposures, with the different outcomes. You have multiple types of exposures in terms of single point of exposure, multiple exposure, acute exposure, chronic exposures. Also with outcomes, you have acute transient outcomes, you have chronic outcomes. I mean, you have a plethora of different types of exposures and outcomes that are important in the study design that needs to take into consideration. In terms of data sources, as scientists, we all know that the data sources have strengths and limitations, and we have to leverage these data sources on their strength and help to minimize some of the limitations. Similarly with the methods, we have to also apply our knowledge to the strengths and limitations of the methods as well. I use this to help frame, and as Andrew just mentioned, in order to come up with an alternative or a solution, because of the complexity, there's really not a one-size-fits-all. I see this as a suite of opportunities to explore multiple methods for those outcomes that are applied to that method, as well as the data sources that apply as well. Also, too, I just add that the spontaneous reporting system, it has its strengths and its limitations. Okay, we want to build upon those limitations and supplement them with activities like signal detection, but also build on its strengths of large national coverage, global coverage, as well as identification of rare events. However, using these large healthcare administrative databases, at some point they'll be able to as well assess rare cases as well, which we couldn't do back in the early years, decades ago. So these are, given the limitations, these are things that we have to consider. Now I'd like to share with you our experience. We spent a number of years really assessing this whole space. And as an epidemiologist and a scientist, you normally start with a feasibility assessment. What is the appropriate approach to do this? And so what are the data sources, what outcomes? And we actually decided to do a hypothesis-free signal detection. We started as a pilot. And over time, we learned from that. And then now we have a fully implemented procedure in place with appropriate guidelines, also roles and responsibilities, as well as governance in place as well. So what we have is, for each molecule, we have a safety surveillance team. That safety surveillance team consists of a physician, a surveillance scientist, as well as an epidemiologist, as well as members from my case management group who process individual cases. We meet on a periodic basis to discuss and look at all the available data to monitor and identify any new safety concern. This group, the safety surveillance team, is responsible for then bringing on this activity, writing a protocol, and following all the procedures as well with this. So in terms of data sources, right now we're using one single data source, a large healthcare database. The method that we're using is the high-dimensional propensity score method that was developed by OMOP. Many of you may be aware of that method. One of the nice features of this method is that it automates the selection of covariates to include in the model for propensity scores. The model that we use is a stratification approach. Some of you may be aware of our approach. We presented this at multiple times at the International Conference for Pharmacoepidemiology, ICPE, so some of you may have been tracking and following this as well. In terms of the scope, we only apply it to newly marketed drugs. So it's not the plethora of portfolio of drugs that we have, it's only newly marketed drugs. And then those products must have a sufficient number of cases or patients who have been prescribed that drug in the database. The number that we use is in excess of 1,000 cases in terms of, looking at the opportunity to start exploring using this approach. And also, it's important, too, that I think somebody else mentioned, not all products would be able to apply these methods or this approach, particularly if you have small numbers in the databases, and we're seeing this with many of the oncology drugs. So oncology is a unique area. How many have heard that oncology is unique? And they are unique because of the small numbers, so it may not apply to oncology drugs. I mentioned governance. We do have approvals. We have, in order for a team to consider this, they must meet that threshold of having, the drug must be on the market for at least over 2 years or so, or have a sufficient number of patients in the database. They write a proposal and a protocol, and it goes to several of our senior medical directors who then will approve that. And so then the frequency. The frequency, as I said, it was over 2 years on the market. You must have a large number of patients in the database. And then in terms of frequency, we typically, as a guide, you can run the model every time there's a database refresh, or the surveillance team, they document everything as well. They can determine if they need to do more or less types of runs as well. Also, the team can document when they see no value, this extra activity is not providing any additional value. They've already understand the safety profile of the molecule because it's not really by definition a newly marketed drug anymore. It's been on the years, over the 4, 5, 6 years or so. So they document that as well. So in terms of the methodology, as I said, it's a high-dimensional propensity score with stratification. Starting with the criteria of greater than 2 minutes. I started then with greater than 1,000 patients or so. Also, we use multiple comparisons. We talked about comparison. The comparisons that we use are drugs within the class as well as outside the class or in other classes. So we use multiple comparisons. I think that's important as well. That safety, the number of events that must be in the outcome that we use has to be 5 or more for the outcome. And real quick, we have a threshold as well, predefined threshold. Must have a hazard ratio of greater than 2, confidence interval of greater than 1 on the lower bound. Then all those drug event combinations that meet the threshold, they're reviewed by this team, and they determine whether or not this is not a signal, they close it out, or it may be a potential signal. The other thing, if it is a signal, the team would just document, just like we do with normal spontaneous. You find a signal, it will work through the process with the appropriate assessment, documentation, as well as review. So our actual experience is that it is not, because these are newly marketed drugs, the volume has not been huge. We have not seen any major excessive number of drug event combinations that met the threshold. The ones that have, they've been expected. They've been associated with disease indication. So nothing that has not been manageable. The multiple comparison has been helpful, so you can use that to help really assess the impact of false positive and false negatives. It's important, though, when looking at the output, to use your clinical, medical judgment, as well as product therapeutic judgment as well, too, and your experience in pharmacovigilance. In terms of next steps, we are applying, so we're saying that high dimensional. We're also looking to add what Nicholas presented, the self-control methodology is side by side with that as well too, and visualization in there as well. And that would be very helpful in really assessing the impact of any potential signal. Clinical judgment is very important, as we said. Remember, this is signal hypothesis generating, not hypothesis evaluation. So causality has not been evaluated. Further review and assessment are needed for causality inferences. This bridges the gap between clinical trials and spontaneous data. It's a supplement to the existing pharmacovigilance activities, and it augments the activities. As I said, it's not a one-size-fits-all, multiple suite of tools as well as methods need to be considered. I think this is also a great opportunity for collaboration. Thank you. Look forward to the questions.
Thanks, Kenneth. And next is Miriam.
Well, thank you very much for the opportunity to speak here. And I have my speech here on the computer screen because of jet lag, I guess. So I don't want to forget anything important. So the perspective I'm bringing here is I'm coming from Europe, and there are not many Europeans here actually, but in Europe we started a project in 2008, actually at the same time as OMOP was starting, and it was called EUADR. It was one of those European Commission funded projects that they give some funding and then it disappears again. So EUADR has ended. It ended in 2013, but I think we learned a lot of important lessons that I do want to bring here. So first of all, EUADR had the scope actually to design, develop, and validate a computerized integrative system that utilized data from electronic healthcare records, but also biomedical databases. So it was not relying on electronic healthcare records alone, but wanted to facilitate the triage process or the refinement process by linking it to available knowledge that we were combining. So I would like to talk a little bit about that. So what we learned there, it's actually necessary to have a fully integrated workflow that where you start with the federated databases, which of course Sentinel has, to have the analytical tools. And I think we, today we have learned a lot about all of the tools that are available. I'll come back to that later. But what we have not spoken about a lot today is the facilitation of the triage system or the refinement. And I think this morning I was struck a bit in that, there seems to be this phase of signal detection and then actually a long period, a potential lag period where there is a process of refinement. And I think what could be improved is actually to build in this opportunity for refinement already at the moment you also do the signal detection. So it is actually possible to run multiple methods in parallel, so you have them available once you do the refinement. But also there are tools now where you can, and Odyssey has a nice tool, I think, to look at whether there are known reactions from literature, from labels, and from, so you can actually automatically, or you can already have knowledge about whether this is a known reaction. You would want to know the strength and all the, of the reaction or the impact actually, and Stephen Evans was talking about that, so you can actually have that available as well. But also, to look at whether, the explanations or the alternative explanations, such as protopathic bias, which we have not spoken about a lot, but which actually, according to my experience, is one of the major problems. Confounding and other biases can explain this. You can actually pre-define designs that would specifically look at those topics. So not all of the designs, and there is a very nice review by Arnaud et al. From last year, from the French group in Bordeaux, reviewing all of the methods that are currently available, and each and every one of them has, there's no size fits all, but they have specific characteristics and advantages and disadvantages. But if you combine them in a good way, you can actually use the different systems to look at the at what component you would like to look at. So there's a nice example of this, which was done by a collaborator of us in Précy Colombe, and she looked at the signals generated in the EODR around myocardial infarction, and then used a suite of methods to actually look at the change of estimate if you use a self-controlled design, or if you test for protopathic bias with Leopard or other tools. And you can actually then assess immediately whether this is a potential signal or not. So there is some experience in putting those things together. I think what is really nice in that paper was a step that I haven't heard about today, which is about the biological plausibility. It seems that we are focusing a lot on expert review to look at biological plausibility. But nowadays we have all of this, potential using biomedical databases to actually create pathways and to see whether, proteins are actually there that can explain this reaction. So I would like to, ask for integration of that system as well, so that at the end, those that are reviewing or doing the refinement have like a dashboard on which they can actually look at all of the available evidence at once and do not have to go back later, so that this makes things faster, I think. Maybe even, and I know it's difficult in this audience, you can automate some of these steps. So I just want to put it there. Now, after the EODR project, what has been done is with some graduate students that were doing research on these, on the associations that were found, they were actually doing some additional work, which was to compare this with, the signals generated in electronic healthcare databases with those from spontaneous reporting databases. And so there has been some work ongoing to compare it with FAIRS data and with VG-based data. And so there are some lessons learned that I think are important. First of all, we all share the conclusion that EHR data will not replace spontaneous reporting databases. They can augment it. So I think our experience led to, when we compared the signals that were found in EODR versus FAIRS or a VG-base, it was clear that some signals you cannot easily find in electronic healthcare databases, whereas you find them easily in spontaneous reporting databases and vice versa. Now, what is it in spontaneous reporting databases that is difficult, especially when you start comparing? They have opposite temporal trends. Like in spontaneous reporting databases, you will find many more reports once there is a knowledge about an association, whereas in electronic healthcare databases, usually the association goes down because of risk minimization activities. So it's not good enough to look at all time backwards if you do this type of things. You need to account for timing and knowledge about the reactions. Spontaneous reporting databases are much influenced by lawyer reports and patient reports that may not have the same quality as serious adverse reactions, so you need to account for that. And we generally concluded that the spontaneous reporting databases are better for the idiosyncratic reactions that may not even have a specific code in the electronic healthcare records. So whereas the electronic healthcare records are much better in picking up frequent conditions that are multifactorial and are not easily recognized to be drug-induced by a medical doctor and therefore not reported. So they have complementarity, and it's important to recognize that. Now, one of the questions that I still have after today that I would like to have, it's good, I think, to think about is how to combine Sentinel-based signal detection as well with the FAERS signal detection. And I think it would be important to realize that there can be disconcordancy between those, and the question will be how to deal with that. I think also towards communication. And then there is another important thing which has been addressed, that is the power to detect. And from the EODR, and I know there have been many power calculations based on the TreeScan, but from the EODR system, what we learned is that that 20% of the drugs make up 80% of the exposure, actually. So whereas on the frequently used or the long-term or the chronically used drugs, you will soon have enough power to detect, there are many drugs that are utilized that are either short-term or very infrequently that you will not have power to detect important events potentially. So when we, there is a paper on how, is there enough leverage, also by Presi Coloma, and actually there was a simulation done if the size of that data source was increased 10 times, and the underlying source population was 30 million, so 30 million. Even then, you would not have more than 50% of the drugs that would have enough power to detect an important relative risk. So one of the conclusions there was it's not about increasing the size of similar type of data sources. What is important is to actually augment it with different type of data sources or different countries where different drug use patterns may actually be. So I think that pleads also for the global collaboration in doing this. So I've seen it, yeah. So now coming back to the methods presented, I think they are all excellent methods, and I cannot choose between one of them. I think they each, they may look at different things or different correcting for different types of biases or confounding. So I think we should not go into choosing one or evaluating which is best. I think we should move forward, especially with the computing powers that we now have, is to actually do them in parallel and make them available for signal refinement for the people that are actually deciding about this. So I think I would leave it like that. Thank you.
Thanks very much. And next is Jennifer.
Thank you. I'm excited to be here today, and I appreciate the opportunity. I've thought about these issues off and on for the last decade or so in my role as Sentinel Methods Core co-lead, as well as work before that even within the Vaccine Safety Data Link project. So I'm going to just reflect on some overall thoughts that I have, talk about some lessons that I think may be relevant from the Vaccine Safety Data Link experience, and then give a few comments on the specific methods we heard today. So we were asked to think pretty broadly. Michael suggested we even ask the question, should FDA do this at all? And I think there's pretty good consensus in the room that yes, it's a good idea. A comprehensive safety surveillance system needs to be equipped to look for, to discover things that are unexpected. And I think claims and electronic health record data have a lot to offer and can complement the VAERS and FAERS systems. And ideally, these systems are going to inform each other and work together to signals from one system can be brought in and used to help guide other systems. I think the main question that keeps coming up is at what scale? The more, the volume of data that we look at, the more pairs we look at, the less customized we can have a method be. And that leads to more false positives and false negatives. The fewer pairs we look at, the more things we might miss. And so I think regardless, with these data, and I'll agree here a lot with what Andrew was saying, these are data that are not collected for research purposes. The signal to noise ratio is relatively low, and so I think it's really important to prioritize. Otherwise, we will be overwhelmed by false positives and false negatives. And the question, I think, really is, well, how? And I would argue, I think what's important is to focus on the intersection of pairs where, one, we can make the biggest difference, prevent the worst problems, and where the data are the most fit. So quality data. And I think there's two ways to think about doing this. You know, let's build it up from zero. Let's start by figuring out which pairs those are, as opposed to start with all by all and exclude things that we think are hard. I'm not sure which way is necessarily the best to go, but I think building it up from zero is maybe where I would start. Thinking about serious outcomes, drugs that are going to be expected to have widespread use, so, you know, really important events that we want to not see happen, and instances where a particular drug is going to be used a lot, so even events that would occur rarely may turn up. I think it's also, in that vein, important to be prudent about how often we might conduct a signal detection activity, and I would argue that perhaps relatively less frequent might be better. We need to worry about having adequate power, and I think Miriam brought that up as well. We don't want to be falsely reassured by running these things and not finding things that, oh, everything's fine. I think we need to be thinking about, well, what power do we really have to see something at any given time that we looked? The other question I would step back and ask is, you know, with this signal detection activity relative to other activities that FDA undertakes, you know, what is really going to be the value added for this particular activity? And I think it's worth thinking through that very carefully, and there's been a lot of good discussion on that already. Two things have come up, or at least come to my mind. So casting a broad exploratory net so that we can feed what we find from this system into other systems that can be more rigorously evaluating what's going on. That's one piece of the puzzle, I think. And the other would be, we just want to identify big unexpected problems that we won't otherwise catch. So those are two things. Reflecting on how to deal with looking at lots of things in an exploratory way, and what can we realistically find from it. I think it was interesting. I was reassured to hear someone in a previous session say, most of what we're going to find are not the big, huge problems. Most of what we hope to be, what's out there are smaller effects, which is good. We don't want big problems, but the problem with that is these data are messy and I don't think are well suited to find small effects. Their bias is going to overwhelm a lot of that. So we need to think our way through that problem. So in terms of some lessons that I've gleaned from my experience with the Vaccine Safety Data Link, I'll talk about a few challenges and a few strategies that have been helpful. I would agree with several people who have talked about the need for a priori plans for follow-up, sort of internally, as well as transparent reporting plans that may happen externally, and putting those in context of risk and benefit and the level of evidence that's being generated. Even though I'm a statistician and I love methods, I think regarding methods, the methods are not the main issue here. The data are the issue here in terms of what we need to address. But with regard to methods, I think more approaches are better than fewer, and I don't think it serves us to restrict to a single focus, because any method that we perform will have limitations, and we can simply learn more if we apply different methods that have different limitations and advantages. I think simpler is better. We want to be able to easily communicate what we're doing. So as I alluded to, I think the biggest challenge here is more on the dealing with the data and the quality of the data, not the methods. And I think Martin Kulldorff put it very nicely. You know, if you give any method the data that are not good, if you have bad inputs, you're going to get bad outputs. And what we found within the Vaccine Safety Data Link, I think I would categorize the two biggest problems with regard to data are outcome misclassification, and along with that, the timing of outcome occurrence, as well as bias due to confounding. So either poor design or poor choice of comparator, or lack of being able to adjust for things we need to. The other challenge has been brought up in the communications section of this day, sort of the reporting of the results. We need to fairly interpret and commensurate with the quality of the level of evidence that's being generated, what we see when we conduct such an analysis. I think in the VSD, they originally started with data mining before moving towards, and signal detection, before moving towards what I would say is their most prominent methodology now, which is a targeted, proactive, sequential safety surveillance. So selection of a handful of outcomes for any given vaccine and really targeting surveillance for that. Complementing that with, well, let's add an outcome that's more nonspecific. Like in kids, you don't expect to see hospitalizations after vaccination. So kind of having a catch-all bucket for those things we don't expect in advance. So we target a small set of things that we're worried about for various reasons, and then we cover our bases with this nonspecific outcome. So that's kind of one way in which the VSD has landed on that. But in the early days, there was a lot of signal detection, very broadly casting nets and generating what they've referred to as a phone book of results, and just volumes and volumes of information. And it was honestly hard to know what to do with that information, and for a variety of reasons. And we've come a long way since that, and all the methods and a lot of the processes people are talking about today have come a long way since that. But I think there is a risk of generating enormous amounts of information that we just don't know how to synthesize or process. So we need to think carefully. I want to mention power again, because I think there is potential false reassurance that we can have due to false negatives. Miriam mentioned in many cases, and others have mentioned, even though we're dealing with big data, in the end we may have relatively small numbers of events, and so we need to think about methods that don't make large sample assumptions. We need to worry about outliers, because there may be very little information driving our results, and so those are things that haven't come up that I think we need to be worried about or think about. And then the interpretability of the results. What is the quantity in the end that we're generating upon which a decision to follow up or take some action will happen, and can we quantify that in a way that is clinically relevant? Okay, so some useful strategies that have come up. Thank you. So what I've learned, I think, is that if we don't do any tailoring for a given drug outcome pair as we apply a method, so what I mean by that is tailoring of some design choices like the outcome definition, decisions about the outcome definition, choice of comparator group, et cetera. If we just apply a single approach to all or a lot of drug outcome pairs, we're going to have a lot of inaccurate information being produced, and we're going to waste a lot of resources. However, as Andrew mentioned, we just don't have the opportunity to do full customization. We're not doing a full confirmatory or observational study. It's just not feasible with so many pairs. And so how do we find the balance? How do we find something in the middle that's a compromise? And so within the VSD, there's been recommendations for thinking about a hybrid approach. So let's have a customized, and I mentioned it already, a customized list of some things that we know we should be worried about, plus more generic groupings of sort of everything else. And that could be as simple as a single outcome that's any hospitalization in a certain number of days, or it could be much more granular depending on how much time you want to spend in that realm. I think we need to go beyond ICD-9 codes at places where we can, and Sentinel has data partners where we can go beyond ICD-9 codes, comparing lab information procedures when we know they're relevant to defining an outcome that we care about. I think to get a handle on the scale, one useful thing to consider doing is really to restrict the number of outcomes or the types of outcomes that we look at, in particular to outcomes where we think the bias may be the least. So I think what we've found for vaccines is that restricting to serious events, so inpatient events or ED, emergency department events, restricting to incident events, looking at shorter risk windows, primary diagnoses codes, avoiding things like day zero, and then in the end, looking at multiple risk windows. I think other people have brought that up as well in terms of sensitivity analysis. So there's ways we can take this all by all space and really take good first steps forward by picking outcomes or defining outcomes in such a way where we minimize these major problems that we tend to see with misclassification. I really like the ideas of pairing analyses with control outcomes. I think you can think of much more broadly than that: control outcomes, control exposures, control time windows. And I would like to think we could go farther than we have with the use of control outcomes and other control aspects beyond some sort of sensitivity analysis or gauges on amount of bias that might or might not be there. But can we use these control outcomes to really correct the bias that we may or may not be seeing in the analyses that are of interest? I think in addition to, am Am I at time? Okay, sure.
We're saying a lot of interesting things. So let's finish right now.
I have a little too much here. We can catch some of this in the discussion, and that's fine. I guess I'll end on talking about the reporting. I think that it is sometimes lost in the discussion. We need to find ways, one, to synthesize a lot of information. I really like the idea of the graphical displays that people were showing. That's a really excellent way to take a lot of data and really be able to pinpoint and see quickly what's happening. I guess I wanted to react to one comment that Andrew made that I thought was really interesting. And so we need to worry about the nature of the secondary data. And it's not possible in detection, except in broad brushes, to tailor to some of these problems that will exist. And I guess I would challenge that and say, you're going to make some investment in this. We have messy data. There's going to be an investment in figuring out what's real at some point. You could do that at the beginning and try to do more tailoring of your outcome definitions, of your confounder adjustment, et cetera, so that in the end you have less to sift through. Or you could punt that at the beginning, so to speak, and then you have a lot more time to spend in the follow-up phase. So I guess I'm in the prevention camp, but you're going to hit that problem at one end or the other. And I'll leave it at that for now.
Thank you. And Bill.
Well, thanks very much, Mark. It's a great pleasure to be here. I've learned a lot today. I think one of the things that I find most heartening is this distinction between the signal detection and then the subsequent evaluation, and finding things that we want to subsequently try to draw some causal inferences about. It's really, I'm very optimistic, kind of thinking about the future in terms of our likelihood of continuing to improve in terms of being able to draw causal inferences from observational data. And I've been asked to sort of talk a little bit about, where we might go in the future, perhaps from partly a wishful thinking sort of standpoint, but I don't think it's all wishful thinking. One of the things that we have learned a lot about, and this is, I think, is largely attributable to the work that Sentinel has done, but also kind of the parallel epidemiological work, the work that OMOP has done that Ken was talking about, is we've learned a lot about design and how to set up these pharmacoepi studies. And just as Martin kind of put out a list of best practices, there's another sort of list that's a complementary one that has to do with observational data analyses, and it goes something like this. We want to look at, when we're looking at an intervention with a particular drug, we want to look at an active comparator. We want to look at new initiators on both those products. We want to be able to control for adherence or exposure to the medications. This idea of high-dimensional balancing, whether it's with propensity score or with its inverse probability weights or similar kinds of technologies. And then avoidance of design flaws. Those can be other design flaws. They can be very subtle. So, a great example, I think, is the kind of the discordant results between the Nurses' Health Study and the Women's Health Initiative, both of which were looking at hormone replacement therapy. We had almost a decade of evidence from a very rigorous, well-done, non-randomized Nurses' Health Study that had indicated that hormone replacement therapy was protective against cardiovascular events. And then along came the Women's Health Initiative, a randomized trial, another really large randomized trial, which came to exactly the opposite conclusion. And immediately everybody said, oh, that's randomized. That's the problem with observational studies. You can't draw reliable inferences from them because it wasn't randomized. So the supposition was that there must have been kind of major unobserved confounders of some sort that were leading to this differences in conclusions. But it turned out, subsequent reanalysis of the data turned out that it wasn't that, or at least it wasn't primarily that. It was more this issue of timing.
And so we talked about sort of timelines and looking at events happening at different exposure periods. And the Nurses' Health Study, the non-randomized study, had been just looking at over the breadth of experience of the women that were in the sample. But the Women's Health Initiative was looking at a very specific 2-year risk window following the initiation on hormones replacement therapy. When the observational data from the Nurses' Health Study was reanalyzed using that same 2-year window, the results were extremely similar. They were virtually identical. So that's a design problem. And of course, the Women's Health Initiative, which gave us this randomized trial result, gave us something to point at. It highlighted this difference in the exposure window. And so when we reanalyzed the observational data, we were able to get to the same answer. The challenge is that with observational data, we never know for sure whether we've got it right, you know, and whether there's something that we're missing. So that's a challenge. Another reason I think, though, that we're going to do better going forward in terms of causal inference, aside from design, which I think is primarily the biggest thing is the design, is the data. And we're getting, if you think just in the United States about the effects that the Affordable Care Act had, or more correctly, the HITECH Act around health reform in the U.S., which mandated meaningful use of electronic health record data. That created just a massive electronic health record data infrastructure in the U.S. that didn't exist prior to the passage of that legislation. Almost all the medical record data that we had in the U.S. was paper, with the exception of hospitals and certain kinds of clinics and so forth. But now we've got this ability to be able, from a technical standpoint, to be able to link electronic health record data with medical claims data, which heretofore has been kind of the bulk of the data that we've been analyzing in Sentinel. As you think about sort of the ability to measure clinical outcomes directly with medical record data, which we typically don't have in claims, the ability to measure severity, have variables we don't have in claims data like blood pressures and body mass index and hemoglobin A1C levels and lipid panels and things like this. I think that's going to help us to control for unmeasured confounders much better than we have been able to do in the past. And it extends beyond that. You can link to social demographic data from market research companies, and you can link to data from area health resource file at the county level, which has about 6,000 variables in it, that has all kinds of information, everything from environmental exposures to information on hospital beds, numbers of doctors in that county of different specialty and so forth, the kind of measures of healthcare supply in particular that may be extremely important, and very good measures of race and ethnicity, employment status, income, and so forth that come in from the decennial census. We're going to have data on device feeds, continuous information on device feeds, and at some point, the ability to link to genomics information. Whether we do this as kind of a massive database that has everything in it, or whether it's done on a study-specific basis, that ability to link data, we don't have to send around protected health information to do this anymore. There are salting and hashing methods where the data holders have the algorithm. They pass the algorithm through, and it will take information like social security numbers and names and addresses and dates of birth and so forth, so encrypt all that information, and you can link on encrypted IDs. So there are ways to have privacy-preserving linkage that will improve our ability to enhance the richness of the data we're working with. One thing that I worry about, because I'm coming at this more from the perspective of an economist rather than an epidemiologist, is our tendency to think of these data as sort of being patient data only, or patient behavior data, or clinical patient response. And it is certainly partly that, but when you think about what sits in these administrative data systems, it's almost all generated by providers. So the diagnosis codes, the procedures codes, the tests that are done, and so forth, this is all coming from providers. The patient isn't coming up with that stuff. So if you think like an economist, you say, oh, the patient recommends that it represents the demand side here, the demand for healthcare services, and we've got on the physician side or the provider side, we've got healthcare supply. And that means that if we interpret this data as being treatment effects that are being driven by patient clinical characteristics only, we're likely to get biased estimates. So economists think about this in the context of identification. What kind of information do we need to be able to identify the patient characteristics, or the patient effects, and what information do we need in order to identify the physician effects? And basically the solution is to bring in more data. And it's paradoxical because the information you need to identify the patient effects is more information about the physicians, and vice versa. In order to identify the physician effects, you need more information about the patients. The other thing I just wanted to make one brief comment on was around the potential for machine learning. Michael mentioned earlier in the first session around some work that's already been done using Lasso and ElasticNet that suggested there were some incremental improvements using machine learning. I suspect that will get better as these data linkages come in and the data content feeding the models improves. But machine learning methods are not causal. They're classification and prediction methods. They're very useful, I think, in the context of the way we've been thinking about signal detection for identifying things that may be correlated with safety events or signals of safety events. But ultimately, we'd want to take those results that come out of that machine learning kind of approach and apply our traditional causal modeling framework to them. And one of the beauties, though, I think, of the machine learning is that it doesn't require us to do all the data mapping and all the pre-specification of what the variables are. So, which keeps us kind of locked within our traditional theoretical framework. It's hard to sort of discover new things when the only things you test are things you already know from the literature. And so I think that's the beauty. The machine learning, by having this ability to trowel through these large linked data sets that may be very messy in terms of the completeness of the data, they can be very helpful for hypothesis generation. And then finally, just some of the more recent work in this area is starting to bring our causal modeling frameworks that we've been working with for the last decade or so and machine learning together, and in particular targeted maximum likelihood methods, where the models are taking account of risk exposure and outcomes and modeling both of those pieces for the intervention, for the comparator, but doing the treatment evaluation on the basis of the predicted values for those things. So rather than trying to pull parameters out of the models. And that allows the machine learning methods to be able to use just a whole variety of different specifications. You can use support vector machines and random forest and lasso models and so forth, and aggregate all those up and use that as the prediction. So a little bit into the future, but I think that we do have, we're making a lot of progress in terms of our framing of observational data analysis from a design standpoint, from a data standpoint, and from a method standpoint. Thanks.
Well, Bill, I appreciate the last opening comments from the panel focusing on a pretty optimistic view of the future and what may be possible down the road. I want to start off this discussion before opening it up to those of you who are here with a few of my takeaways from hearing from all of you. And the first is just picking up on Bill's point, there seems to be a lot of consensus that moving in this direction, building out Sentinel capabilities related to signal detection is an approach whose time has come, and that's thanks to the considerable progress and achievements of the kinds of methods that we heard about in the last session and that you all are very involved in applying already in many cases. I did not hear a specific call out in favor of one particular kind of high-dimensional analytic approach at the signal identification level, rather quite the opposite, the importance of, given that these algorithms have been refined, the importance of continuing to develop, apply all of them, or a range of them as this work moves forward. Maybe there's some fit-for-purpose issues that will emerge there, but right now I didn't see a clear call for one approach versus another. I did again hear the importance of paying attention to the underlying data quality for these signal detection activities. And I guess the other common theme from this panel was in one way or another, the importance of not thinking of signal detection in isolation from a signal refinement strategy and communication strategy and so forth, as we've talked about today. Where there may have been some differences, though, was around how exactly to integrate the two, whether, as I think Jennifer was saying, well, maybe we should focus on a narrower set of drug and outcome pairs to begin with, where we've got some pretty good priors or reasons to think this is the right place to focus, versus what I think was maybe implicit in some of the other comments, maybe having a broader net, but also a well-specified set of thresholds or criteria or a dashboard that could maybe automate even some of the signal refinement activities in conjunction with doing signal detection to, again, help focus efforts where they're really likely to pay off. So let me stop there and see if there are any reactions to my quick summary effort, as well as other points that you all might like to add based on what you've heard from the other panelists. Again, with this very practical focus on, okay, where does Sentinel go next in terms of integrating a signal identification approach. Any further thoughts or comments?
Thanks, Mark. I agree with your summary. I just wanted to kind of pursue a bit the issue of using a suite of methods for the alert stage prior to the review and consideration of next steps, because I think we've talked about it, but it's challenging in the sense of if multiple methods give different findings and we start seeing discordance, or we see discordance across 3 out of 4 databases, so an alert in 3 of the databases but not in the 4th, it becomes it's easy, I think, to oversimplify and think, okay, it's only an alert if it always stands out, or vice versa, and it will depend on the nature of the exposure and the outcome. Why, for example, we may say, well, if it stands out in that particular database, then it's alert, but if it isn't standing out in that database, well, it doesn't mean so much. So there's an element of the nature of the data we're looking at, the methods, the nature of the outcome, the nature of the exposure, and maybe even changes over time. So I do absolutely think it's the right track, this thinking around suites of methods, but also trying to think about how we're going to weigh them. And Miriam was talking a bit about this, but a priori, how are we going to think about weighing the difference, if you like, evidence streams into our thinking at the follow-up stage? The challenge being, the more we plan a priori, these are the areas we're going to focus on, maybe we run the risk of missing the truly unexpected, because we're putting too much in terms of our prior assumptions. So no good answer to it, but I think it's really challenging how we combine those elements.
I would like to respond a bit on what was tried before. So if we talk about like orthogonal evidence or different type of evidence, for example, I think all the methods like TreeScan and the temporal discovery patterns, etc., they all run on generating an estimate of risk from the same underlying data sources. But there are other type of data sources, such as the literature, where you find already evidence, labels. Then there is biological plausibility, and these are all like type of independent sources of evidence that you can combine. And the way this has been thought about before was to combine them in like a Dempster-Shafer model, where you can combine independent pieces of evidence. So this is the approach that was taken then by EUDR, where you could actually tune the you know, whether you would favor one more than the other. So I do agree with you that you cannot pre-specify, you know, a priori everything where you will be looking at, but it needs to be flexible. But I think the point I wanted to make is that it's important, if you do so, that you have all of these evidence next to each other rather than sequentially. I think that will gain, then we will gain a lot of time.
And I would like to ask anyone who does have an additional comment, question, or idea to come forward. Ideas related to signal identification. This is signal identification meeting in two senses of the word. One, it's signal identification in the Sentinel system, but it's also signal identification in terms of ideas, issues that FDA and all of us should be making sure get on the table as this work moves forward. So start over here, and again, please identify who you are when you ask your question.
As I listened today, I was wondering, you know, what are we trying to solve for by signal detection, and what are we trying to achieve in terms of signal detection? And I think, you know, for me, you know, I think the synopsis and the discussion by Jennifer Nelson did really a great job of summarizing the issues around detection of signals. And what are we trying to solve for is really embedded in that question. So thinking about that, I think she said some very important things in terms of the system is not trying to identify small effects. It's really to try to find out the big things that we don't want to miss, because historically there have been some big serious safety issues that were either found late and there was a lot of harm. There was a period of the time in which it could possibly have been detected but was not detected. So if you think about what we're trying to solve for and what would be a pragmatic way of thinking about detecting signals that are high magnitude, big effects, that are public health, that have a big public consequence, because the small effects are going to be messy, right? So given that we all understand that data quality is important, and I think that's a given. I think the other things that Jennifer said, which was very important, was that sort of epidemiology 101, which is multiple methods, multiple approaches, different data sources. These are things that we need to do in order to give us greater assurance. So what I would like to ask the panel is, what does good look like for signal detection? What are your views on that?
Yeah, really good question. If you all have any additional thoughts, I think you did a nice job of summarizing some of Jennifer's points. Several of you came at this issue of how this signal identification kind of strategy can best complement and augment FAIRS and the traditional long-term approach. Miriam, you talked about maybe better for identifying adverse events that are adverse outcomes that occur normally or in the course of treatment that might not be as likely to be reported. But really appreciate any other thoughts, further thoughts in relation to this question.
I think the obvious one is the denominator and the background rates, and it's easy, I think, to see spontaneous event reports and see what looks like a scary number of reports coming in, and think that you may have an issue, but you really don't know unless you know what the background rates for that particular patient population are. And I think, personally for me, I think that's one of the greatest contributions that the Mini-Sentinel and Sentinel system has provided, is this ability to have a denominator and be able to estimate background rates so you can know whether or not the signal that you're seeing is differentially higher than the background rate.
Just to add, Solomon, excellent question, and that's one of the reasons why we had set our pre-specified threshold to greater than 2, which OMOP and others have also done various projects and investigation in comparison and performance of methods. And really, when you're using observational data, it is ideal for finding large effect size. Now, can the methods be more refined to lower that threshold? A lot of the activities that we're doing in disproportionality analysis with spontaneous data, the effect size of about 2 is also used. For many of the actual hypothesis testing studies, they normally try to identify effect size of 2 or greater as well too. So going below 2, I think is more research need to be conducted and evaluate how we can leverage and enhance the methodology to look at more refined things, 1.8, 1.6. I mean, those are some of the activities as well. But other thing also, it's important too, as you mentioned, Mark, supplement, augmentation. It's to be supplemental. You should look at all the available data, not just the output that comes from signal detection. You have to use your clinical judgment. That is very, very important to put those drug event combinations that are above the threshold into context. What are we seeing with all the other available data as well, from literature to spontaneous cases, clinical trials, so all of that. That's very important, not to use this activity in isolation.
Thank you. Please.
Hi, Martin Schumi, Janssen Research and Development. I'd like to respond to Bill's comment on the use of machine learning in observational data. And what I'd like to point out that I didn't have time to discuss in my presentation is that we actually used a lasso to fit the propensity models. So each and every one of those tens of thousands comparisons that you saw, we used tens and thousands of variables derived from the observational data. And the reason why I think that is important to bring up here is that even though you're running these analyses at a very large scale, that doesn't mean that you can also use a large amount of the data to correct for confounding. And in fact, in that study, we used negative and positive controls to show that we achieved that correction for confounding. So just wanted to point that out.
Thank you. Any further comments on that? Good point.
No further comment.
Good point. Diana?
Diana Zuckerman, National Center for Health Research. I just wanted to follow up on the pre-specification issue because I think that's so important, and actually use the hormone replacement therapy study as an example, where the point you were making about what kind of study design was interpreted as the reason for the different results. But that wasn't the only reason. I mean, there were a lot of changes made about how are we looking at outcome, and are we only looking at women taking hormone therapy for X number of years or a longer number of years? Did they start premenopausal or postmenopausal? And actually, when NIH reanalyzed their data, they held firm on what the results were, but other people who reanalyzed the data had a different interpretation. So it's always complicated depending on whose perspective and which variables you're looking at, and I think that's a good example of all the issues we're talking about today and the importance of, as much as possible, deciding ahead of time what it is you're going to look at, but keeping your mind open for possibly looking at other things later.
Yeah, I think that's a great comment. That's why I sort of couched it as in terms of one of the many factors was this exposure window. But I think we have there's a literature that exists that has tried to compare observational studies to studies from clinical trials, and with clinical trials being our gold standard, of course. And contrary to what I think most of us might think, or what the general public would think, we would think there would be a large degree of discordance in those results. But that's not actually the case. That observational studies in general yield average treatment effects that are very similar to those of clinical trials. And that's even without attempting to mimic the design of the trial. without trying to find the inclusion-exclusion criteria, the same sort of patient populations that were studied in the trial. So I think that we should feel like there is great value to observational data analysis in terms of being able to assess treatment effects, but we should be always cautious of what we might be missing, because we can certainly be wrong with observational data, but it's a pursuit worth going after, I think.
Yeah, I think the point of what to pre-specify versus what to leave open to explore is really well taken, and it's tricky. We're not conducting a randomized trial where everything should be pre-specified. In fact, I would argue, I think we're on the opposite end of the spectrum. We are exploring. That said, well, so I like the idea of exploring risk windows because we don't often know when we expect to see some of these events. I like the idea of exploring different comparators, different designs, because there's never, in these databases in particular, one perfect comparator group. What I like pre-specifying is sort of goes back to the point that another person made, is pre-specify the scale of what we want to do. What's the main purpose of this activity? Pre-specify those outcome definitions as robustly as you can, and pre-specify the quantity that you're going to generate a next step on. And this was talked about earlier in the day too, thinking about relative measures or attributable risk measures. So defining whatever that is going to be in a clinically meaningful way. And I think I would agree with those who would suggest that the absolute risk is probably, well, they're different measures, going to bring something different to the table, but that's the thing that's going to translate to impact. And I think so that would be an important piece to pre-specify.
I think it's, I agree absolutely with the comments, and I think it really emphasizes also the importance of transparency, that we're really trying to be as explicit as possible about what we've defined a priori, what impact we think that's going to have, and then look at it after the analyses. And I think that kind of links back to the previous comment from Martijn Schumi about methods and use of LeSous, because I think that we can gain a lot from more complex methods, but we have to be mindful that as far as possible we don't want to introduce unnecessary complexity in our choice of methods, because that may mean that our alerts are less easy to understand what happened. And I'm thinking of the work we've done in our pilot work, where a really big challenge is, what do you do when you see it there? Do you really understand what the implications were that led to it being highlighted? And the reason why I mentioned the LeSous is we did some work, it was in FAERS or FAERS-like data, Vigibase, 10 years ago now, using the LeSous, comparing it to the more standard methods like GPS, and the information component used at Vigibase. And it was really interesting, the results, because we assumed that it would outperform the standard disproportionality measures that are used because of the fact it was allowing for co-reporting of drugs. But we found it was marginal in practice, and actually we found the results harder to interpret than the GPS type outputs, so that at that point, and things have progressed since then, but at that point we felt that the added complexity that it provided in spontaneous report couldn't outweigh the fact that it slightly performed better. So I think we have to be wary of this trade-off between complexity and whether we really gain extra performance from it, and really have a kind of laser focus on transparency of our outputs and how they rely on the way the data is collected and the assumptions around data collection and structuring.
And efficiency, great, thank you. I think we may have two more comments that we'll try to get in before we wrap up. So Bill, over to you.
Okay, yeah, I just wanted to introduce, I guess, another layer of complexity. Something that hasn't been discussed here, but I know is always a worry, is whether a given drug-event combination that never used to appear suddenly appears in higher frequency. And so the question is whether you use all the data or whether you just try to focus on something recent that seems to be bad. There are about 3 reasons why you might get a change, even though a molecule is a molecule is a molecule. One would be certain drugs or vaccines, especially biologicals, are kind of hard to produce, and you could have a batch effect in which things can just go wrong, and we saw that a few years ago with heparin, where there were many deaths with a supposedly safe drug. A second reason could be the drug was recently approved for a new indication, and suddenly it's exposed to a different population, and that could cause a change in that drug event combination. And the third could be, although the drug's been on the market a long time, a new drug has just been introduced, and the drug interacts with the new drug. And so in all these cases, it would kind of be important in these surveillance idea to have some other analysis that tries to detect if something bad is happening just in the last year.
Mark, just a quick comment. I had one of the things that's worth thinking about, I think, is that roughly 80% of the data we have is unstructured. And that's particularly true with electronic medical record data. And try as we might to extract useful information from medical text, it's turned out to be extremely difficult to do. In this machine learning world, in the last 5 years, there's been a dramatic improvement in one specific type of machine learning, which is kind of the one that undergirds the artificial intelligence work, and that's deep learning. What deep learning does is you don't pre-map everything. You let the computer find the mappings. And so for the kinds of relationships like we were talking about with TreeScan, where you had these terms that were similar to one another, but they were at the same level, deep learning would discover those and figure all of that out. It's been made possible by the gaming industry and the development of graphical processing units, where this is kind of jumped exponentially in terms of the improvement of those methods. Now, the bad thing about them is that they are totally opaque. So you really, from the standpoint of being able to talk about and convey these models and what they're finding to doctors and to patients and so forth, they are the ultimate black box. But work that's been done recently by Google and UCSF and University of Chicago, using UCSF and University of Chicago medical record systems, has used deep learning in predicting hospitalization and hospital discharge just on the unstructured text. And those models have been shown to be superior to anything that's been published previously. So it's not that it's a panacea. It took hundreds of thousands of computation, like GPU hours, for just the thing to run. But it is a sort of a new world in terms of trying, I think as we go forward, a lot of what we're going to have to learn to do is to work with unstructured medical record data, particularly from electronic medical record systems, and that may be a path forward in that area.
Got time for, very briefly, and then one final quick comment. Yeah.
No, I would just like to come back to the comment made by Bill, I think, which was about the impact of time and time trends, and I think it cannot be underestimated. So I think it's one of the refinement steps. I think what needs to be done is that within database you look actually at time trends, and because it's showing healthcare patterns, and I think that's just a part of consistency that needs to be checked as a routine.
Thank you.
Yeah, I have I'm Bill Wang from Merck. I have one general comment, but two quick questions. First, I want to applaud our FD colleague for really setting up a great framework and think about the detection, the refinement, the evaluation, the communications, but also think about the quality, precision, and statistical validity. I think that's a great framework. I was really impressed by the discussion today. Two questions. Regarding the statistical method, we talk a lot about the detection method, but in a way you have to think about this as a whole, right? from the detection to the refinement to the evaluation, because it's data continues to flow, continues to collect. So the question there is the method there, the thinking of the method, not only cover the detection, but also more fine-tune the evaluation, answer a specific subgroup question, but using a continuous thinking of methodology. So that's one question. But another question is, we talk about orthogonicity today, or different source or replicability. So I've been thinking there are efforts going on with US FDA, but sounds like there are also efforts going on with the European agency, with Japan's. Is there a collaboration or international collaboration that we need to think about where we can actually help each other? And because the communication is a global impact, global communication.
Really good question to end on. Many of you brought up the similarities and the potential complementarities of and benefits of sharing data across countries. So how much of this effort should be U.S. focused? How much of it can benefit from looking globally, but very briefly? Miriam, do you want to?
Yes. Yes, I would like to comment. I think in general there should be global collaboration because it's not confined to one continent, so I totally agree. Now, the reality being that the US is ways ahead of Europe at this moment, because whereas Europe has some, initial funding to develop methods, I mean, EMA does not have a system in place as Sentinel, unfortunately. Because there's no structural funding to do this. So there is a network of centers, but there's no money for it. So it's all based on these, finite projects that then initiate and then end, actually. So I think there should be a way to collaborate. I'm not sure yet how that will be realized in Europe. There is now an IMI project that will focus on converting European data sources into the OMOP common data model. Whether that's going, it's just started. So whether that's how that is going to be of help here, we just have to learn it, I think, over the coming years. But it should be done. But Europe is lagging behind.
Very quick. Yes, absolutely agree with Miriam's comments about the importance of it. Internationally, yes, definitely. We need to work together in terms of definitions, reference sets, good practice in terms of clinical ontologies, and I think there's lots of learnings we've heard throughout the day around how not to do this and best good practice approaches. So yes, absolutely, international collaboration will be very useful.
Great. I think a nice note to end on. Now, in the interest of time, I'm going to do my thanking comments for this panel in the same context of the thanking comments for the wrapping up the rest of this event. So if you all just bear with me for 1 minute. This has been quite a day, to quote Diana Zuckerman. It is complicated. However, there has been a tremendous amount of progress, and I think a lot of good ideas discussed here on how FDA can go forward with implementing signal identification capacities in the Sentinel system. A lot of this is happening already, and some great discussion about both the challenges, the trade-offs, and the growing opportunities and value for incorporating this. Back in my days as FDA commissioner, I did not get to attend briefings very often on important methodologic issues like this one. I always loved doing them, though, not just because it was a cool change from dealing with the latest public health crisis or what have you, but because these are the kinds of ideas and insights that are going to develop better evidence, and this better evidence is going to transform healthcare and health. So I want to thank everyone who's contributed today for pushing forward on the next frontier for the Sentinel initiative. I believe FDA is planning on moving forward with developing pilots in this area, as you heard earlier. There will be more activities in this area, so stay tuned. The slides and materials from this event are going to be available on our website at Duke Margolis by later this week. In addition, we are going to have a significant part of our upcoming Sentinel annual public workshop meeting in the spring devoted to next steps on some of these issues as well. and definitely more coming from FDA too. So thank you all for this important step in moving forward on signal identification. In particular, I want to thank all of our speakers today, including those on this panel up here with me. Thank you all very much. And this would not have been possible without a lot of hard work by FDA. I think you saw that reflected in the presentations made by FDA today. So Mark Levinson, Simone Pinheiro, Monica Munoz, Daniela Stojanovic, all of you, thanks very much. But a special thanks to Michael Nguyen, who put a tremendous amount of time and effort into pulling us all together and enabling some further progress at FDA. So thanks very much to our FDA colleagues. And last but not least, thanks to the Duke Margolis team, Greg Daniel, Morgan Romine, Elizabeth Murphy, Nicholas Harrison, and especially Adam Atten for a lot of work to get this together. Thank you all too, and all of you, safe travels, and look forward to next steps on this very important topic. Thank you very much.